might be the one to come up with a vaccine against COVID-19, but we also are set up to possibly tackle future pandemic and be prepared for it, and then tackle other major diseases, because if you think about cancer and heart disease, they're basically a pandemic that people have got complacent about, right? Because cancer and heart disease and Alzheimer's and various other diseases tend to impact a lot of people yearly. So this is what we're using, the same model of open science, and the people who have joined us from all around the world, the scientists, we're going to further tackle things like that. And we've done a pretty good amount of work in 4 months, impressive amount of work, that being, as you will hear from various people in our group, in our lightning talks, the stories that are emerging. So just a brief, description of Covert. You can see the growth trend we have. Anyone can join us who's interested. We're very open for more collaborations. In Covert, we have many subgroups, as you see here. This is sort of our work, how we work in Covert. And as you see, these many subgroups, and we keep forming new subgroups with people joining depending on their interests. And the whole goal is publications, and we're throwing symposiums, and then eventually, hopefully, maybe ultra public policy decisions, and come up with vaccines and so on. And we're also closely collaborating with other COVID groups like N3C, which is the National COVID Cohort Collaborative, which Melissa Handel, who's the director of it, is going to be giving a talk later in our symposium. So you'll hear that. And we're also very keen on collaborating with other COVID groups. We've done a lot of activity and achievement for the past 4 months. There's a lot of papers. You can see one is already submitted, a lot more to be submitted in this 4 weeks. We're already testing some potential therapeutics with our members in SARS-CoV-2 in vitro models and animal experiments, and we've done a lot to actually promote. And the big announcement we have is now we have become an official nonprofit. This is especially due and thanks to Wiley Law Firm, where after our first symposium contacted us with the help of Brian and Shimola, our signature science, and special thanks to him also. And with Wiley Law, they were very kind, and they did pro bono work to help us set us up to become a nonprofit just last week. So we couldn't have done it without Wiley, and we really appreciate their help and their continuing help that have been able to allow us to be where we are. So this leaves us also to have maybe potential funding from donations of anyone here or the audience that wants to potentially knows anyone who wants to contribute to COVID research. Any amount of donation will help. We're going to be setting up crowdfunding sources, so keep an eye out for that. And if you're interested, please contact me or Todd, and you can find our contact information on our website. So keep that in mind, because all this, for people who donate money to Covert, all the money will go basically to COVID research in our group. And of course, we could have done with all our support from all the people who have been supporting us, which is all these people here. So I'm going to pass this along to Todd. So Todd, if you want to take it, I'll stop sharing my screen.
Yep, thanks, Efshin. So yeah, I'm going to highlight some of the just attendance numbers. I won't repeat what we just stated, but very excited to be here today with everyone. Thank you all for joining, and thanks to all the speakers and organizers. So I'm an assistant professor at Rice University in the Computer Science Department in Houston, Texas, and my research focuses on primarily developing computational tools for large-scale genome alignment and comparison. I want to highlight a couple things here, symposium highlights and acknowledgments primarily. We are nearly 700 attendees as of a few minutes ago, which is really exciting. We span 20 countries, 150 plus institutions, and 34 US states attending this event today, which is again, really, really exciting. We have 23 speakers covering 15 plus topics. So I'm just, again, very enthusiastic and looking forward to listening to all these great talks. And for acknowledgements, I just want to do a brief acknowledgement here, and we'll do more as we go on. But of course, thanks to Efshin Beheshti for co-lead of Covert and everything he's done. Eliza Uzi for all the admin support and all the great work she's done in putting this event together, and all the behind the scenes stuff that goes on. Of course, the Rice Computer Science Department and Ken Kennedy Institute for co-sponsoring the event. The provost at Rice, Provost Reginald DeRoche, just really appreciate the fact he'll be giving closing comments today of the event. And then, of course, a postdoctoral scientist in my group, Dr. Leo Elworth. He's today's moderator and a co-organizer of the event today. So just wanted to highlight and acknowledge a few things. And then with this, just very briefly in the last minute I have, just again, globally, we pretty good representation around the globe, 20 countries represented today, primarily from the US, but then we have good presence in India, Brazil, Argentina, Portugal, and Germany. Across the US, again, 34 states represented, primarily the highest attendance from Texas, California, and Maryland, but good attendance across the US. I'm gonna break down by city, a lot of attendees from Houston, but overall across the US, again, just kind of spread out. First symposium breakdown, we had 60% academia, 31% others, which included primarily industry, and 9% government. We haven't crunched all of the final numbers yet as registrations were flying in the last 3 hours, but the updated numbers look like we have a much more even spread across government, academia, and industry. So really excited to see that. Huge presence across the government today, including FDA and many others, DOD, DHS, colleagues, former colleagues at MBFAC. Really excited to see that, the group of folks we have here today. And with that, I'll hand it over to Leo. Just really excited. We have an excellent group of speakers today, covering a broad range of topics. And then with that, I'll just hand it over to our moderator for today, Dr. Leo Elworth. And with that, just can't wait to listen to all the talks. And thanks again. And I'll get a chance to present some of my research later on in the day.
Thank you so much, Afshin and Todd. I have to give a few thanks, and I have to go over a couple of logistic items, but in the interest of time, I'm going to just do it a little bit later so we can get the ball rolling here. So Dr. Brinkman, are you ready to share your screen, and we'll go ahead and start getting you set up here?
I am.
Okay. Hi. Yep. So our first speaker today is going to be Dr. Fiona Brinkman. Dr. Brinkman is a distinguished professor in bioinformatics and genomics at Simon Fraser University in British Columbia, Canada. She's most known for R&D of widely used microbial genomic software and databases, as well as an immune database and platform. She's a fellow of the Royal Society of Canada, Thomson Reuters highly cited researcher, and is on several committees and boards, including chairing the Scientific Advisory Board of the European Nucleotide Archive. Finally, she's one of the leads of the new Canadian COVID-19 Genomics Network, which she's going to be speaking about today. Welcome, Dr. Brinkman.
Thank you. My pleasure to be here. I have to say this looks like a very exciting group. I'm going to first, basically as the first speaker, I'm going to talk fairly generally as it's an interdisciplinary audience, give some general info, peppered with some new data we've learned from analysis of Canadian genomics data. So, hello, bonjour. It's my pleasure to provide this overview of this new Canadian COVID-19 Genomics Network, otherwise known as CANCAGEN or CANGICO en français. So this initiative has been possible thanks to, oh, wait, 1 second. Thanks to a $40 million federal investment in Canada involving 20 million going to genome sequencing for SARS-CoV-2 virus and 20 million for human host genome sequencing from COVID-19 patients. The goal of this project is really primarily centered around coordination of efforts and capacity building to better respond to the pandemic. And specific goals include, coordinating and funding SARS-CoV-2 and human host genome sequencing efforts up to 150,000 viral sequences and 10,000 human genome sequences. Then there is also, we want to better integrate genomic data with clinical and epidemiological data, facilitate sharing of data more nationally in addition to internationally, it really expanding on current efforts, essentially capacity building for this pandemic and for future outbreak pandemic preparedness. I'd actually like to do acknowledgements early. I want to acknowledge the leadership of Genome Canada, one of our primary funding bodies for genomics in Canada, that really allowed this to happen, along with these key implementation steering committee members that are basically spearheading this, led by Terry Snutch on the viral side and Naveed Aziz on the human side. As a microbiologist, I'm getting used to being referred to as non-human right now. This is a very large, diverse group of participants involving a consortium of national and public health labs, with on the viral side, Gary von Domsler and the National Microbiology Labs playing a key role in Canada. along with Will Sow and the provincial lab spearheading and filling in gaps in our sort of other data collection, the genomic, the epidemiological clinical data, which in genomics we tend to refer to as metadata. So the hospitals, universities, research institutes, large-scale genome sequencing centers, and industry are also playing key roles with support of Genome Canada. So why are we doing this? So, well, primarily it's about coordination. More coordination with viral sequencing and associated clinical epidemiological data collection between provinces, because in Canada, healthcare is a provincial jurisdiction, so there is a tendency to have the provinces work more as silos, and we really wanted to break down those barriers. We can also capitalize on Canadian past leadership in some human genomics areas, studying the role of human genetics in the outcome of COVID-19. And this can include specifically tailoring this to Canadians and our genetics and metadata to complement studies spearheaded by other countries. While this is very focused on Canadian data, we will further align, or we already are aligning with global sequencing efforts, including COG UK and Spheres in the USA. A critical component, though, as I mentioned, is really capacity building, both now and for the future. So we're, developing this for COVID-19, but obviously want to make sure we can handle other potential outbreaks as well. So really, while we're doing this is because of all these benefits this data provides, but I'd like to spend most of the rest of the talk reviewing some of the examples of insights gained, or sorry, I'm saying, there's a lot of benefits, I guess, to this data, but really what I want to do is talk about some of these benefits with some example insights gained to date. So why sequence SARS-CoV-2 genomes? So, well, a key benefit, of course, is being able to better track or confirm transmission trends, capitalizing on the lineages of the virus that develop, that can allow us to better track spread, not just internationally, but also regionally, provincially, nationally. So you can see here the first viral genome collected from an individual in Wuhan, China in late December 2019. So for those not familiar, this is the sequence serves as a reference sequence. It isn't very big, only 30,000 nucleotides, whereas our genome is about 3 billion base pairs. And the S denotes the spike protein that you always see in pictures everywhere of this little spiky picture of a virus that binds to the ACE2 receptor in our human ACE2 receptor in the lungs, and is sort of a major component that is studied. Over time, this virus transmits through the population and can mutate. For example, here's an isolate collected in Germany in late January that acquired 2 mutations relative to the original Wuhan 1 isolate, and we can use these mutations to generate a family tree of sorts of relatedness between different isolates. So over time, this virus spreads and diversifies. Diversity and location information, along with other metadata, sort of inform the outbreak. For example, you can even just see visually in this tree here, or just this this image here shown from the GSAID resource, how many of the North American isolates have been sampled that came from Europe, and how in Canada we clearly got isolates from Europe, from Asia, US, Middle East, and even you'll see a little line going here from Australia. So this is another tree, just to show an example of data. This time in the US is colored yellow, and in Canadian isolates are colored orange. So the since in the global tree, the colors are sort of too similarly red for global viewing. But early analyses really emphasized how there were multiple introductions of Canadian isolates into Canada, predominantly from the US. But as you can see from this tree, we also gave back to the US, so to speak, with some isolates you can see are nestled within a bunch of Canadian isolates here. There's a US isolate here and here that suggests these isolates are of Canadian origin. And so this information has been really key to help confirm what public health measures should be taken. For example, we saw here in BC, the virus was being introduced from the US and Europe and other locations multiple times. And this aided policy development regarding border closures. So this also can be used to check if the isolate's genome actually reflects travel history or not. This is not always the case and can be very useful information, but I'd really like to emphasize the importance of not just looking at the genomic data and saying, oh, this isolate is from this location based on the sequence. You really do have to look at the whole collection of data, the clinical data, the epidemiological data, in particular epidemiological, combined with the sequence. This is another tool basically for doing these analyses. So basically what's useful though is this can help inform contact tracing efforts, identification of clusters of cases, and in some cases, the genome sequences matches the individual travel history. In some cases it doesn't, and actually was helpful in identifying other aspects of transmission that was occurring just from where that person who got the isolate from somebody else, where that person probably got the isolate. So why we sequence SARS-CoV-2 genomes, there's other, of course, benefits as well, not just transmission and cluster investigation. There's also this, looking at evolving characteristics that might impact detection methods, PCR and serology, clinical outcomes like strain severity and transmission and the effectiveness of potential drug and vaccines. But there's good news on this front. In Canada, we have confirmed what others have seen, that this coronavirus genome mutates relatively slowly, about 1 mutation every 2 weeks. And this provides hope that vaccines developed, will continue to be effective. This is not a fast, rapidly evolving coronavirus. Evolutionary modeling by researchers such as Sally Otto at UBC in Vancouver also suggests that there's currently selective pressure for the virus to not evolve to cause more severe disease. However, we are putting selective pressure on it to be more transmissible. And there are variants being highly studied at this time that have some evidence they might be more transmissible, like the D64 to G64 variant, some may be familiar with, that's being pretty intensively studied, that involves a change in the spike protein on the outside of the virus. And there is some evidence of in the lab, but it's important to realize that a couple of things. One is the lab data, you really have to sort of couple that also with looking at the epi data, because sometimes what happens in the lab isn't quite exactly what is happening in the population. Also, you've got to watch out for things like founder effects, where you can have something that looks like it's taking over something, but it's just by chance that particular virus got lucky. It went to basically a party where there was 50 people and it spread or whatever. So it's non-trivial to analyze these things. But one thing we can do is ID these regions of sequence frequently changing or not changing at all to inform this drug target vaccine development and drug vaccine effectiveness. So up till now, I've been talking about the VirusSeq project, looking at the viral genome sequencing. In my last few slides, I'd like to bring up the HostSeq project, and then some of the biggest challenges we face in this Cancogen initiative. So why are we sequencing host genomes? Well, basically, COVID-19 does display significant inter-individual clinical variability, ranging from silent infection to lethal disease. We need to characterize the role of human genetics in determining the clinical response, in addition to the other data that we have. The hope is, that we can use this to aid delivering new biomarkers that predict people at highest risk, because we do know there's these very unexpected cases where somebody gets a very severe disease that wasn't expected based on their age, rare idiopathic severe disease in the young, or rare unexplained even resistance to infection in people who are older who have comorbidities. So we really want to look at this more closely, also development of precision therapies as well. So the way this project is going with the viral sequencing, it's very public health lab linked sequencing samples. In this case, this is based on sub-projects with research ethics board consent, and then they collect the specimens, do sequencing, and then the data is returned with controlled access for future studies. So it's very much a reviewing project by project, certain hypotheses and studies being proposed that warrant human genome sequencing with ethics approval. So we have a number of partners that we're engaging in all this, with some examples shown here, including both, just building national networks more than we've had in the past, focused on this particular virus. And then, getting together with international partners, primarily around the area of what we call metadata harmonization and organization. So we're really just initiating this project, getting things going, ramping up sequencing. I'd really like to emphasize the biggest challenge I think we've had is to date is dealing with the data harmonization and data sharing across jurisdictions. Never underestimate the impact of such challenges, which involves, a real combination of, different sciences, including social sciences challenges with how to encourage data sharing. I'm a big proponent of open science and open data, so it's great to be on this at this meeting. But though challenging, it's also gratifying as we address these issues also for the future. We're really trying to get this right, ideally building this Canadian pathogen genomics network that could also have applications in other areas, in agri-foods, in forestry, both for pathogen and host. And really setting this up for a model for data sharing across jurisdictions within a country, which is addressing some issues that might be relevant for sharing across countries. So in closing, I'd like to really emphasize that if you're at all interested in this work, we're just getting going, so please don't hesitate to contact us. And we'd love to see if there's ways that it might be suitable for you to get involved, partner, or just learn more. So thank you very much. I'd be happy to take any questions.
Thank you very much. Okay, we can start with an audience question here. So what factors into the selective pressures on SARS-CoV-2 infecting children if schools open? Any general thoughts on that?
I'm actually just gonna, sorry, if you could bear with me for a second, I'm just gonna try to get my the chat open just so I can see too what's going on here. So sorry, thank you. Can you repeat that?
Yeah, sure. Any thoughts on factors of selective pressures on SARS-CoV-2 infecting children if schools open? Or maybe just in general, schools opening and SARS-CoV-2 link.
Yeah, so I guess really what you're, so I guess there really, in terms of selective pressure that occurs, it really is about, I guess what you're, is being asked is, are the schools going to cause a change in selective pressure on how this is evolving? Certainly, I think this is still being actively studied, the role children play or do not play in transmission of the virus, because they disproportionately have very mild to no infection or asymptomatic infection. I can honestly say that I think in the end, what we have to keep in mind always is that we are always, by nature of just what we are doing with testing and contact tracing, we are selecting for more transmission, but not necessarily more severe disease. I think there is, I will just add a comment, but that's getting a little outside of genomics and how genomics can inform things, but just say that there is a need to balance, of course, the issues of schools opening and the risk to those who are vulnerable, which is a notable one, with also the risk to the children of just getting education and mental health for many people who are with their kids at home, you know. But I have to say that I don't know, and I'd be happy to think about that more, about the specific impact of taking a young population in or out of the model. But we certainly have some excellent researchers, Carolyn Kajin at SFU and others, who have been doing some mathematical modeling of this as part of the Cancogen or as part of COVID-19 research.
Yep. Okay, we've got less than a minute left. I'll do one more here from the audience. What's the Canadian design for signing up cohorts for collecting viromic data and also the host genomic data?
Yes, so for the host side, it's very much based on a project proposal that's put forward, almost like a research proposal that would just go through regular ethics approval. Some of these projects have already been approved for analysis, and so it's relatively straightforward, but in some cases they haven't. But then the idea is that you're getting this genome sequencing done similar to you would for any human genome sequencing project, but also key is to link up with the public health data so that you can get the corresponding viral genome sequence. I will put a cautionary note that depending on the participant, we may or may not get the virus genome successfully. There is certainly we want to get as much viral sequencing done as possible, but for example, a sample that has a low viral load, somebody who's a bit asymptomatic with a more mild infection, there may literally not be enough virus in the sample to be able to get genome sequencing. So there's certainly a lot of active research, the one being able to characterize these samples more, because some of these, what we call high CT value, high CT sequences or sort of low viral load are basically very much of interest as part of understanding transmission.
Thank you. Thank you very much. All right. Dr. Muirville, are you ready you can go ahead and share your slides whenever you're ready.
Yeah,
okay.
Definitely. Can everyone
Awesome.
Okay first?
Yep.
I want to make sure that goes smoothly. Great. All right.
Yep, and I'll go ahead and just say one quick thing before you get started. Everybody, there's a Q&A box within Zoom. I can speak more in a minute on it, but if you want to ask any questions, just post your questions into the Q&A box in Zoom. And our next speaker here is Dr. Cameron Muirville. He's a postdoctoral fellow in the Sabeti lab at Harvard University. He's developing Cas13-based technologies for viral detection and destruction. In 2016, Cameron completed a PhD in systems biology at Harvard, jointly advised by Pamela Silver and Peng Yin, working at the interface of synthetic biology and nucleic acid nanotechnology. In 2011, he received an AB from Princeton University, majoring in molecular biology with a certificate in quantitative and computational biology. He was awarded a Fannie and John Hertz Foundation Fellowship to support his graduate studies and was named one of Forbes 30 under 30 for 2019. In January 2021, Cameron will be joining the Department of Molecular Biology at Princeton University as an assistant professor. Welcome, Dr. Muirville.
Thank you so much for that wonderful introduction, and thank you to the organizers for the invitation. I'm really excited to share with you some of our work. I've spent the last couple of years really focusing on all sorts of different CRISPR-based technologies for both detecting and destroying nucleic acids, and one in particular that we've been working on quite recently is one for doing massively multiplexed nucleic acid detection. That's a technology called Carmen, and that's going to be the focus of my talk today. And I'll really spend a lot of the talk explaining to you what this technology is and then how we've been applying it to detect all sorts of different types of viruses, including SARS-CoV-2. So without further ado, I think this slide is almost superfluous at this point in time because now more than ever, pathogen detection is of the utmost importance. But we like to think about it in different contexts, whether you're detecting infections in a patient, pathogens circulating in a population, or as we now see, outbreaks and pandemics. And really, these different types of applications have very different needs in terms of things like throughput and multiplexing. And so That's how we like to think about this problem and break it down, where by throughput, what I really mean is the number of samples that we want to test simultaneously. And multiplexing is the number of different targets, and that could be different viral species or sequences or strains that we want to detect at a time. And so we can think about this as a two-dimensional set of axes, and really what we want can be outlined in some of these different situations. So in an outbreak, you might only need to test for a smaller number of things, but you're going to test lots and lots of samples. Whereas if you have a really sick patient, you might want to test them for lots and lots of things if you don't know what is causing that infection. And then disease surveillance falls sort of somewhere in the middle. You want to test for a fairly large number of things and also screen a representative portion of the population. And so unfortunately, a lot of existing technologies really are in the lower left corner of this graph. You can't test for that many things at a time, and often we are limited in our throughput as well, unless you're talking about a single plex assay. So that's sort of this the challenge. As I see it, is we really want to move up and to the right on this graph. And just as by way of background, some of you may be familiar with some of these CRISPR-based nucleic acid detection technologies. There's one called Sherlock in particular that is the basis for Carmen, and it involves basically extracting nucleic acids, doing amplification, and then converting that amplified double-stranded DNA into RNA that can be detected with Cas13, which is this really amazing RNA targeting CRISPR effector protein. It's kind of like Cas9, except that it cuts RNA rather than DNA. And once Cas13 finds its target, it becomes active, really highly active, in fact, and it can be used to cut a reporter sequence. And that basically amplifies that initial binding event into many, many subsequent cleavage events, thereby giving you a lot of additional sensitivity and specificity relative to other methods. And so a typical Sherlock is a single amplified sample with a single detection mix containing a single CRISPR RNA. And recently, just back in 2018, some of our colleagues in Feng Zhang's lab demonstrated 4-plex multiplexing by combining different Cas12 and Cas13 orthologs together. But our goal here is really a lot more ambitious. We want to achieve 100-plex multiplexing with tens of different samples in parallel to really scale up diagnostic testing to the size of the problem. We want to be able to test lots of different samples for lots of different things. And so that's what we set out to do with this technology called Carmen, which stands for Combinatorial Arrayed Reactions for Multiplexed Evaluation of Nucleic Acids. And I should say all of this work was done in collaboration with a really talented postdoc in Paul Blaney's lab at MIT, Sherry Ackerman, who is pictured on this slide. And so one of the great things about Carmen is it lets you perform thousands of different tests in parallel by miniaturizing the reactions, and that reduces reagent consumption. It lets you test basically a set of samples against a set of CRISPR RNAs, and you get this giant matrix of data. And now I'll walk you through a little bit more about how that works. Essentially, we take advantage of miniaturization by taking the same Cas13 detection technology and miniaturizing it into nanoliter scale droplets. And each droplet is given a color code that is a ratio of different fluorescent dyes so that we can differentiate sample 1 from sample 2 from sample 3, and then also detection mix 1 from detection mix 2 from detection mix 3. And each of those will contain unique samples of interest or unique CRISPR RNAs that have a different sequence. And then we can mix all of those droplets together and load them onto an array of microwells. And each microwell on this array, and there are tens of thousands to hundreds of thousands of wells on one of our arrays, contains just enough space for a pair of droplets. And that lets us form all of the pairwise combinations of these different samples and these different CRISPR RNAs. And at the beginning of the experiment, we can image our array to see exactly which samples and which CRISPR RNAs are in which positions using a fluorescent microscope. And then we can merge the pairs of droplets by applying an electrical current, and that initiates the Cas13 detection reaction and gives us a fluorescent readout if indeed that particular CRISPR RNA detected something in that particular sample. And that allows us basically to comprehensively test a large set of samples for many, many pathogens. So I'll just start by showing you a little bit of data. When we developed this technology, Zika virus was the one that everyone was really worried about. And so we initially demonstrated this for detecting Zika. And what I'm showing you here is basically a limit of detection experiment where we're diluting Zika complementary DNA and performing Carmen. And what you can see is that we get a lot of technical replicates because there are many, many copies of each present on the chip. And what I'm showing you basically is the distribution of fluorescences at each of those different concentrations in a dilution series, and then representing that same data on a heat map by taking the medians of each of those distributions. And I'm going to show you a lot of heat maps going forward with those medians rather than showing you all of the individual data points because there are far too many to show on a slide. And you can see that those data points do come indeed from individual droplets that are either bright or are not bright, depending on whether or not the virus has been detected. And we did a quick comparison just to compare our standard Sherlock to Carmen, and we found similar performance between the two methods for Zika detection, which was quite encouraging. So now I'm going to talk about a couple of different applications for Carmen for detecting species, subspecies, and SNPs. And then if there's time at the end, I will also talk a bit about some of our more recent work on SARS-CoV-2 during the pandemic. So I'll start with species detection, and this really is where we got a bit ambitious. We asked the question, can we design a panel to test for all of the human-associated viruses at once? And it turns out there are 169 different viruses that have at least 10 available genome sequences, or at least that was the case when we designed this panel. There's now at least 170 because, of course, SARS-CoV-2 is on there. Anyways, at the time there were 169, and so we designed a set of primers and CRISPR RNAs using some software to try and identify each of those specifically. And then we comprehensively tested cross-reactivity using 8 of these chips for a total of 28,500 tests. And how did we do that testing? So we basically tested every single target against every single CRISPR RNA using a relatively high copy of each of the targets to make sure that these assays were really specific. And that was something that initially was of concern and is one of the reasons in general why people haven't done this type of testing at scale, because you're concerned about cross-reactivity between different targets or different probes or what have you. And fortunately, that experiment worked quite well, as you can see in the heat map on this next slide. And what the expectation here, if the assays are all working perfectly, is that we will see a diagonal line where each specific CRISPR RNA interacts with one and only one target, its cognate target. And indeed, for about 94% of the CRISPR RNAs, that is the case. In our very first experiment, only 85% of them worked. After one round of optimization, we got to 94%. At that point, we said, that's probably good enough for now. But of course, if we continued optimizing those last couple percent of designs, I'm sure we could get those to work well. 2, the median performance of each CRISPR RNA is really high. And that's actually one of the advantages of this method, is that each CRISPR RNA is really working independently. And so if there are issues with one particular CRISPR RNA, it doesn't affect the performance of the rest of the assay. And I think that's really powerful. If there are any issues, it doesn't affect sort of the overall performance. And this is after 1 hour of detection showing fluorescence values. And on this graph are represented basically all of the viruses you've ever heard of, and probably quite a few that you haven't, because it's really a pretty extensive list. We also tested this on a number of different patient samples from dengue, Zika, HIV, and hepatitis C, as well as nasal and throat swabs from influenza. And here you can see some of the results of that testing. What's really interesting is you can detect the viruses themselves, which I've annotated. So there's a cluster of, for example, flu detection or dengue or Zika, but we also pick up a handful of other viruses that we confirmed with sequencing were present in these samples, highlighting our ability to potentially discover other viruses that are infecting people, not necessarily the ones that are making them sick, but perhaps as a co-infection. And we identified a few such viruses. I just want to dive a little bit more into now the comparisons between Carmen and next generation sequencing, because this is something that, as a lab that does lots of sequencing, we're really interested in investigating. And so we looked and basically compared the results of Carmen and NGS, and we found in general really quite good concordance between the two methods, sort of shown on this slide. There were some situations where we detected a sample with Carmen, but not with sequencing, or the opposite. And that's largely because for any given amplicon, Carmen is more sensitive than sequencing, but sequencing can also use the entire genome's worth of information. And so in some cases, what we found with sequencing is that the amplicon we were trying to detect was not present, but other parts of the genome were present. And so sequencing picked up that sample, but Carmen did not. And I think that's another reason why multiplex testing is going to be incredibly important as we think about the future of diagnostics, because if you can tile a genome with multiple different probes or CRISPR RNAs, that will really increase your sensitivity to detect whether or not that material is present in a sample. And also the turnaround time is much faster. In these experiments, our turnaround time was about 6 to 8 hours. That is something we are currently working to streamline and optimize. Okay, we can also do subspecies identification. I'm going to go through this a little bit quicker because I realized that I'm running a little bit lower on time here than I had anticipated. But basically, we can subtype influenza. So we did H1 through H16 and N1 through 9, which are the canonical subtypes of influenza. And that works quite well, shown with synthetic targets here, H1 through 16 and N1 through 9. As well as with patient samples here, we have a set of H1N1 and H3N2 patient samples, and we're able to correctly subtype all of those samples, showing 100% concordance with PCR and with sequencing. Okay, so now we're zooming in a little bit more, and one of the really cool things I would say here about Cas13 is that it can actually be used to detect single nucleotide polymorphisms in addition to these higher level sort of species and subspecies identification problems. And here we chose HIV drug resistance mutations as our canonical SNPs of interest, and there's a set of 6 in particular in the reverse transcriptase gene that we focused on. And here what we do is we actually use a pair of CRISPR RNAs, and one targets each allele. And by comparing the relative activity of those CRISPR RNAs, we can tell if the mutation is present or not. The idea being that if the CRISPR RNA matches the sequence, it's going to have more activity than if there is a mismatch due to the presence of a particular mutation. And indeed, that's something that we saw, again, both using synthetic targets for this panel of 6 CRISPR RNAs, as well as in a handful of patient samples here. I'm just showing you a subset of 4 where patient 2 has this K103N mutation that we can pick up, and the other 3 patients do not have any drug resistance mutations. All right, so in summary, we're really excited about this technology. It can offer a really high degree of multiplexing for detecting different species, detecting different subspecies, as well as SNPs. And in all cases, we were able to validate that by testing the method on patient samples. And so I just want to take a moment here and put that back into context, and remind everyone of that graph that I showed you at the beginning of the talk. And our human-associated virus panel sort of is a fair ways up and to the right on this graph. I'm showing you a line here because the design of Carmen assays is actually relatively flexible, and it's really limited only by the size of the chip. And so you could imagine a different assay that used a subset of the human-associated virus panel, and that would let you test more samples at a time. Because if you're testing for fewer things, there's more room to sort of add additional samples. It's also relatively inexpensive. So the cost per sample is about $10, and that's for a 100-plex assay. The cost does scale with the number of targets that you're testing. And I'd say there's a lot of synergy with next generation sequencing here. I think one of the really cool applications that we're just beginning to explore is using this as a way of triaging samples for sequencing. With that, I'd like to really acknowledge a large team of people that made this possible, including my advisor, Pardis, a number of really wonderful people in the lab who helped out. This was a really cool collaboration with Paul Blaney's group at MIT. Their lab really built the underlying microfluidic and sort of microwell array technology that Carmen is based upon, as well as collaborators in the CDC who provided some of those flu samples that I mentioned. And I just wanted to show you as a sort of brief teaser at the end, a little bit of the work we've been doing with Carmen for SARS-CoV-2. So we've developed a respiratory panel that goes after 21 different respiratory viruses. We've sort of tested it out using synthetic targets, and in general, that all works quite well. What I'll just leave you with is some initial testing we've done with SARS-CoV-2 patient samples. This is a set of 6 samples that are positive and 4 that are negative, and we're able to pick up those infections in those samples.
Thank you very much. So I believe we have a 10-minute break now, but I'm just going to go ahead and spill over a little bit into it to ask a few questions. We've got some nice questions from the audience actually ready to go for you. So the first question, how do you design and select primers for so many different sequences at the same time? Is it done manually?
There is software for this, and actually one of my colleagues, a really talented graduate student in the lab named Hayden, has developed a software package for this called Adapt. That he is currently working on a publication to describe that method. There's a little bit of information. We've released a set of Adapt designs for respiratory viruses in a preprint with just a couple months ago. And hopefully we'll be able to share the entire software package soon. We're really hoping to get that out there, because that's a really important part of this whole system. If you can't design the sequences, then it's hard to make these assays work.
Okay. How about, sorry if anybody hears a little lightning, it's kind of storming here right now. If you rule out like known pathogens, let's say, what about using it for like unknown pathogens?
Yeah, I would say that is one of the limitations of the approach, is that you generally need to know the sequence of what you're going after to design some of those primers or CRISPR RNAs. There are some cases where you can go after highly conserved regions and maybe suspect that something is present without knowing exactly what it is. But that is why I think that sequencing is really powerful. It's to give you that initial information that you need, and then our model is, or our vision is very much that once you have that sequence information, you can use it to make these diagnostics, even better. What
about comparison to NGS, just sequencing and qPCR based methods? And they ask specifically in terms of like consumption of resources of all the consumables and things like that also. Yeah, definitely. So I'm going to go back to the earlier slide here for a moment. Yeah, so in terms of cost, it depends a lot on how you're doing your NGS, but the cost is somewhere between a factor of 10 or more lower per sample than sequencing. This depends a lot on how much sequencing you need to do though. And for viruses in particular, the challenge is that they often are a small percentage of what's present in a sample. So you need a lot of reads in order to detect something. We saw greater performance for Carmen on a per amplicon basis, but when you include the entire genome, I'd say the sensitivity is similar. And that is one of the advantages of sequencing, is that you can detect lots of different regions. In terms of qPCR, the performance of Carmen on a per locus basis is quite similar to qPCR. And I think the key advantage is that we're able to test for lots of different things at a time, and also test lots of different samples at a time. Thanks. I'll ask 2 more final notes. It looks like you kind of already put it up on the slide here, but do you want to speak at all to time and expense at all on any of these? Yeah, definitely. So in terms of time, for the version of Carmen that is presented here, that was about 6 to 8 hours per experiment, and that depends a little bit on how long you need for that that last detection step, which can range in time. We have been working really hard to optimize that and improve it. And I think now we're at more like 5-hour turnaround time for some of our latest implementations of the technology. In terms of cost, yeah, you're looking at around $10 per sample. Per test, the cost is extremely low because you're testing, in that case, 100 plex. So the cost would be 10 cents per test, which is where you really that's what really enables you to test for things that maybe you wouldn't test for if you had to run a whole qPCR reaction dedicated to that test. All right. Thank you very much. Yeah, thank you. All right, so everyone, we're on break for just a few more minutes, and then we're going to get started officially back up in about 7 minutes for Dr. Peter Hotez's talk. All right, we've got about a minute or so left. Dr. Corbett, are you here by any chance right now? I don't see Dr. Hotez online quite. I don't see Dr. Hotez online quite yet. If he maybe is held back a little bit, do you think there'd be any chance we could do like a little bit of a swap and have you go like now?
Sure.
Would that be okay if we need to, or?
No, absolutely. That would be okay. I probably I timed the notifications on my computer.
Okay.
So just clean up some things.
Okay. Sorry about that.
No, it's fine.
We'll give him another minute here and see, but just in case we have to just swap. Yeah, there's also there's a pretty bad storming going on in Houston. A lot of us are actually based in Houston, myself, Peter, Todd. So also everyone, please just be patient with us if a couple of us have some power issues flickering in and out.
Yeah, no problem. This is what you deal with when it's virtual.
Yeah. All right. Yeah, why don't we just if you're ready, why don't we just go ahead and have you present if you're all right with that.
Yep, I'm pulling my
I don't see him. Okay, thank you. All right, everyone. So we'll just do a little switcheroo here. Peter may be held up or may have lost power. Who knows what's going on there? So we'll just do a quick swap. So
I'm trying to pull my slides up. They're
yep,
coming slow. Yes, they're big.
Yep, and I don't know if you want to share your video. Also, you don't have to. It's up to you. But if you want to, feel free. I don't see anything currently.
I mean, why not?
Okay. Okay. All right. Yep. And sure. Go ahead and share your screen there. And so our next speaker is going to be. It looks like we'll just do this quick swap here. So our next speaker will actually be Dr. Kizzmekia Corbett. Dr. Kizzmekia S. Corbett is the scientific lead for the coronavirus vaccines and immunopathogenesis team at the National Institutes of Health, National Institute of Allergy and Infectious Diseases Vaccine Research Center. Her work focuses on developing novel coronavirus vaccines, including mRNA 1273, a candidate vaccine against the current coronavirus that's causing COVID-19. In response to the ongoing pandemic, the vaccine concept incorporated in mRNA 1273 was designed by Dr. Corbett's team from viral sequence and rapidly deployed to industry partner Mrna for FDA-approved phase 1 clinical trial, which started only 66 days from the first release of the virus sequence. Phase 2 studies began on May 29, 2020, and FDA feedback has been received for phase 3 studies. Alongside mRNA 1273, Dr. Corbett's team boasts a portfolio which also includes universal coronavirus vaccine candidates and novel therapeutic antibodies. Additionally, she spent several years working on a universal influenza vaccine, which is slated for phase 1 clinical trials in the upcoming year. She has 15 years of experience and expertise in influenza, coronaviruses, and other pathogens. Welcome, Dr. Corbett. Unless we want to do,
I think she's on mute.
Unless we want to do a quick swap back. Dr. Hotez, I see you are here now. Sorry, we weren't sure if you were having maybe any issues or anything. Are you ready to present? We can do the normal.
Yes, if you don't mind, because I have to.
Yeah. Let's.
Sorry, I have to go on. I have to go on CNN after this. So
sorry about that, Dr. Corbett. I'm sorry.
It's okay. No.
Okay.
Oh,
okay,
I'm sorry.
So
am I?
No, we weren't sure.
In the wrong time slot.
Nope. You're good. You're good to go. We weren't sure. Maybe you had like a power outage or anything like that. A lot of people I know are dealing.
Oh
with
no,
that.
I joined earlier, and then
Okay.
I understood that I go on at 11, not 10. So.
Okay. Yeah, you're good. If you want to go ahead and share your screen, we'll go ahead and kick it off.
Okay, great. Yeah.
I'll
be brief.
Sure. Okay. So our next speaker. Sorry, everyone. Our next speaker, Dr. Peter Hotez. He's a professor of pediatrics and molecular virology and microbiology at Baylor College of Medicine, where he's also founding dean of the National School of Tropical Medicine and co-director of the Texas Children's Center for Vaccine Development. Professor Hotez is a world authority on neglected tropical diseases of poverty and vaccine development. He served as U.S. Science Envoy for the Obama administration in the area of vaccine diplomacy. Professor Hotez is a member of the National Academy of Medicine and American Academy of Arts and Sciences. He obtained his MD and PhD from Rockefeller University and Weill Cornell Medical College and was a pediatric resident at Massachusetts General Hospital. He's the author of more than 500 scientific papers and 4 books. Welcome, Dr. Hotez.
Thanks so much, and apologies if there was a bit of a miscommunication, but honored to be here and always to work with Rice University. So we develop global health vaccines and have been pursuing this concept of what I call vaccine diplomacy, and I'll explain what that means. And so today I'll summarize briefly our efforts to develop a low-cost COVID-19 vaccine for global health, but also some of the other geopolitical issues around that in terms of fighting a growing anti-vaccine movement that's gained strength around COVID-19, as well as this ethereal concept of vaccine diplomacy, which is falling apart because of some new trends in nationalism. So this is, by the way, a new instant book that just came online. In case people are interested, I like to write books. I wrote a book in 2016 called Blue Marble Health, which looks at World Health Organization, Gates Foundation data, showing that that paradoxically, most of the world's poverty-related diseases are actually in low and middle income, are actually in wealthy nations, but among the poor living in wealthy nations, including the G20 nations. And it's a concept that's not really caught on yet, despite what I think is a pretty good database for this coming out of 2 different sources. But the idea is that's the poor living among the wealthy in the G20 that account for so many of our poverty-related diseases like tuberculosis, leprosy, leishmaniasis, and COVID-19's going by that same playbook. And although we don't think of it as a neglected disease or as a poverty-related disease, in fact, this I think is what's happening right now in the United States as the virus is racing through the southern half of the country in our southern metro areas. I think it's overwhelmingly people living in poverty who are piling into our hospitals and ICUs and getting sick. And right now the estimate is around 13 million cases globally. And unfortunately, and it's mostly the poor who are bearing the brunt of this. And right now in the southern US, we've got the new numbers coming out today are roughly that 1/4 of all the world's new COVID cases are occurring in the southern US in a swath going from Florida to the Gulf Coast to Texas, New Mexico, Arizona, and Southern California. So this is the epicenter of the COVID-19 pandemic. And in Texas, things are very rough right now. We just got a new update from the Philadelphia models, which I haven't had time to put in, but it shows that as bad as things are right now, we're going to double by the end of the month. And the cases are really piling into our Texas Medical Center, which is our big medical center here in Houston, the world's largest medical center, into our ICUs and hospital beds. And also staff is getting exhausted as well, hospital staff. And but the other piece that we don't have all the numbers on are the deaths are overwhelmingly in Black and brown people, African-American, Hispanic, Latinx populations. And this is why I've been so out there in the public domain, because I don't think this point is being, hit. We're not calling this out, that we are failing to protect some of our most vulnerable populations living in poverty who are essential workers, who have to physically be in the workplace in order to get a paycheck and live in neighborhoods where social distancing is more problematic. And it's really awful what's going on right now across the southern part of the United States. And we have to, and I feel like we as scientists, we have to speak up and do something about this. And in the lab, we are working to develop COVID-19 vaccines. And this is a recent slide from Dr. Fauci, Anthony Fauci, who spoke at the National Academies a few weeks ago. And one of the things that people often fail to realize is that essentially all of the vaccines are working by the same principle. We need to induce a protective immune response by creating immunity to the spike protein of the virus. And in particular, an essential component of that is to induce high levels of virus neutralizing antibody that will bind to that spike protein and prevent virus attachment to its host ACE2 receptor and then gain viral entry. And the question is, what's the best way to do it in terms of efficacy and what's the safest way to do it? And there's differing opinions. We think a recombinant protein vaccine is going to be needed for that purpose. And that's where we're listed there on Dr. Fauci's slide, but there are nanoparticle approaches, adenovirus vector vaccine approaches, DNA, RNA vaccine approaches, and all of them have pluses and minuses. And what we're seeing now is that these are all moving into different stages of clinical development. For our vaccine, we based at Texas Children's in Baylor in the Texas Medical Center, we partner with PATH, which is an organization not too many people know about it. It's a very important organization based in Seattle. Historically, it's done a lot of work with the Gates Foundation to develop the malaria vaccine for Africa and the meningococcal A vaccine for Africa. Now we've created this partnership with them to accelerate global health coronavirus vaccines. And our center is focused mainly on recombinant protein vaccines using microbial fermentation technology, either bacteria or yeast. We also do mammalian cell and insect cell expression, but we like the microbial fermentation because of ease of technology transfer to low and middle income countries where they can actually make the vaccine. So a big component of our activities is around capacity building. And the truth is we're not really virologists. We are mainly, we work on parasitic disease vaccines that we've accelerated to clinical trials for schistosomiasis and hookworm and river blindness, leishmaniasis, and Chagas disease. But about a decade ago, we teamed up with a group from the New York Blood Center led by Shibu Jiang and Lanying Du, who had worked out a very promising approach for coronavirus vaccines, where they found that whereas the full spike protein of the virus would induce immune enhancement, they could prevent or block that by just focusing on the receptor binding domain part of the virus. And we partnered with them to use our ability for scale up to develop coronavirus vaccines. So we've been doing this for about the last decade. And the nice thing about our approach is these are well suited for this situation that we're starting to see now. As bad as things are in the southern US, I'm worried things are going to get much worse as this virus races now through the favelas of some of the northern Brazilian cities such as Fortaleza and Manaus and Belém, but also now in the urban slums of Dhaka and Delhi and Hyderabad and everywhere else. And so this is our big approach, and we're seeing a lot of devastation now in Latin America, and I suspect soon we're going to start seeing this as we proceed through the summer across the southern hemisphere into Africa and into South Asia. And our approach is this: it's those green blobs there are Pichia pastoris, methanol utilizing yeast. And the reason that's relevant, this has been used to scale up production of the recombinant hepatitis B vaccine, now manufactured locally in Bangladesh and Brazil and Cuba and India and Indonesia. So we feel our approach is going to be very useful for technology transfer, that where they already have the capacity for making recombinant hepatitis B vaccine, we could do this for our COVID-19 receptor binding domains. And we have two candidates that we've developed. One is the original SARS receptor binding domain, SARS-1, which we had already manufactured in 2016, and that we think could be used as a heterologous vaccine for COVID-19. But in the meantime, our group has really worked rapidly to apply the same approach to specifically focus on the RBD of the SARS coronavirus 2. And that has really, we've been able to work very quickly, in part because all of the subtle technologies that we use to scale up the RBD, receptor binding domain of SARS-1, has worked really well with SARS-2. So both of these molecules, which are the receptor binding domain of the spike protein, induce high titers of neutralizing antibody, We also found that focusing on the RBD removes the immune enhancing epitopes associated with the full spike protein. And again, we use that yeast production system that's been used for hepatitis B. So this is an example of what our in vivo studies look like in laboratory animals. And for this, we've been partnering with Ken Cheng's group at the Galveston National Lab, where he's developed a transgenic mouse model for coronaviruses, as well as looking at mouse adapted coronaviruses. And you can see that we can develop pretty high titers of virus neutralizing antibody against both pseudovirus and PRNTs against a live virus. Now this is for our SARS-1 vaccine, and we get 100% survival compared to control mice. And this is what eosinophilic immune enhancement actually looks like. So a lot of people talk about immune enhancement, but not many people really show what it is. And we've spent a lot of time looking at it. And to cut right to it, these brown dots that you see is immunohistochemistry staining of eosinophils. And this is what happens if you use the full-length spike protein. But if we use our receptor binding domain, we can really, and depending on the ratio of alum that we use or other adjuvants, we can really hold that off. And I think that's very instructive when we look at the receptor binding domain for SARS-2. And here Lanying-Du has shown that the RBD1 will cross neutralize SARS-2, but now we're moving directly to the SARS-2 as well. And another advance that we found that works for SARS-1 and SARS-2 equally is, with yeast proteins, you always worry about excessive glycosylation, which can interfere with immune responses. And indeed, the SARS-2, like the SARS-1, if you don't remove certain glycosylated amino acids capable of glycosylation, you get this smear, which represents differing levels of glycosylation. And by expressing the protein without the N-terminal amino acid, we have found we can preserve the immunogenicity and get a very tight band over here. And the reason that's useful is, one, if you're trying to do quality control for production, you want to show that you can do it the same way every time, and that's helpful. But also, we get much better levels of virus neutralizing antibody and protection. And so all of those things that we spent years working on for SARS-1 has worked for SARS-2. And we're hoping to announce in a couple of weeks, maybe next week, this partnership that we're doing with India to accelerate that vaccine as a low-cost Pichia-based recombinant protein vaccine for India. So we're not part of Operation Warp Speed. That's all focused on the 5 pharma companies. We do see a path to also developing this in the United States, and we're working on that as well, provided we can find a potential pharma partner interested in doing that. And this then brings us to Operation Warp Speed, which has a lot of merits in terms of accelerating those 5 vaccines. You've heard about the Mrna vaccine now going into phase 3 trials, and the others will follow. The problem has been the messaging coming out of Operation Warp Speed, while well intended with that name, has actually worked against us, because we have a very aggressive anti-vaccine movement here in the United States that I've gone up against for the last few years. And new polls coming out of Reuters and the Associated Press find about only half of Americans will accept COVID-19 vaccines, even if they become available. And many in academic health centers and universities are surprised to hear about this, in part because they don't keep their eyes and ears open about this, how aggressive and pervasive this anti-vaccine movement has become. Unfortunately, the language and the absence of communications coming out of Operation Warp Speed have been very tone deaf to the fact that if you look at what the anti-vaccine tenets are, what the movement claims, they're playing right into it. And so they claim that vaccines are rushed. What does the anti-vaccine movement claim? Vaccines are rushed. They're not adequately tested for safety, that there's a cozy or conspiratorial relationship between the pharma companies and the US government. They claim this concept of health freedom, that the government can't tell us what to do. And then they claim that vaccines cause autism and other chronic diseases. And I've spent years diffusing that bottom one because I have a daughter with autism and wrote a book called Vaccines Did Not Cause Rachel's Autism. But the way Operation Warp Speed is operating is really helping to promote all their other agenda. So,
Dr. Hotez, I just want to let you know you've got about a minute left before we're going to.
Oh, really? That's it? Okay, I'm sorry. So
sorry about that.
So this is what we're seeing, though, this very aggressive rise in kids not getting vaccinated in Texas. And this is the Health Freedom, which is now a movement, which has expanded from vaccines to social distancing, contact tracing, and parading against masks. And a lot of this is now turned into this larger anti-science movement that's targeting me and other scientists in Texas. And so I see the autumn as being a time of great instability in the United States. We're already seeing big declines in vaccine coverage, and we're still dealing with the fact that things cause autism. And this is my effort to combat that, writing this book, Vaccines Did Not Cause Rachel's Autism, and putting out positive messages about vaccines. And the last point I'll make, and then I'll stop. The other thing that's happening, other than promoting the anti-vaccine movement, I don't like, is we're starting to hear about the Chinese vaccine, the American vaccine, the British vaccine, the European vaccine. There's even a term for it now, vaccinationalism. And this is very damaging, because the truth is, if we're going to successfully develop these vaccines and vaccinate the world, we need the opposite. We need vaccine diplomacy. And I like to point to the story of when Albert Sabin developed the oral polio vaccine. Not many people realize it, but he did it jointly with the Soviets at the height of the Cold War after the Sputnik launch, where the two countries put aside their ideologies to develop that vaccine. And that led to the near elimination of polio and eradication of smallpox. And if we don't fix this problem, we're also not going to achieve our goals. So I'll stop there. We have other vaccines that we develop for poverty-related diseases, and just say that we're in this epic struggle right now where we're developing vaccines, but we have very powerful 21st century forces that are going to make this difficult: anti-science, urbanization, poverty, climate change. And this is why you need a place like Rice University that's got all of that capacity in these other areas, and trying to figure out a way to create this better dialogue between the biomedical sciences, the physical sciences, and all of these, all the social scientists and humanists committed to these concepts. So I'll stop there. Thanks so much.
Yeah, thank you very much, Dr. Hotez. And we'll leave some of these questions in the chat, everybody. We have another vaccine talk coming up next, so we can actually keep them. And I think Dr. Corbett's going to be able to also chime in on a lot of these. So thank you very much for your time, Dr. Hotez. Appreciate that.
And thanks so much for inviting me. I really appreciate it.
Yeah, and Leo, can we just grab one question?
Yeah, go for it.
Rushes to CNN.
I would say for you, it's going to be about, how is the population dealing with these vaccinations? So for instance, this question: in polls, is the percentage of Americans that say they'll get a vaccine for SARS-CoV-2, is it different from the percentage to say the flu vaccine?
Yeah, well, remember, the anti-vaccine movement also works on the flu vaccine also. So that is another problem. So that's a good point. So our uptake of flu vaccine is also awful. And now we're coming into flu season, and we know that co-infections of flu and COVID-19 occurred all the time in China in December and January of December last year and January of this year. So that's going to be a huge issue. And the rate of uptick of the flu vaccine is really low. And that's why in one of those slides that I had to rush through, that we're trying to look at messaging around the flu vaccination as well. The point is, we're going to have our hands full, especially now. We're also getting a low uptick of measles vaccinations. And if we have to deal with this triple epidemic of COVID-19 or influenza and measles, that's going to make things very dreary this fall and winter.
Maybe, sorry, Leo, I was going to jump this, Afshin. Maybe I could ask a question to you, Dr.
Sure.
It's something another in the Q&A. We know recently, we've been starting to see that the long-term effects, that immunity and antibodies are lost in some patients. So how do some of these vaccines, will the memory last in these vaccines, and will it be effective if the immunity is lost in patients due to this weird behavior of COVID-19 and SARS-CoV-2?
Yeah, and so this is a question everyone is struggling with. And, with SARS-1, we saw antibodies that seemed to last for a few years, in some cases as long as 12 years, and maybe longer than that. With this one, it depends. Some people seem to maintain high antibody titers for a while, others lose them pretty quickly, especially, and there's a rough correlation with the severity of disease, so the ones who are asymptomatic. And so that's unknown. We do know there are a lot of upper respiratory coronaviruses where people can get repeatedly infected. And that may be the case for people who get asymptomatic illness. The only thing I can say is, even though antibodies are waning, in some cases to the point where they're undetectable, there may still be memory B cells or long-lasting plasma cells, so that they'll respond very robustly to, if either they're infected again, they'll develop a quick antibody response so they won't develop severity of illness. Or remember, those populations, if they get vaccinated, may have a boosting effect also. So it may not be as dire as some are projecting this to be.
Okay, thanks. Yeah, that helps. Thank you.
Okay, well, thank you again. I appreciate the opportunity.
Thank you. Dr. Corbett, can you give me a quick reminder? How are you doing on time? We can flow a little bit into the break, depending if you are up for it, because we have a break after this.
That works for me.
Okay. Okay, great. Okay. All right. So our next speaker, you heard her introduction, but maybe if anybody wasn't here, I'll just do a real kind of brief highlights here. Dr. Corbett is the scientific lead for the coronavirus vaccines and immunopathogenesis team at the National Institutes of Health, National Institute of Allergy and Infectious Diseases Vaccine Research Center. She's actively focused on developing novel coronavirus vaccines, including mRNA 1273. She's also on working on a universal influenza vaccine slated for phase 1 clinical trial. And I'll leave it at that. I'll just go out on a limb here to say that I think her work speaks for itself, as you'll soon see. So welcome, Dr. Corbett.
Thank you for having me. And also, my PI wants everyone who organized this meeting to know that he graduated from Rice and he would have kindly accepted the invitation. So I am Kizzmekia Corbett, and I am a senior research fellow and the scientific lead for the coronavirus vaccine and immunopathogenesis team at the Vaccine Research Center at National Institutes of Health. I work under the direction of Dr. Bernie Graham, where generally we study prototype pathogen preparedness, vaccine development, and I'm going to give you a small snippet of our story surrounding our efforts towards a SARS-CoV-2 mRNA vaccine in collaboration with Mrna. And so developing coronavirus vaccines is not new to the Vaccine Research Center. Actually, that predates my tenure at the Vaccine Research Center in the SARS-CoV-1 days, where in 2013, there was the SARS-CoV-1 virus that was discovered. There was this widespread epidemic, but soon the virus did die out. Nevertheless, in an effort to develop a vaccine for SARS-CoV-1, the Vaccine Research Center developed a DNA vaccine platform. And I just show you this slide because it really shows the timeline from discovery of a virus to initiation of a first-in-human trial for a particular vaccine candidate out of the Vaccine Research Center over 10 years ago. So this was over a year towards a phase 1 clinical trial. And since then, we've taken on this prototype pathogen approach to pandemic preparedness, where the idea with this is that you research a known prototype virus within a given family to a large enough extent that you know so much about that prototype virus that you're able to apply that knowledge to other viruses within that family, and apply that knowledge so widely that you're able to, how I like to call it, plug and play your vaccine development efforts in the onset of a pandemic, similar to what we've done with this particular COVID-19 pandemic. One of the other kind of bullet points under that prototype pathogen preparedness is that you would ideally want to develop those countermeasures or vaccines through phase 1 clinical trial. which will help to speed the development of the vaccine if there shall be a pandemic. And there's more information on this approach to pandemic preparedness in a commentary that Dr. Graham and I wrote in the Journal of Clinical Investigation. So as we think about prototype pathogen preparedness, we think about SARS-CoV-2, which is one of the members of the coronavirus virus family. It is about 80% identical, at least in the spike protein, which Dr. Hotez gave a really good introduction to SARS-CoV-1. And so with that, because of much of the extensive work with MERS and SARS and other coronaviruses that have happened really over the last, concentratedly mostly over the last 6 years at the Vaccine Research Center, we were able to, on the onset of this pandemic, initiate a rapid response for vaccine development that landed a phase 1 clinical trial starting only 66 days after the sequence of the virus was released. And I will detail many parts of this timeline, but in 15 minutes, I really am going to focus on 2 major aspects. And the first of which is being some of the work that we did prior to the pandemic that really laid the footprint for how to respond in this instant. I don't really need to give too much detail on the spike protein, only that it is the target vaccine antigen. And if you elicit antibodies to the spike protein and to the right places on the spike protein, as Dr. Hotez alluded to, you will likely have a very good vaccine response. And so we use the spike protein as our target antigen. And specifically for us, we use the entire spike protein as our target antigen with sort of a twist in that one of the things that we do is called structure-guided mutagenesis for vaccine immunogens. And that allows us to, as you can see with all of these proteins that are similar to the spike protein, they have this fusion machinery, which is the way that they do their work or allow for the virus to get inside of a cell. They fuse with the host cell. And so the ideal vaccine antigen is actually, in many cases, the stabilized pre-fusion version of any of these proteins. And that concept was really proven as a concept with RSVF, which is my boss's baby, is what I like to call it, where there are years of work being put into really understanding how and why the pre-fusion version of RSVF is more immunogenic than the post-fusion version. And so I really just show you these. I hope you can see my pointer. I'm not really necessarily sure, but the pre-fusion version is this squatted shaped version that has these red areas on it that are really, really, really potent immune epitopes or antibody epitopes. And then when it flips into its post-fusion conformation, those are occluded or hidden. And so you can see why this would be a better vaccine concept. And so we took that knowledge and we wanted to apply it to MERS coronavirus. And this started, when I started my research fellowship at the Vaccine Research Center. And so what we did was we, in collaboration with Jason McClellan's lab and Andrew Ward's lab, we solved the structure of HKU1 coronavirus spike protein, which is a human coronavirus that is a seasonal coronavirus that people get, every so often seasonally, causes the sniffles. It was really easier to solve for the structure, but it really let us understand on a very atomic level what details were around this fusion machinery sitting here in the second part of the spike protein, and how we could really lay that beside the MERS coronavirus and the SARS coronavirus spike protein and scan it for mutations that might stabilize it in its pre-fusion conformation, and I say we stumbled upon, but obviously there were hundreds of mutations that we tried. Nevertheless, we stumbled upon 2 mutations at the apex of the central helix region here and the heptad repeat 1. Those prolines, as we call them, the 2P version of the spike, allowed for MERS and SARS to be expressed better in cell culture compared to wild type. Also, you can see by negative stain EM, the 2P version of the spike does not readily flip into its post-fusion conformation. And so now we have this stabilized pre-fusion spike protein, and it proved to be extremely immunogenic, even more so than the wild type protein, and even more so than the receptor binding domain that Peter Hotez was talking about, and the S1 region of the protein. And interestingly enough, as you think about pandemic preparedness, you could effectively transfer those 2 proline mutations into the backbone of several spike proteins and get your pre-fusion conformation. So we did that. On January 10th, when the sequence came out, we plugged and played those 2 proline mutations into the backbone of the spike protein to make protein so that we could solve the structure in collaboration with Jason McClellan, which came out on February the 19th, but also in collaboration with Mrna, who we'd been working with for several years in a collaboration to really understand mRNA delivery of our vaccine antigens, and applying that concept because of the speed of mRNA to pandemic preparedness. And so mRNA is a platform where you can put into a lipid nanoparticle and messenger RNA sequence of really any protein, and then allow the body to make that protein. And in so many ways, it's fast, as we're seeing, reliable because it's been tested multiple times through phase 1 clinical trial in humans. And it's kind of how we think about universal, but I have it in italicized, but at least it can be applied widely across different virus families. And we'd studied mRNA in the concept of MERS prior to this pandemic, and shown that MERS actually served as a proof of concept for delivering the S2P antigen in animals. And so we showed that the MERS S2P elicits robust protection in animals at 1 microgram and 0.1 microgram doses. And you see this beautiful protection of the long viral replication in these animals that are challenged with this lethal virus. It's also without any evidence of enhanced illness, which I'll come back to a little bit later. And so we had this kind of book, black book, so to speak, of data essentially waiting for a pandemic. And so it really allowed us to what we call at risk, apply our concepts to SARS-CoV-2, and then have Mrna then made GMP available material for clinical trial, and then also clinically relevant material for us to immunize animals. And so we immunized several strains of mice with the SARS-CoV-2 mRNA. And so you'll know probably from all of the news that it's called mRNA 1273, and it elicits robust binding and also neutralizing antibody responses in 3 strains of mice. This is with a 2-dose regimen of the vaccine, but also with just 1 dose, which really, really begs the question of its utility towards use in a pandemic such as this, or in an outbreak setting. Also, as you think about safety and avoiding those vaccine-induced enhanced illnesses that many other vaccines, particularly for measles or RSV, and then in vivo for SARS-CoV-1 have shown to do, you want to induce a Th1 biased T cell response, or at least a balanced Th1, Th2 T cell response. And so we looked at the IgG2A and IgG ratio in those mice and showed that they have a balanced Th1, Th2 like antibody isotype response. As well, looking at the CD4 T cells, and we also looked at CD8s, but I just show this here for brevity. You can see that all of the blue are Th1 biased cytokines that are being expressed by the CD4 T cells, and they are antigen specific T cells that are expressing this, whereas the orange and red cytokines, which are more like Th2 cytokines, are expressed to lesser, and that is a dose dependent type of response. So at the higher doses, you get more of the Th1 biased T cell responses in these mice. And we were able to protect mice from challenge with a mouse-adapted strain of SARS-CoV-2. And so giving 2 doses of mRNA 1273 at 1 microgram, which is actually a very low dose. And so one of the reasons why we started at this low dose as opposed to what other people have been starting with in the vaccine field is because we had so much knowledge that guided us to a dose based on our Mrna's work. And so at this, very modest dose, we were able to protect mice from lung viral replication and also viral replication in 3 out of 4 of the mice in their nasal terminates. And so we have this protection in the upper and the lower respiratory system. And also not just with 2 doses, but with 1 dose as well. And so given just 10 micrograms or 1 microgram, and then challenging the mice 7 weeks later, we were able to protect those mice from lower respiratory virus replication. And so, where is the vaccine now? As of this week, the phase 1 clinical trial preliminary report has come out, and it is on a trajectory towards a phase 3 clinical trial. We have several other sets of animal data that I cannot show because it's not public yet. But what is public now and has been recently added to my presentation is just one brief slide to show you what the immunogenicity looks like in an actual human. And so this study was led by Lisa Jackson in Seattle, and it shows mRNA 1273 given at 3 doses, 250 micrograms, 100 micrograms, and also 25 micrograms in a phase 1 clinical trial of 15 participants per dose. You can see that there is this dose dependency in the S-specific binding antibody responses, where you get binding antibodies after 1 dose, and then with a boost, you get an increase in those antibody responses. And then the thing that we have been really excited about is that in a comparison with a panel of human convalescent sera, so those are humans that have gotten infected with SARS-CoV-2 within the last month or 2, having taken their sera, we are in the upper quartile of those antibody responses. And even more so when you look at the neutralizing antibody responses, particularly in just zoning in on the 100 and 200, well, the 100 microgram dose, because that's what's going into the phase 3 clinical trial. All 100% of every single participant in this phase 1 trial has a robust antibody response, no matter what their dose was. And we are getting in the upper quartile of the human convalescent sera. In fact, you can see about even 1/3 of people don't even have a detectable neutralizing antibody response to SARS-CoV-2 after natural infection. So this was really good to see. The phase 2 clinical trial has already started and has completed enrollment. And so you'll start to see some data coming out of the phase 2 clinical trial with 600 participants. And hopefully by the end of the month, we can start to vaccinate up to 30,000 people to get an efficacy signal with this vaccine to just ask, does it work or does it not? But we are very excited about the immune responses that we're eliciting in animal models and in humans. And without further ado, I will say thank you to everyone on our team at the Vaccine Research Center who has been kind of ride or die with this before the pandemic. And it was just our small team, but now it's really a Vaccine Research Center, all encompassing effort and with all hands on brains on deck. I'm so thankful. And then all of this animals work is online in the preprint. And we have a ton of collaborators all over the country, particularly Mrna, UNC, University of Texas, Austin, and Vanderbilt, that have helped to really bring all of these efforts into fruition over the last 6 years, and particularly in this moment, in this rapid response. And I'm sorry that I sped through that, but I thought I had 20 minutes until he said 15, so here we are. And thank you for having me again.
Thank you for joining. No problem. All right, let's do a couple questions from the audience here. So the first one is going to be, I think, kind of a repeat here. So let's get your take on it here. How long does the immune response to the mRNA vaccine last?
Yeah, and so we don't know how long it lasts. That's actually just the really frank response. And so our first person was vaccinated in February and got their second dose in March. And so we're about 3 months out. And so time will tell how long these immune responses last. I will say that it will not be surprising for there to be a dip in the immune response, because every vaccine shows a peak immediately after the vaccine, and then there starts to be a waning. But as long as you get to a point where you wane no further than what is the protective threshold, which we also have no clue about at this point, we think we will be fine. Given the fact that many of these sera were taking after, a couple months after a person is vaccinated, at the same time point that many of the people for which the convalescent sera were collected, we're really optimistic that we at least are getting over a threshold that may be durable.
Thank you. Someone's asking about vaccine manufacturing. Is it a significant bottleneck to the deployment of the vaccine? And if so, how much faster would Moderna's mRNA approach or other novel approaches be than the conventional vaccine manufacturing?
So I'm trying to stay away from saying conventional, because I want conventional to change in all aspects of everything. You got to think a little revolutionary, right? So conventional meaning the types of vaccines that we currently get in our muscle, whenever we get vaccinated, whole inactivated vaccines, protein subunits. They do take quite a bit of time and also resources, because they're made in cells. You have to purify and blah, 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 blah. Using the body to make the protein, generally you can get over that one hump. And that's why nucleic acid vaccines are ideal in this type of setting. That being said, no one company that has a vaccine in the pipeline is going to be able to vaccinate the world. And so that's why everyone and all of their different products have to come together and kind of make this work. I think that people, all of the companies are doing their best to make sure that they're manufacturing ahead of time. So with the idea that there will be FDA licensure, and I think that Moderna has pledged 500 million doses or something, I don't know. But nevertheless, manufacturing is going to be a somewhat of a bottleneck, but it's I think people are doing as best they can.
Okay. Here's a question from the audience. Will a universal vaccine elicit a broadly neutralizing response, like broadly neutralizing antibodies?
Well, that would be the hope. That would be also why I probably will have a job even after this vaccine, if it goes to market, is because it's not the end, right? I kind of, at the VRC, I was raised under the universal flu vaccine program. And so understanding that universal vaccinology is what would be the ideal standard for any of the 24 virus families that can potentially infect a human. So yes, that would be the hope, but that's going to take some effort. We don't really understand what the cross-reactive epitopes are, blah, 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 blah. I think there might be a presentation by Andrew Ward, who actually collaborates with my team on universal coronavirus vaccine development, somewhere online. Neil King also. So you can look those two presentations up somewhere. Mm-hmm. But we do have that kind of matriculating in the background, and it's a good point.
Okay, let's do one more here, another upvoted one. Do you know, is there any research associated with vaccinating pediatrics candidates or pregnant women?
Yeah, so I'm slipping on the company, but I'm pretty sure I know actually there's a company, and I don't want to say the wrong name, but someone definitely has a platform that is going into clinical trial in pregnant women. As of yet, all of the phase 1s and phase 1/2s have been in healthy adults or elderly. So there haven't been any children vaccinated of yet, but there are platforms that people would just historically deem safe in children, and it would be the next place to go.
Okay, let's do another one. I like this one. Is there any concern using the spike protein as the target, with recent data indicating that it may be mutating or evolving? D614G is given as an example. What are your thoughts on that? And for instance, is this going to be something that's going to have to be like more of like a seasonal thing? Is there any possibility for that where you'd have to kind of update it, things like that?
Yeah, and so coronaviruses have a innate proofreading mechanism. And while they are RNA viruses, they don't mutate at such a rapid speed that we are concerned about a polyclonal vaccine response being evaded. There are tons of data suggesting that, when you hone in onto one particular part of the spike protein, you might get more escape as opposed to the full length spike protein. And we, lots of people with different vaccine candidates have tested their vaccine sera against the D614G mutation virus and shown that there is really nothing to be worried about. I think that we're good for a few years if we have a few years worth of protective immunity, right? So it's all about durability at this point.
Thank you very much. We're moving into a break now, unless Todd or Afshin, if you want to add any other questions to the mix. Otherwise, we're on about a 10-minute break here.
I'm just gonna ask one really quick question. Sorry, Leo, to Dr. Corbett. I'm just curious, what strains did you use in those experiments? The data was fantastic, and as we all know, the mouse strains are kind of limited right now with experiments relating with SARS-CoV-2.
Yeah, so that's why everybody should go to UNC and become friends with Ralph Barrick, like me. No, so we use Ralph Barrick's mouse model at the University of North Carolina, and this particular one, I think it's on bioarchives, but he mutated 2 amino acids in the receptor binding domain of the infectious clone for SARS-CoV-2 and made it susceptible to a wild type mouse. And so it has local replication in the lung and the nose, and so kind of like a human. So it's not like an ACE2 transgenic mouse where you can like have a mouse that dies from like brain or whatever problems. So that's the model that we used, and we actually really like it. And the reason why I know we don't tell this kind of story when we publish things because it's sloppy or messy or science. So the reason why we actually initially tested the 3 strains for immunogenicity is because we started at the same time that he started making the mouse model, and he didn't know in which strain the virus would take. And so it all came together for the good, I guess. So wild type BALC mice can have localized replication with the mouse adaptive strain of SARS-CoV-2.
Ashton, can I ask a quick question? It's Bruce.
Yeah, Bruce, yeah.
Great talk. Thanks so much for your work. Is there any information yet or a plan for immunizing people who become seronegative, had possible COVID? Do you have any signal yet? And what do you think is going to happen?
Yeah, I don't know. I do, hear the conversations, but I don't speak in those conversations because honestly, I feel like my job is a lot easier than having something on the market and deciding who gets it. So I don't think that there's a conversation around seropositivity yet in regards to who gets the vaccine because we don't know what a protective threshold is. Right now, I think everyone's thinking it's going to be a free for all kind of thing. Yeah, because as you saw with you can't say, well, you already had coronavirus, so you can't get the vaccine. Meanwhile, 1/3 of people aren't even mounting a antibody response after getting the virus. So you kind of just, until you have more information, it has to be for everybody.
Are you guys going to use ELSA spots for T cell antigen specific reactivity profiling too?
Yeah, so because we do flow cytometry, we generally don't do ELI spots. So we just do kind of the hardcore ICS 19 color thing a lot of times. And we have done, we have re-stimulated some T cells, re-stimulated some T cells from vaccinated animals, and done, like milliplex and looking at cytokine secretion that way.
All right, I think let's give everybody a chance to just take a couple minute break here before we start up the next round of speakers. Thank you so much, Dr. Corbett. That was wonderful. Thank you.
Yeah, go Rice Owls.
Yeah.
Thanks so much, Dr. Corbett. That was a wonderful talk. I really, really appreciate it.
Thanks.
Dr. Joyner, I see you here. We've got 2 minutes left, but if you want, you're welcome to go ahead and start getting your slide set up and everything.
I am here.
All right.
Let me figure out how to share my screen here.
Yeah.
Always the challenge.
Yeah.
Let's just see where I am.
And just a quick reminder to everyone in the audience. I don't think you need a reminder since I've been seeing a lot of good questions being asked, but there is a QA box down at the bottom of Zoom that you can use to ask questions, and you can upvote the ones that you want to see asked to the speakers.
Let's see, where are my? Do I use whiteboard or what do I need to use here?
No, do you see, when you hit share screen, do you see an option for your slides there somewhere?
I should, but I don't. Let's see. We try again here.
Sure. And in the meantime, I think I'll go ahead and give you an introduction here.
Fantastic. Okay.
Yeah, so our next speaker, everyone, is going to be Dr. Michael Joyner, MD. He's a professor of anesthesiology at the Mayo Clinic. Dr. Joyner's laboratory is interested in how humans respond to various forms of physical and mental stress during activities such as exercise, hypoxia, standing up, and blood loss. Dr. Joyner and his team study how the nervous system regulate blood pressure, heart rate, and metabolism in response to these forms of stress. They're also interested in how blood flow to muscle and skin responds to these stressors. These responses are studied in young, healthy subjects, healthy older subjects, and people with conditions such as heart failure. Welcome, Dr. Joyner, and I think it looks like you're all set up and ready to go.
Thank you very much. And so how did a nice exercise physiologist, environmental physiologist like me get invited to a party like this? That's always the question. So we'll start here and just talk about what's happening in terms of the expanded access program for convalescent plasma in the United States. So the overview of the talk is straightforward. What is convalescent plasma? I think everybody here knows. Talk about use of convalescent plasma to treat COVID-19. Focus on our expanded access program. Give you some insights on clinical trials that might be out there or should be out there. Lessons learned so far. Is it safe? And give you some preliminary insight on what we think in terms of efficacy, and talk briefly about future directions. So everybody here knows about passive immunity. At some level, I think convalescent plasma is the kind of first best shot on biological goal until you get either hyperimmune globulin, monoclonal antibodies, or obviously a vaccine, transfer of antibodies from a recovering patient or animal, post-exposure to prevent disease, and also to treat disease. And this is really what we're focused on. Again, many people in the audience here know the long history of this sort of therapy. Early use with plasma because that's what we have. And then ultimately we hope to shift to a concentrated hyperimmune globulin preparation. Again, the conceptual model is straightforward. You have a COVID patient who's recovered, do a blood draw, virus neutralizing antibody, serum or plasma, and you can use it for prophylaxis or therapy. This is a classic example of a use in 1918 in the flu pandemic then. I believe this was a naval officer who started getting convalescent sera, and you can see the defervescence here. Tom Luke, who's doing some very interesting thing with humanized cows and convalescent monoclonal antibodies, did this wonderful meta-analysis of everything you could find on the 1918 flu epidemic and showed that convalescent blood products really made a difference. And overall, the treatment group that he was able to identify, only about 16% or 54 out of 336 died, whereas about 37%, 452 out of 1219 died in the individuals who did not receive plasma. Many of you know about Argentine hemorrhagic fever. This is perhaps the best example and where there's been probably the best randomized clinical trials. The point is that the case fatality rate among cases treated with normal plasma is 16.5%, while the rate in those patients treated with immune plasma was 1.1%. So I think this is probably the most dramatic and best characterized example with potential relevance to what we're talking about with COVID-19. SARS in the early 2000s, some evidence that people that were treated earlier got out of the hospital faster than people who were treated later. This also shows the challenges of doing this sort of work, because by the time you can get the protocol set up, the IRB approval done, and the convalescent plasma collected, a lot of times the pandemic or the outbreak is waning in that spot. And again, that remains a challenge today. So has it worked in the past? Yes. Many examples of prophylactic use of various forms of passive immunity. Early use seems better. What about rescue therapy? And we've certainly seen a lot of that with convalescent plasma in our expanded access program. And again, concentrated products follow plasma. So how did a nice guy like me or an innocent person interested in hypoxia get involved in something like this? Well, I was made aware of it via Twitter in the Wall Street Journal based on an op-ed that Arturo Casvidal published. I then raised awareness here at Mayo locally. There was an emergent network that really sprung up around Arturo, but Arturo and I had been friends for a while, and so part of that network was already sort of going on. We began to engage the Food and Drug Administration in March. We worked closely with Peter Marks to develop the EAP program. Again, there are a number of RCTs going on, and I'll talk a little bit about the results so far that I can share. So this is Arturo's February 27th Wall Street Journal article. Arturo is a friend of mine, so I emailed him. And the main thing I added to this as an anesthesiologist is I pointed out that the blood banks, the blood collectors, collect around 30 to 40,000 units of red blood cells a day in the United States. And to me, at least, that indicated that there was substantial infrastructure for potential collection of this product out there. Now, would it be easy? Would they just be able to collect it as they always have? No, but did the chairs in the blood banks exist, provided you could fill those chairs with convalescent patients? I think the answer was yes, and that was really my contribution to Arturo's initial insights. And this is the summary. So here's the timeline. We on the 30th of March, shortly after original early use in Sinai and Houston, the FDA contacted me about an EAP. Why Peter Marks emailed me, I do not know. I look forward to meeting Peter face-to-face someday and ask him that question. My family also looks forward to asking that question because I've been working pretty hard and haven't seen much of them recently. The IRB approved this. We have a rapid IRB turnaround time, and we were able to be the central IRB. These were really critical. We then began with our internal website teams to develop a rollout, which included really a completely electronified process, which allowed sites, MDs, and patients to enroll, develop some simplified workflows, develop really a navigator process where people could email us and we'd give them a call back to help them, develop case reporting tools via REDCap, and really, like I said, a full service communication center. So on the 7th or 8th of April, our first patient was transfused. As of yesterday, we had transfused 61,000, or enrolled 61,895 patients, and we have complete records on 39,388, 2,627 sites, 11,000 physicians. There's a lag between enrollment and reporting. We believe about 85% of these people have been treated. But as this second wave began to emerge, there's some really big time reporting delays from overwhelmed facilities and physicians. We're trying to understand that, trying to simplify our case report forms and make it easier for people to get those updates to us. But this is one of the real challenges we're having right now. And it's a little concerning to me as somebody with their name on 62,000 consent forms. So we're going to try to figure out how to do a little bit better job on that in the coming week or two. You can see that we're all over the United States. This animates up, and you can see as things just sort of take off from here. And you see the big spike in New York City area now, and you can see things really growing down south. This is what it looks like as of yesterday. So what do we know about the cohort? And this is 27,425 patients transfused prior to June 24th. We want to do that because this has given us time to really focus on this and do some quality assurance and drilling down and data curation. You can see most are middle-aged, 60% are men, a lot of obese patients, 19% are African-American, and 36 or 37% are Hispanic. So we have excellent diversity here. You can see that these are pretty sick patients. About 30% were mechanically ventilated, 55% in the ICU. About 70%, 68% were characterized as having, severe or life-threatening COVID. And you can go down these respiratory markers, but the thing I find most interesting, as an anesthesiologist who certainly in the operating room occasionally takes care of patients who come down from the ICU, is 13% have multi-organ dysfunction or failure, and about 11%, 10.7% were septic. So I think it's fair to say there was a whole lot of salvage therapy or rescue therapy going on in our cohort, and I believe that continues as well. So our charge was to provide broad access to COVID-19 convalescent plasma, perform a robust safety analysis, and to explore efficacy. So what do we know about safety? This is a paper in the JCI in the first 5,000 patients published, I believe, in May. Transfusion-associated circulatory overload was extremely low. Transfusion-related acute lung injury also extremely low. And allergic transfusion reactions were extremely low. These are among the lowest incidence of these plasma-associated transfusion problems that have ever been seen. And we've really been shocked by this. We've looked at this in up to 20 or 25,000 patients, and these rates remain very, very low. So I think in terms of both safety and access, we've demonstrated through the EAP that this product is safe to give, especially when you consider just how sick our patients are and the difficulty in making a taco or trolley call in somebody who's mechanically ventilated with 3 or 4 medical problems. Now, the initial plan was to take a stab at efficacy. Perhaps we thought, frankly, we were going to enroll. Our original IRB approval was for 5,000 patients. We thought we would then do some sort of post hoc matching and this sort of thing, and in the third or fourth quarter of 2020. And then we thought we'd build a biobanking and start looking at things like neutralizing antibodies later this year or early next year. But obviously, as enrollment took off, we wanted to know if we could do this a little bit faster. And also, there was great interest from various arms of the U.S. government about whether we could do it faster. And potentially, this is not a randomized trial, extract some efficacy signals out of this. So again, here's my disclaimers. We gave a guesstimate dose of an uncharacterized product to all comer patients, disease stages least likely to show a positive effect if you look at the old data on the use of serum to treat pneumonia from the 1920s and 30s. We gave it at any hospital, teams with no training, and many elements of the plane we are flying were built while flying it. So nevertheless, potential signals of efficacy embedded in the enrollment and safety data, the volume of plasma administered, not all units are 200 mLs, it's variable, and some patients got 2 units. The time to treatment from diagnosis, we think we have a pretty good handle on that. Comparison to other cohorts, and then exploratory analysis using neutralizing antibody titers. And all of these things, volume time and neutralizing antibodies, potentially have elements of randomization and placebo control, or at least a dose response relationship. So we started to get almost immediately stories like this, what I call the Lazarus stories, people that got better fast. This is a 58-year-old guy who was on 60 liters of O2, careening toward the ICU at the University of Wisconsin. They needed to think about it, so he ended up getting intubated, but they gave him the plasma shortly thereafter, and he was extubated rapidly. We hear a lot of stories like this. They're rife with confirmation bias, but they are, in fact, interesting, and the timeliness of them really makes you wonder. This is data from China. It's a randomized clinical trial that was suspended for lack of enrollment. These are the improvement rates, convalescent plasma versus control. But this is when you take the least sick patients, non-ventilated patients, you get a big improvement versus those with life-threatening disease. This is the Mount Sinai case control study. Again, most of these people weren't mechanically ventilated. You see this coming down, and you see these are the treated patients, 4-to-1 matching. These are the untreated patients. So again, the sort of signal you saw in that flu data I showed you earlier. This is something that Arturo Casvidal and I developed, where we looked at the first 3 are basically randomized trials. The first 2 were stopped earlier. This one from Iraq was finished. These are case control studies, all of them in pretty sick patients, except for Zia et al., who were a large number of less sick patients, not too many on oxygen in China. And you can see that the mortality difference is pretty striking in every single trial, and the average is about 40%. If you take the Z data out, the chi-squared on this, on almost all of these, is positive and trending toward positive in the RCTs. So I think, again, this is at least helpful evidence, or evidence that makes us optimistic. So are you absolutely sure? No, we're working on detailed outcomes analysis that the FDA and BARDA have related to time to transfusion, the volume of transfusion, and titers in plasma versus outcomes, and in the comparison toward other cohorts. Again, this analysis is done. It's currently being embargoed while it's discussed at the US government. But I can tell you, based on what we have seen, that the ideas about using antibody therapy from the 1920s and 30s for respiratory diseases indicate that early administration is important. And I believe that our findings will, in fact, reinforce what was common knowledge before World War II. Remember, things are complicated. And they work together, and we don't always know exactly what's going on. And we believe there's a lot of emergent properties in convalescent plasma besides antibodies. And there are clearly people who recover with very low antibody titers. So I think that people can get into a little bit of white whale chasing about antibody titers equal efficacy. So cast of characters from the outside, Mayo folks. And what's next is questions. Thank you very much.
Thank you very much, Dr. Joyner. So we don't have any audience questions yet, but I actually have a couple comment internally to me here. One question from a grad student here, Nick Sapavel, is talking about how would this work as far as like an international collaboration? So if you have, some nations that are just now recently getting hit, and so maybe they don't have the antibodies yet, how would that work exactly? Like collection, distribution of plasma for,
right.
for these? Yeah.
Theoretically, very easily, but the problem is blood banks are regulated at the national level, and there's, in general, it's very, very difficult to send blood products across borders. Not impossible, but difficult in any bulk, and that also includes, to some extent, to refined HIG products. That's a little less problematic, but so in general, this is typically going to be a local or regional solution. I don't know what the rules are in the EU, but again, I think it's difficult for them to ship blood to us and us to ship blood to them.
Okay. There's a lot
Leo,
of,
is there
yeah.
Can I ask a question?
Yeah, go for it.
So this is Leon Alkalite, JPL. I'm just curious, and I'm not a physician. Is there any matching or compatibility aspects to the transfusion, or is everybody equal here, or
no,
gender?
Same. They got to be ABO compatible, just like a regular plasma donation,
Okay.
transfusion. But again, that's old hat for the blood bankers, and it's not been a barrier at all.
Good news.
Yeah, I also had another internal question coming in asking about blood type, if there's any other thoughts related to blood type.
No, I mean, other than making sure we have enough donors, so we have a wide inventory of all sorts of types.
Yep. And
hey, Leo.
Yeah.
Could I?
Yeah.
Bruce, you had a comment in there. I don't know if you want to mention it, since you have mic privileges to Michael here.
Sure, thanks. Okay, so Yeah, fantastic work, incredible impact. There's several angles here. One is, Jeff Ravitch's immunoglobulin subtypes, the glycosylation state of those, the IgG3. Is there any signal in there for differential effectiveness?
So this is let me give you a kind of a high level answer to that. So I think this is a homebrew product, and that people collect. And I think there's a lot of interest in what else is going on besides neutralizing antibodies. And I think that for example, spike proteins. And I think there's a huge need for basic and translational research on what else might be happening, because about 10 or 15% of people who recover from fairly severe COVID infections don't have a lot of neutralizing antibodies. In fact, 5 or 10% don't have any. But they're clearly protected. They clearly fought the disease off. They've clearly recovered. So, I call these people the left-handed recovery people. So what are these left-handed people doing? So I think that's really, really important.
I actually, to follow that up, I think, initial responses, you get out of the ICU, you go home, you're not on any supplemental oxygen, big success. Sorry, it's not over yet. I'll talk about the long haul stuff in a little bit, but the fine structure of the outcome, exactly how much do you recover? Is there a difference between those individuals that don't?
Yeah, no, that's being looked at. Correct. But I think the other thing that's interesting, we had a talk on a cardiology grand rounds. I did not attend, but people were sending me slides and telling me about it. And so our pathologists who are expert in other forms of respiratory infections, flu and regular pneumonia, say that when you look at this looks just, of course it isn't the flu and it isn't pneumonia, but they say the sort of systemic responses that people get that eventually kill them do have a lot in common with the flu and horrible cases of flu and pneumonia. And as you know, that people, the recovery from the flu or pneumonia is serious enough to get you mechanically ventilated and spend time in the hospital. It's not a linear, straightforward thing. It takes a while, and there's a lot to it. And in addition to the recovery from the infectious disease and the inflammatory process, and bed rest itself is a tremendous problem. And I know there's some people, that have some links to the space program here, and that, that's a huge. I mean, I've served as an advisor to NASA on countermeasures for long-term space flight, and just the inactivity, lack of weight, the weightlessness, loss of muscle mass, loss of bone mass is a huge problem, huge challenge.
Yeah, I think we're gonna have to. So I'm sorry to interrupt. I think we need to get to the next speaker. We're running over just by a minute or two here. Thanks.
Okay. Thanks so
everybody.
much.
Thank you, Dr. Joyner.
I wish I could stay for the whole thing, but I got to go talk to the FDA.
Yep.
See you later. Thanks so much.
Dr. Alcali, are you ready?
And
All right. Feel free to go ahead and share your slides there. So our next speaker is going to be Dr. Leon Alcali, who has been at JPL for 31 years since getting his PhD at UCLA in 1989. He's currently a tech fellow of the laboratory and manager of the Office of Strategic Partnerships. He's currently managing the JPL ventilator project, which was developed at JPL in response to the COVID-19 pandemic. The technology has been licensed to 28 countries across the world, including Brazil, India, Canada, Australia, Mexico, Indonesia, Nigeria, and many more. Welcome, Dr. Alcali.
thank you. Thank you very much. It's a pleasure to be here. Do you see me okay? And do you see my, or not me, do you see my slides okay?
Yeah, looks
good. Can you hear me fine?
Yes.
Wonderful. Well, again, thank you for inviting me. This is a brief overview of a recent project at JPL. And when you normally hear about JPL, you don't think about ventilators or medical engineering or medical devices. So this is a kind of an odd activity for us, but one that has generated a lot of enthusiasm, a lot of energy, and hopefully will make a difference in this fight against COVID. So this is just a brief slide introducing JPL. If you haven't been or seen or know about JPL, it's one of the NASA 10 field centers, of which it's the only FFRDC, federally funded research and development center managed by Caltech through a contract with NASA. We have a pretty sizable business base. We are focused on robotic space exploration. That's our mission for NASA. Over 6,000 employees, and pretty much a lot of it is just specialized lab space to implement all these great robotic missions. And I give this one slide here of the next Perseverance or Mars 2020 rover, which is actually launching in just less than 2 weeks on July 30th to Mars. And I'm just giving this because of its relevance and timeliness, but it's really what we do at JPL. We build these kind of rovers. This is a nuclear-powered rover, mobility system, onboard intelligence, many instruments. It will actually, for the first time, demonstrate a number of things on Mars, including in situ resource utilization, generating oxygen on Mars from the atmosphere, and many other things. It can rove for many tens of miles and so on. And so this is where we're coming from. That's our kind of day-to-day job. And then we got kind of brought into this world of COVID, and it all started at a very grassroots level. And I'm just summarizing the whole story here, is that a few engineers got together, said, why can't we just put something quickly together to help the world? And the vision at that time was quite simple. Let's put together something that everybody in their homes can build. A ventilator that they'll get parts from Home Depot. They can go online, download some design drawings, and make it yourself ventilator. That was the original, somewhat naive, but kind of a vision to help the world, because we were hearing all kinds of stories from Italy and all over that the shortage of ventilators was so dire and so kind of resulting in deaths that the more important thing was to just increase the volume of production of these devices. So we started by calling the doctor, and we got in touch with Dr. Michael Gurevich from Huntington Memorial Hospital here in Pasadena. And March 16th, we kicked off the first study, first just dialogue with an expert, and then as I show here in a fast summary, within 37 days we had built 2 ventilators and demonstrated at Mount Sinai Hospital in New York City. Also had the opportunity to share this and demonstrate it to the President of the United States, all within 40 days. And then we issued a worldwide license call, which I'll go through in more detail. This is how the ventilator looks like. This is one of the 2 ventilators. This is the compressor ventilator. There's also a pneumatic one. I'm not going to go through the movie, the video link here. You can go on YouTube and just search for NASA ventilator in 37 days. That was the title given to the news release and so on. But we've licensed this to, now the number has grown to about 30, just to correct a little bit, 30 companies across the world, of which 8 of them are in the US and 22 are in different countries, 5 of them in India, 3 in Brazil, and so on. This is our team. This is actually a team picture. The whole project was executed, at home, except for about 10% of the people who had to be in the lab to build the actual ventilator. So this group was quite large at times. I frankly did not know a lot of the people, excuse me, who were on the project, but we all executed this remarkably efficiently, perhaps more efficiently than any other project that we normally do, even without COVID. This is the leadership team. I'll just go through listing, showing you that these, at the top of the project, we had Roger Gibbs as the project manager. I was in charge of all external relations and partnerships, and also part of the leadership of the the project management and so on. The only person that's missing here that is on the leadership is Rob Manning, who also is the laboratory chief engineer. And you see also Michelle Easter, you'll see here, she was in charge of the test bed. Mike Johnson was our lead engineer. David Van Buren, Stacy Boland was in charge of medical operations and liaison. RB Karapetian at the bottom right was in charge of production. Let me go through a few of the key elements of the strategy, which is really kind of my day job, and we had to formulate this rather quickly. And this is also a strategy that is also looking back. It's not like this was all known at the beginning, but it kind of evolved very quickly. As we were not experts in this field, but we wanted to provide an engineering and a technical solution, we immediately had to seek advice from experts, medical doctors, healthcare providers. We reached out to experts at Medtronic. We reached out to Mount Sinai Hospital, UCLA, USC, other hospitals, Mount Sinai, Huntington, and so on. We realized very quickly that we need to go into a rapid design and prototyping phase. So as ideas came up, we were breadboarding and prototyping. We realized we had to partner with other government agencies. I'll go through that rather quickly. Early on, we decided that we cannot go through this process without getting FDA approval. And whether it's the licensee or the partner or JPL, we knew one way or the other. And actually, we called a few hospitals and we said, if we build this, will you use it? And they said, if it doesn't have FDA approval, doesn't matter what you build, we won't use. So that kind of differentiated us immediately from a lot of open source ideas that were not going to go through FDA approval. So we immediately backed out and said, we're going to go through FDA approval. We're going to build something that hospitals and doctors want to use. It's not going to be the simple Home Depot kind of a ventilator that anybody can build. It's got to address real requirements and real needs. And so we resorted to a lot of JPL established processes. We started with defining the requirements. We had our doctors sign off and say, yes, if you meet these requirements, we'll use it. And then again, in a JPL standard way, we went through a brutal series of peer reviews, technical, medical, regulatory, operations, manufacturing, supply chain. All of these were peer reviewed, which included both internal and external reviewers. We immediately also realized that we can't interfere with the existing legacy ventilators. So we had to stay away from all the supply chain that involves any existing ventilator. And we were able to do that. And finally, we had a big decision to make between open source and licensing, and we went for the licensing approach, which I think was a really critical and very good decision that we had to go through, and I can certainly talk more about it. This is just showing Dr. Gurevich standing here in the center, talking to Dr. Matt Levin from Mount Sinai. We use some of our existing capabilities. We have roaming robots in our laboratories. And you see here on the left, Matt Levin on the computer, and you see towards the right, on the right-hand side, just in front of Mike Johnson, you see Stacy Boland, who was also from LA on the robot and driving around, looking at the different aspects of the design, all kind of huddling around the design and testing things and so on. The key attributes of the patient is that this is an adult, is either sedated or intubated, so we're sedated and intubated, and relatively stable vital signs, and yet in need of a invasive ventilation due to their COVID condition. These are some of the requirements. Frankly, there's a whole document of specifications that the doctors were telling us. And they were, I would say, aggressive in the terms of PEEP values and oxygenation or oxygen fraction use. and tidal volume and so on. So we took these as starting conditions, and quickly, within 2 weeks, we designed. Mike Johnson was the lead for this design, and then from the drawing board to functional design, and immediately to breadboarding. Here you see on the right a high-end lung simulator that we actually had to borrow from a facility outside of JPL. It was, even for JPL, it was too expensive for us to buy our own lung simulator. Always we had the doctors in the loop telling us what's wrong and what needs to be changed. We got it to work. That was pretty historic moment. And then we had, especially in this case, Michelle Easter was very critical in managing the test bed that we were developing. And in particularly, you see here also the team that had to deal with the production. So we had a tag team between the prototyping group that was building, and then as soon as they source parts, they call the production team, and the production team would say, wait a minute, that part is being used by such and such ventilator. You can't use it. We have to find a different part. And or there would be a case where a part would be available, but not available in volume production. Or there's other factors that would limit it. So we had to go back and forth between prototyping, supply chain, production, to come up with a design that will be not FDA approved, but have the capability to be manufactured by our licensees. We were very, very excited when we sent this over to Mount Sinai, and they were able to replicate our curves and the graphs on a simulant. This is a high fidelity dummy patient that they were able to recreate the performances. We have a matrix of all the configurations of pressure, volume, timing, and this is a patient-assisted device, so it detects the patient breathing, and it also has software to avoid breath stacking. So it's a pretty complicated device. This one is a pneumatic design. The other one is a compressor design. And we also sent it afterwards to Mount Sinai I'm sorry, this one, both are Mount Sinai. And then after that, we sent it to UCLA, to the laboratory of Dr. Tisha Wang. They also did an independent assessment and testing of the ventilator. And then we went out for worldwide licensing call. And we were, when we issued that call, we were just astounded by the response we got. About 300 companies around the world went onto our website and registered. We received about 100 proposals, and out of those, we down selected to a manageable, at this point, 30 licensees. And here they are listed here. Since this, we've added another one from South Korea, and forget which other country, but we've added we have a total of 30 licensees. And just last week, on the 10th of July, we shipped one to India, and this is at one of the licensees at Bharat Forge in Western India. That they actually.
Dr. Aklai, I'm sorry to interrupt. You got about 2 minutes left. I'm almost done. Thank you. This is Bharat Forge in India, and there you go. I'm done. All right. All right. Yeah, thank you for that. I've got a couple internal questions here again for you. One question: how rapidly can manufacturers switch to producing the new ventilator devices? Are there any additional requirements for potential third-party manufacturers?
Yeah, so we've once they've been approved for the license, then they have access to our server that has all the details of the design, everything from parts, boards, software, hardware, you name it, they have it. So I don't know exactly when the first will occur, but I think we're looking at a period of a few months before the first manufactured ventilator is actually going to be recreated.
Yeah, thank you. And another one, have there been similar supply problems with the high flow nasal cannula? I hope I'm pronouncing this right. Cannula devices. And do you think your model of rapid manufacturing can be replicated for HFNC?
So we're looking very, very carefully. We've had a series of lessons learned on how quickly we did this, which really stunned JPL. The first question that JPL and management was asking is, why can't we do our Mars missions like this? Why we, if you can work so efficiently, why can't we do everything like this? And it doesn't work that way. Certainly the pace is not sustainable. People were working 15, 16 hours a day, 7 days a week. There was no weekend, no holiday. There were some consequences, people were exhausted from this kind of pace. So it's not easy to replicate. What we've actually been doing is picking out real lessons learned in terms of what are things that can be replicated from a management point of view. Also from a topical point of view, are there other things that JPL engineers can take on as challenges to solve, not necessarily COVID related, could be other areas. And that's certainly something that we're very interested in doing. I know that Mount Sinai Hospital was particularly interested in continuing other opportunities with JPL. In terms of the first part of the question, we've made sure that the interfaces from the ventilator to the patient are all based on standard tubing, standard interfaces, HMEs, filters. So there's nothing non-standard about the interfaces between the ventilator and the pneumatic or oxygen or tubing into the hospital existing equipment. So from that point of view, it's standard.
Yep. All right, thank you very much. Well, with that, it's time to turn it over to the lightning talks. So thank you very much for your talk. And yep. There we go.
Yeah.
And yep, Afshin, thank you. Just say thank you again for organizing and running these segments. So I'll hand it over to you.
Thanks, Leo. Thanks, Leon. That was great. So these segments are just going to be quick snippets of what we're doing within the COVID International Research Team, Covert. So just to give people a little preview, and any audience members or any of the panelists here and speakers want to collaborate with us and join our group, we'd be more than happy to include everyone, since we're all about open science and including as many people as possible. So I'm starting it off. Let me switch to my slide here to discuss the mitochondrial subgroup. So the mitochondrial subgroup, it's going to be really quick overview. So these are, again, reminding from the introduction for people who might have missed it. These are all our subgroups in Covert, and I'm focusing on mitochondrial, and throughout the day, you're going to hear about a lot of these subgroups, not every subgroup. So these are the people involved with our subgroup, and how Covert works, it goes in and out of people coming in and out of meetings and groups, and a lot of cross-pollination between, overall different subgroups. So what started us looking down the path, we started looking at this public database from Wuhan and doing all the omics analysis. I'm not going to get in the nitty-gritty. We noticed the suppression of mitochondrial happening. And what was interesting is when you started looking at the literature, why, let's say, the hardest hit populations, for example, or symptoms that might occur, like in this case, loss of smell and taste, as we all know. Well, that's heavily related to mitochondrial suppression and calcium signaling being increased as we see. Another, population that's hit, of course, is my slides advance. There you go. Or the elderly, right? And if you know, with age, mitochondrial decreases. So again, if your mitochondria decreases and you have that extra hit, the elderly, of course, going to get hit more. Obese and other things and so on. So every of these targeted or the more susceptible people that are getting targeted by COVID tend to have mitochondrial dysfunctions occurring. So the virus itself, when it comes in and does further mitochondrial dysfunction, mitochondria is involved with all your bioenergetics and energy. Of course, these people are going to get hit harder. And we have now further data from Chris Mason's group, who's part of our group. He has this large nasal swab RNA-seq data from COVID positive and negative patients. I'm not going to get in the nitty-gritty here, but these are the people involved analyzing Chris Mason's data. And with viral load, we see, again, impact of mitochondrial suppression happening. And interestingly enough, the mitochondrial genes that are being upregulated are upregulated because they're trying to overcompensate for all the downregulation suppression of mitochondrial genes and proteins. Again, so we have a proteomics expertise here with Hossein. Hossein from CHOP, he dove into the proteomics literature. One of our speakers coming up, Kristen Mooch, actually had a really beautiful paper looking at proteomics in SARS-CoV-2. So he started dissecting all this. And again, the theme pops up in proteomics. When you really dive into the mitochondrial, we see suppression happening in mitochondrial, and there's heavily things related, like glycolysis, for example, is related to mitochondrial suppression being increased. And then we also have people in our group from Doug Wallace, who's basically the founder of all this mitochondrial genetics. This is from him. And he has, when he dove into like looking at SARS-CoV-2 and how it relates to virulence and with mitochondrial, as you can see here, I'm not going to read this, and you guys can read it yourselves. You see that indeed, a lot of the symptoms, a lot of the facts of COVID-19 or SARS-CoV-2 viral activity is heavily related with mitochondrial dysfunction and suppression, including from inflammasomes are tied in there and pathology and so on. So he has some ideas actually to be, potential ways to combat it with mitochondrial dysfunction in mind. These are some of it here. And what's interesting is another paper that was released looking at protein interaction with the genes, that was by the Krogan lab. Doug went in here and dove in and looked at the different regions of SARS-CoV-2 with the, the NSF and the ORF regions. And indeed, you find that about 50% of the SARS-CoV genes are related by bioenergetics or the mitochondria. So it's interesting. And when I dove into like the nasal swab samples from RNA-seq from Chris Mason's lab, and then compared the genes that might overlap from the genes that are targeted by these different regions, indeed, a lot of these are suppressed, and the ones that are activated are expected to be activated when there's mitochondrial suppression occurring. So again, there's that theme of mitochondrial suppression overall happening. Again, I'm going through this really quick. It's a 5-minute lightning talk. So, and these are some strategies, some really quick strategies that Doug, is thinking of tackling it and looking at mitochondrial suppression and how you might use that to, combat COVID-19 infections that are happening. And it's a host response here. So maybe it could be used as a therapeutic to combat all the, damage that this virus does. So this is some support that I had personally, and there's other support from all the other members from NIH and other things that I didn't have time to include. But I have time for about 1 minute of questions because I'm right at 4 minutes and 30 seconds. So I'll stop sharing my screen, and this is a quick overview of what's happening. So if anyone's interested in mitochondrial suppression or mitochondrial activity and want to collaborate with us and help us out and have ideas, I'd be more than happy to talk to you after this. So is there any questions? Let me go to the question panel. Sorry, switching over. I don't see any questions. I don't see Leo. Did you see any questions that might? Oh, there's a question here from Rob. Okay, any evidence of SARS virus changing the mitochondrial genome? It has been reported in bacterial infections. So that's what we're looking into, and there seems to be that SARS virus itself, as Doug had dove into those papers, and we've been seeing with our analysis, definitely it impacts the mitochondrial expression, but the genome I haven't heard of too much yet, and there might be someone looking into that. And one of the issues is that a lot of people ignore mitochondrial, so that's why there's not much information out there. And I think, as Doug would say too, that that might be a mistake. We need to start with the host response, at least concentrate on the mitochondrial impact. So any other questions, please email me or on the Q&A. So we're going to go on to the next person. Quick lightning talk. So the next person is Paco Inguanta from University of Lisbon. Paco, you there? He's going to talk about long non-coding RNAs in our microRNA subgroup. I think you're on mute, Paco. I don't hear you right now. Oh, Paco, I think you're on mute. Maybe we could try to unmute you. Let me see.
I'm here. Sorry.
Oh, there you go. There you go. Okay.
You are right.
Okay,
you are right. So sorry, everybody. Okay. So
no problem. We're gonna start the timer now. So go ahead.
I'm going to share the screen. Can you see this? Okay. So
yep.
For you, if you don't know me, so I'm assistant professor in the medical school in the University of Lisbon and researcher at the Institute of Molecular Medicine, also in the same university. And basically, I'm going to represent the subgroup of the non-coding RNAs in the Covert consortium. So I'm going to tell you something about a specific kind of non-coding RNAs. So as Afshin already pointed out, so we are dealing with this amazing data set from Chris Mason, So we have roughly 700 samples of patients, and more or less 50% of them are infected. So we classify the samples by using the qPCR data in the low, medium, and high viral loads. And one of the most striking conclusions when we analyze the RNA-seq data is that the infection levels induced a transcriptional shift in the patients. So you can see that the amount of upregulated transcript increases as much as the infection load. So when you look at the kind of transcripts that are induced by the infection load, you can see here that there is a significant amount of the transcript profile that it's encoded by non-coding RNAs, mainly by long non-coding RNAs. For you in the audience that don't know about long non-coding RNAs, I have to tell you, and here you have data from the GeneCode database, that these guys, we have roughly the same amount of long non-coding RNAs than protein coding genes in our genome, and these are very versatile regulators. So they are expressing typically very low levels, and they are very important because they can constitute a very versatile toolbox for the regulation of gene expression. Why? Because these guys can interact with many partners, so they can interact with DNA, with RNA, and also with proteins. And they can do many things. So they can signaling, they can decoy, they can guide, and they can also do scaffolding of molecular complexes. So they are involved in everything that you can imagine. So starting from the chromatin to the cytoplasm. So you can see here some of the regulatory functions that these guys have. And this is because we were interested. And when we look at the profile of differential expression of these long non-coding RNAs, and we look at specifically of to the most differentially expressed, and you can see here the samples that are infected, and this is the viral load. So you can see here clearly that there is a striking pattern of upregulation of some of these guys in the infection by SARS-CoV-2. But also you can see here in the orange group that you can see these guys also upregulated in other viral infections. So these are common. Some of them are common. So here you have the dispersion levels by groups, and you can see the dispersion of the data. You can see that some of them are really overexpressed and they are correlated with the viral load, but they are also overexpressed in other viral infections, but with a low level. We also determined that some of these long non-coding RNAs are correlated in their expression. So you can see here the heat map of the Pearson correlation coefficients, and you can see here one cluster and another one cluster of these long non-coding RNAs that are correlated in the expression. One of the main problems to find the function of these long non-coding RNAs for therapeutical purposes is that the majority of them are not well annotated. So you can see here these very weird symbols, so meaning that these long non-coding RNAs are not well annotated in databases. So we started basically to look at the ones that were upregulated in SARS-CoV-2 infection and well annotated. And here you can see something that we came out, which is sort of a functional analysis of long non-coding RNAs. And as you remember, I told you that these long non-coding RNAs can act as a scaffolds of molecular complexes. And one of the things that they do typically is to bind RNA binding proteins. So you can see here the profile of binding to RNA binding proteins extracted from experimental validated data of some of these upregulated and annotated long non-coding RNAs in the SARS-CoV infection. Of course, we are also taking advantage of Christian Munch data, and I'm sorry for the spoiler, Christian, because he's going to talk after that. So, and you can see here that some of the RNA binding proteins that are bound or are experimentally validated to bind these long non-coding RNAs are also upregulated in SARS-CoV-2 infection. So meaning that they are co-regulated, so they are correlated. Very interestingly, there are some long non-coding RNAs here that are nice. So here, this one is also has been described in hepatitis C virus infection as something that the virus needs for infection. Here, these ones have been described in influenza infection, and these ones are related with the splicing and with RNA transcriptional regulation, mainly because they bind some of the AGO2, AGO3, and AGO4 proteins that are related with post-transcriptional regulation. So basically, I hope that you get convinced that these guys are important because we find these transcriptional shifts. We have determined these upregulated long non-coding RNAs, and we can find that we can see RNA binding proteins bound to these long non-coding RNAs. So the idea is to find a functional relationship with these long non-coding RNAs and RNA binding proteins, and try to at least find something that is important for the virus. So, and this is my last slide. So I want to thank everybody, Afshin, and all the members of this subgroup of the non-coding RNAs, and thank you for your attention. And I'm happy to get one question if you have. If not, please be free to email to this email, okay?
Yeah, thanks, Paco. I think that was great. We don't have time for your question,
Okay,
but
it's okay.
so if someone has a Q&A question, you could respond there too. You know,
Okay,
so
okay, cool, okay.
that might
okay.
be the best way. So thanks.
Thank
And
you.
then a quick snippet now for Krista Turnus, who's the lead for the microbial subgroup, and talking about the co-infections. So I'll let her take it away.
Great, thank you. All right, hopefully you can see my slide and hear me. Thanks for letting us tell you about the work we've been doing in the microbial subgroup. Listed here on this slide are all the different members of our subgroup. And thank you so much to all the volunteer efforts that they've been giving for this activity. Without the expertise and the time that everyone has spent, we would not be where we are. And I also wanted to thank the OSF for the free project storage space they've given us to share files amongst the team, and TAC for all the computational time that they've given us to be able to analyze many different data sets. So thank you all for your contributions, and we're really excited about this work, and we believe understanding how the microbial community changes in response to COVID-19 disease can help us better understand the disease progression and severity, and also give us some better understanding of other therapeutic options and diagnostics. Today, I just wanted to quickly share about some work we are actively doing to analyze public metatranscriptomes, specifically from bronchial alveolar fluid or BALF samples from humans who are sick with COVID-19. So they have the disease in different stages, and there aren't very many of these samples available, but they are very valuable in that this is essentially lung fluid where we would expect to see the virus to be very active. So we would anticipate seeing a large degree of viral transcripts in the lung fluid, as well as human response in the different transcription levels of the human genes, and then changes in the microbial community, potentially because of the activity of the virus in that tissue. All of the data that we've been doing is open source and available at the link provided on this slide. So feel free to check that out. It's also part of the coronavirus outbreak research collection that OSF has put together. And pictured on this slide are the different samples that we've analyzed from COVID-19 patients. So each line represents a different sample, and the colors are different taxonomic groups from the sample. So yellow is the SARS-CoV-2 virus, green is human RNA, and blue is Phi-X, or the Illumina sequencing control, so essentially a contaminating organism within the sample. And one of the challenges that we're having in analyzing samples from many different studies is that they were all prepared in different ways before sequencing. So there are a lot of different factors to account for there, and we are actively trying to consider all of those different aspects of the sample prep and how that influenced the taxonomic classifications and functional annotations we're getting on the back end. But you can see here that depending on the author's goals, they may have preferentially amplified the virus or the human RNA. But the black bars, or the bars that are not colored, are the remaining microbial pieces of the data set, which is particularly what we're interested in. And as we look at those remaining microbial pieces of the RNA sequences, we are taxonomically classifying them with Kraken 2, and that is a a leading classification algorithm, and we have a nice large reference database that we're using with it. But these classification approaches are limited right now in that if the sequence doesn't match to a known reference genome, there's not much else you can do as far as taxonomic classification goes. And in this figure, we see that the blue bars represent sequences that had no match to the reference genome. The yellow bars represent ones that were classified. And typically, if you're doing a taxonomic classification workflow, you throw out all the blue bars at this point. So there's nothing more you can do. They're unclassified, you move on. And in this case, we'd lose about 25% of the data that we were interested in if we did that. So we've added on to our approach functional annotations of these sequences with the hope that we'll get more information from those unclassified sequences that we'd otherwise throw out, because we'll be able to identify different coding regions and other gene ontology terms and functional annotations that are important for understanding what's going on in the microbial community. And this has been pretty successful so far. So we're actively compiling the data and statistically analyzing everything pretty soon and writing it up. But I've been encouraged at the results that we're seeing. I think we will see some changes in the microbial community, and pictured here is an output of our Seq Screen software, which was developed with funding from the IARPA Fund GCAP program, which aims to help annotate short sequences with functional potential, specifically for pathogenesis descriptions, but really we're annotating all known functions here. And this is showing a sequence that is involved in energy production. And one of the hypotheses that we have is that there are different changes in the microbial community and genes being expressed to produce energy in response to SARS-CoV-2 infection and COVID-19 disease progression, and potentially an increase in reactive oxygen species and changes in the microbial community because of that. So those are all things that we're exploring, and it's really interesting stuff. If you're interested in talking more, please feel free to reach out or check out our OSF page. We're happy to talk more.
Thank you, Krista. I think that was great. If people have questions for Krista, go in the Q&A, and Paco's already answered one of the questions that was for him. So if you don't have for questions, but yeah, so thanks, Krista. And we'll have more lightning talks later. So Leo, I guess I'll pass it back to you.
Sounds good. Thank you very much. All right, let's go ahead and move on to our next speaker. Our next speaker is going to be Dr. Christian Munk, who received his training at the MRC LMB in Cambridge, UK, and Harvard Medical School. He started his independent group 3.5 years ago at the Institute of Biochemistry at Goethe University, Frankfurt in Germany. His research focuses on systems biology approaches to study cellular stress responses, particularly after mitochondrial protein misfolding or infection of cells with pathogens. To do so, he applies quantitative proteomics, including methods to acutely measure protein translation. Christian has won a number of prizes, including the British Neuroscience Postgraduate Award and the Aventis Foundation Bridge Award. Today, Christian will talk about his recent efforts to understand host cell responses after SARS-CoV-2 infection. Welcome, Dr. Munk. And I can't hear you if you're speaking. Maybe that's on my end.
Hey, Christian, are you on mute? We can't hear you right now.
Doesn't look like it.
Maybe your microphone setting on your computer.
Yep.
We see you.
Oh. Can you hear me?
Yeah.
Yeah, we can hear you now.
Okay. This is weird. Something assumed it. Well, it changed. I was saying thank you for having me to talk here today in this really so far great event. And also thank you for this nice introduction of myself and also of my data in the previous presentation. So that's really a good head start. And yeah, I mean, the important point is I'm not a virologist. I care a lot about the systems biology approaches to understanding what happens in the cell upon different kinds of stresses. And as mentioned, we focus a lot about stresses from misfolding, particular mitochondria, but also in stresses that occur upon infections with pathogens. And I will just mention in one slide, because I think this was really technology that for us made a lot of difference for the data we'll show later, that one thing we care about a lot is translation, because this is really, if you think about proteins being the kind of reagents in the cell that have the function and that can change the behavior of cells, translation is the earliest way how we can see any changes in the protein composition happening. And we spent some efforts in the last years, and it was mainly from a PhD student in my lab, Kevin Clun, and it's published here, to set up a new way of doing proteomics to really be able to measure acute changes in translation. And I will not get into much detail. We use pulse cyclic labeling, so cells don't realize that anything is changing. It's just newly synthesized proteins will now have a heavy amino acid, and it works very well. Here's showing translation in HeLa cells after 15 minutes up to 120 minutes of incorporating this amino acids. And you can see you can determine even after 15 minutes already translation rates for a lot of proteins. We have a good dynamic range and it's quite robust, and you can use it, for example, here to look at mTOR inhibition to see the effects on cells and translation for over 5,000 proteins. And this is quite scalable, much more than ribosome profiling, for example. So we can also do very big experiments with many conditions and replicates. And for us, importantly, is we would need very few cells for that. That was also important in the infection experiments. And it's quite cheap compared to ribosome profiling for us. And the current record is about 9,000 proteins. So this is what we had, and we became more interested again in looking at pathogen infections. And then on February 1st, so we'll mention some dates because we had them in a lot of slides. On February 1st, there was an evacuation flight from Wuhan coming to Germany with about 150 people in there, and 2 of them were actually infected with SARS-CoV-2. And then something that started was really great collaboration between me and more biochemists, cell biologists, and the Virology Institute, where within just a couple of days, on February 5th, they told me they'd managed with the extracted virus to amplify it and also to set up an in vitro infection system. So here this is using CACO2 cells, which is a colon cancer cell line. And you can see, I think, quite well that upon 24 hours of infection with SARS-CoV-2, we have the very clear CPEs, or the response to the viral infection and the cells dying and releasing virus. That was also confirmed looking at quantitative PCR at the supernatant of the cells over time, and we saw that we have a release of the viral RNA copies in the supernatant. So we had a system that worked very well to work with. And then we went into the details to study that. And for us, the idea was really just in the cellular infection system to get an idea what's actually going on, what is the host response, to both gain ideas and what might be therapeutic targets, not so much for vaccination, even though there might be some information there too, but more for drug treatments. And the experimental system was like this, where we used these CACO2 cells, infected them with this patient isolates of SARS-CoV-2, and then at different time points, 2, 6, 10, and 24 hours post-infection, we looked at both translation and total proteome using this method that I just introduced in this one slide. And we prepared mass spec samples on that, shot that, and carried out some more extensive bioinformatics analysis of that. And I just quickly want to put this in there because to us it was really important to make this data available. I said we started on February 5th. So this project I'm showing to you now, this is a manuscript we had submitted on February 27th. It was very hard day and night work to get there, but we wanted to make it available. So there's a section on my website where you can have full access to the raw files, how it was labeled. There's even an interactive online tool, and we put all the supplementary tables to the paper that's now in Nature, so that really, hopefully everyone can go through there with whatever expertise you have with a virologist or another background and see whether there's something useful in there that can be extended on. So we looked at different things here. I will just pick some parts out because I think time will not allow much more. Here we looked at translation of viral proteins just to see that the system was working. You can see that over time here, there is translation of these viral proteins increases. So there is also generally an increase in translation rate over time in the system. And something that I just want to mention, because to me that was quite striking. There are a lot of reports in the literature for a lot of viruses where there has been proposed that they reduce translation extensively in cells. Like you often even read about things up to 90% reduction in translation. And unfortunately, most of this data is really based on overexpression of single effector proteins. We even have that now in SARS-CoV-2, where just NSP is getting expressed, and then people look at translation, but there's not so many global analysis for translation, in particular after infection with a real life virus. So this is one of these examples, and quite strikingly, we don't see much of a difference. So the maximum is reached here after 10 hours, where we adjusted 23% reduction in translation. This is still a severe effect if you think about ER stress and diabetes. This is usually the maximum you get. But as I mentioned, for viruses, much more has been suggested. So I think there's actually a lot more work that can be done in there. But we also looked generally now in the host cell proteome, what's changed. We focused on different factors. I picked out two, I thought, nice examples. We first looked at how the proteome changes over time. So here, this 24-hour time points, everything was done in triplicates. There was a cluster of things that were downregulated, but most strikingly, things that increased. And I have to say, we also cared more about pathways that increase upon infection, because then, of course, one could use drugs, inhibitors, to prevent these pathways with the hope that it might prevent viral replication. And this is what we found here. So one of the really enriched pathways in here was carbon metabolism, focusing a lot around glycolysis. And we wondered whether an inhibitor of glycolysis, that would be 2-deoxyglucose, would be able to prevent SARS-CoV-2 replication in our cell system. And indeed, it did so at kind of low millimolar concentrations. That's shown here. And one of the reasons why we tested this, and we're quite excited about that, because they had in other reports already for rhinovirus, where this 2-deoxyglucose had really been beneficial to prevent viral replication even in animals. And another part of that is that there's been a lot of efforts to make these molecules available in therapy. That was a focus on cancer therapy side, where free drugs were produced that try to become more stable and then available, that this could really theoretically become a treatment. That's done particularly biomolecular. And then there's this other aspect that we looked into, and that is just following the profile and how cells, the cellular proteome behaves over time in the infection, the profile you observe. And there we were focusing on what behaves similarly to the viral proteins, with the idea. So these are 9 viral proteins. Of course, we see them increase over time. We were just wondering whether there are cellular pathways that behave in similar ways to that. And there were a couple of them. One of them that was really most striking is shown up here in this network analysis, and it's probably not super surprising, and that's nucleic acid metabolism. Of course, the virus needs bases to replicate its RNA genome to be able to replicate. And there what we found is that this is one inhibitor, ribavirin, which inhibits the IMP dehydrogenase, that was able at low micromolar IC50s also prevent replication of the virus in cells. And even though I said it is kind of expected that some of those inhibitors should work, we still saw quite distinctive differences whether we were inhibiting purine biosynthesis or pyrimidine biosynthesis, and not both of them worked equally well. And I think this for us has also been quite exciting. We're basic researchers. Usually we are far away from real translation to the clinics. But I think also based on our data, there's now this clinical study going on in Canada where they use Virusol, which is an aerosolized form of ribavirin that's being tested in COVID-19 patients. So kind of a summary of where we were at that point is that we have this cellular infection system for SARS-CoV-2. We looked at whole cell proteomes and translation proteomics, and we found that the translation remained largely unchanged. And we also identified 5 pathways. I've only mentioned 2 so far. Those 5 were translation, nucleic acids, biosynthesis, splicing, proteostasis, and glycolysis, that all were really strongly increased upon infection. And what we found is that with specific inhibitors for all of these 5 pathways could really prevent SARS-CoV-2 replication in our cell model. Something on this slide that's still missing in our data that we can't address is kind of these gray arrows. So these are observations, the pathways change, but we don't really know why, what the signaling component is. And of course, proteomics also offers us a tool there to get to that, and that is carrying out phosphoproteomics, where we can see phosphorylation changes or changes in signaling cascades. And it was a very similar experimental setup as before. This time we did it in 5 independent biological replicates, mock infected or SARS-CoV-2 infected. And that ends up in a 10-plex. That's just a tool that we use where we can multiplex samples to pool them into one. We're still limited by the amounts of samples we can get, but we managed to enrich phosphopeptides from there. It was just 10 micrograms across the 10 samples. It's not much, but we managed with that in the end to identify about 16,000 phosphopeptides, quantify them. And I think it's quite obvious here that there were massive changes within the cell upon infection. And this was mainly phosphorylation on serines, which is driven by the method we were using, which was IMAX. So you essentially largely get serine phosphorylation in there. But that was quite interesting. So we looked at different aspects. We identified a lot of viral proteins that were phosphorylated that may explain some of their functions. And we did, again, kind of cellular pathways analysis, particularly focusing on kind of overlaying pathways of the effects that we see with pathways of FDA-approved drugs and their targets to identify pathways that we might be able to target with compounds that play an important role or could play an important role in infection. What was really a striking center, and it was really sticking out quite extensively, it was very easy to find, was signaling around growth factor receptor signaling, EGF receptor, but also some of the other ones. And this is shown here. There's all these downstream proteins here and in these blue boxes, and the circles indicate phosphorylation sites in there. So here was one phosphorylation site, there are two, there are four, and the color indicates what phosphorylation, which direction it changed. And it's from here, the red means it was upregulated, and you can see the whole pathway was really lightening up. What we found is that growth factors receptors were activated, they were phosphorylated, downstream signaling from RAS-RAF signaling and AKT-mTOR signaling was increased, as well as some other pathways that are related, like receptor endocytosis, cytoskeleton remodeling. Well, the real big question for us was then there, well, if upon infection with SARS-CoV-2, we get activation of this growth factor receptors, could we use compounds that block this signaling to assess their effect on viral replication? And I mentioned before, we did not only see EGF receptor, but also some of the other ones. So we thought it might make more sense to focus on the downstream signaling because that is highly overlapping across the different receptors. So we employed 5 different compounds that inhibit different stages here in the downstream signaling, and all 5 of them prevented SARS-CoV-2 replication in cells and about low micromolar concentrations. I also put the Cmax underneath, so the concentrations that can be achieved in patients. And you can see they're all quite in a similar range. And here's another confirmation where we looked at release of viral RNA copies into the supernatant with these different compounds, and they prevented this quite efficiently. So with that, just as a quick summary, what I showed you, we developed a new translation proteomics tool that we named MeProt. We set up a cellular infection system in CACO2 cells with SARS-CoV-2. And using that, we found different pathways that were increased upon infection, and inhibition of these pathways prevented viral replication in cells. And then we went looking at the underlying signaling events, carrying out phosphoproteomics, and there we found that growth factor receptor signaling plays an important role in inhibition of the signaling events prevent viral replication. So with that, I would like to thank my lab. The work was spearheaded by Kevin Clun with the help of Georg Tascher and in collaboration with the virologist, as I mentioned. Really important to point out the different funding sources that made this happen to really quickly move into an entirely new context with SARS-CoV-2. I would also like to thank our proteomics unit that I'm heading, because they supported this a lot. And thank you very much.
Thank you very much for that. All right, we've got some nice audience questions here. So first one, many of the current omics data for COVID research is based on RNA-seq data. Do you think the changes in the RNA-seq is translated to the proteomics? And maybe I'll just add in there, is there an advantage of proteomics over like RNA-seq?
Yeah, I mean, for us, I mean, in the lab, we do use RNA-seq quite a bit, but I think the issue in the end is if you really want to understand functional differences, this is being carried out by proteins, and I think you need to look at proteins to be able to do that. I think RNA-seq is important because it tells you a lot about activation of pathways, transcription factors, and so on, but for the real function, proteomics is important. And I think what we really needed here is something that's highly time resolved, right? That's the issue. If you have a quick infection happening, you also need to quickly be able to monitor the effects of that.
Okay, and another one. For the study, you used CACO2 cells derived from colon carcinoma. Why didn't you do experiments with other cell lines like lung or HELA?
Well, the easy answers, especially in the beginning, it was really hard to find any cell line that was permissive. I think now we're a couple of months further down the line, but in the beginning it was not obvious. Our collaborator, Indrish, he used to work on Sars when it came out 17 years ago, and they set up some of the first infection systems there in human cells, and they found already that CACO2 cells work very well for that. But of course, now we're trying to get in other systems, especially I think the goal is to get in primary systems or something that looks like a bit more like an organ. But it will be important to also compare different tissues, I think.
Great. Another question. Did you notice any upregulation of IL-6 signaling pathway?
I mean, I think generally in inflammation, we saw some responses, but it was fairly mild. And I think this is mainly driven by the cell line. Yeah? So I think for that, what we've been doing now is to get into more towards primary models, especially of the lung. And I think this is where we much more easily now see this inflammation responses. And it looks like IL-6 might quite play an important role there.
All right, and last one here. Were you able to see a reduction on the translation of the ACE2 receptor and other proteins like angiotensin?
So we didn't see, I don't think we saw a reduction in ACE2 translation, but we certainly saw a reduction in ACE2 levels. And we think that might be, and this has been described for other viruses in the past, there might be some shedding going on, right? That after infection, it just gets cleaved off and the ACE2 is being released. And then of course, we would not see that in our cell pellets anymore. So there's something happening there for you preventing a second infection.
All right, thank you very much, Dr. Mook.
Thank you.
Dr. Orozco, are you ready to get your screen share set up there?
Yeah, sure.
Thank you. All right, our next speaker is going to be Dr. Modesto Orozco, who is a full professor in the Department of Biochemistry at the University of Barcelona and group leader at the Institute for Research in Biomedicine. He's also the director of the Structural Bioinformatics Node and the director of the Joint IRB-BSc Research Program in Computational Biology. His main research interest is the understanding of biological systems from the first principles of physics and chemistry, focused in the understanding of the mechanism of flexibility and signal transduction in proteins and in the connection between physical properties of nucleic acids and their function, with a special emphasis in the study of chromatin. Welcome, Dr. Orozco.
Okay, so thank you. Can you hear me well?
Yes.
Okay. Okay, so it's going to be a little bit of a change with respect to the previous talk. I'm a theoretician by training. I'm a quantum chemist, and I will try to give you a different view of the things that we can do, you know, from physics and from basic theoretical chemistry. So, okay, this is like an old cartoon of the way in which the virus works. So it enters here, it should be the ACE2, it internalizes, and then the very first thing that is done is that the RNA is copied, is replicated into a new RNAs and for the new genomic copies, and also in RNAs that from which the protein, the viral proteins are synthesized. And the key player in the business is this protein. It's an RNase-dependent RNA polymerase. It makes a complex. We still don't know exactly how the complex, all the details of the complex, but this is the central element in the replication of the virus. It's a quite interesting protein. It has not a close similarity to any other proteins out of the coronavirus, the beta coronavirus family. There are many other RNA viruses, but actually this protein is quite different to anyone. And considering that the coronavirus are quite recent in terms of evolution, it's a sort of mystery how this protein has evolved and how it's so efficient. Okay, so we had, and thanks to the work of many different groups, we had already very quickly a 3D structure of the protein. We have already two high-resolution structure of the proteins. Actually, it is complex with other peptides, other proteins, NSP7 and NSP8, that are premises. So they are involved, and probably in the real protein complex, it is involved to an helicase and any other. So it is a very obvious target for treatment. because this is a protein that is not working in humans. So in humans, you don't have RNAs copying RNAs. So you have RNA polymerase that copies from DNA, and then you have processing of RNA, but you don't have this type of system. And so RNA polymerase used to be targets for, the treatment of any RNA virus. And this is not a surprise then that the only drug that has been approved by European and American agencies is remdesivir. That is this very exotic molecule here that was designed against Ebola and is an RNA polymerase inhibitor. Okay, I'm not gonna told you about the evidence that remdesivir is useful, is really useful. There is no question now, but I'm gonna take a look at remdesivir from the point of view of a chemist. So when you look, this molecule sounds like a very exotic structure, but okay, this is just an indication that this is a prodrug. So this molecule will work as an ATP analog. So all this will be removed, and then the molecule will be phosphorylated. And then you expect that this ATP-like molecule will inhibit the enzyme, either by inhibiting the incorporation or by incorporating into the nascent RNA and inhibiting the enzyme by reasons that you don't really know. Okay, what else can you find here? So this is position 1, this is position 2, and this is position 3. Okay, this says the enzyme that it looks like an RNA. This part here says enzyme that it will work like an adenosine analog, and this part here says that this molecule won't be recognized by proteins. So this is not something that can be stable in the genome because you are losing here recognition pattern of an adenosine. This guy here is not a surprise either. This is a CC bond, and it will give some stability to the complex. This guy here is a kind of a surprise, because if you look at other RNase inhibitors like acyclovir or whatever, this is they typically lack the 3' OH. So this is a way to stop polymerization. So polymerization goes from 5' to 3'. So if you don't have 3', this is a signal that you will stop polymerization, but this guy has a 3' OH, and this is a surprise. And this is a big surprise. Come on, I mean, if you work in medicinal chemistry, this group means problems. You never want to have a drug with a group here. So it was what first shocked me. And of course, this is a kind of a side product of the chemistry. So this is the way in which, they obtain this molecule. And in the chemical synthesis, you get this cyano, this nitro group here. But of course, they do a lot of chemistry based on that, and to remove this group, and none of the molecules that they developed, that they obtained, was actually so good. So it is clear that this nitro group here is doing something. We didn't know why, but they will come later. So let's go back to the enzyme. This is NSP12. And this is the first structure we start from. So this molecule, this structure was published, I think it was in Science. It was like in the upper form, but we managed to take an alignment from the hepatitis C virus to actually put the RNA there. And it was very nice because we did a very nice model. And just a few weeks later, it was this other electron microscopy structure where it is very clear that our modeling of the RNA inside the enzyme was correct. When you take a look for the chemists in the audience, you realize something that for me was just amazing, is how beautiful is the binding site. This is the active site. And remember, this enzyme has refined by evolution. Coronavirus are very recent virus. It's in the order of 1,000 years old, compared with humans that we have millions of years of evolution. And this enzyme that has not homologs with other virus actually has a beautiful coordination site. So this is the fingerprint of an active enzyme. So you hear the magnesiums that are crucial for reaction. You have here the 3', so this is a 3' nucleotide that is stabilized. You hear this You see this kind of crown of aspartic acids that are coordinating the magnesiums. Everything looks perfect. And if you look in more detail, you will see this wonderful setup. You have these arginines that are actually capturing the phosphate of the incoming nucleotide. And this is the control site. This is the point where the enzyme knows that he's incorporating a ribonucleotide and not a deoxyribonucleotide. So these are the controls. There are many of these control sites that are made by serines that actually checks that you have a hydroxyl here, a 2' hydroxyl, and this is a fingerprint of a ribo. Remember, you don't want to make, if you're a virus, you don't want to make a copy in DNA. You don't want to do a retro transcriptome. You want to do an RNA copy. And you have this type of control again in here. So this is the 3' region, and you see that the enzyme is checking again that this is right. Okay, you want to have, if you're a virus, this virus has not a very high level of mutation, so it's quite precise in terms of the copying. And you see here how beautiful is the recognition side. So you see that there is this wonderful pocket. You have this a wonderful pairing here that recognize, in this case, it's an adenosine that it will recognize an UTP, so a uridine that will be placed in front of it in the incoming nucleotide. You see how beautiful is the shape complementarity, and you see how beautiful is the recognition for a ribo, and the hydrogen bond pairing. So what happens with this wonderful, with this wonderful type of recognition? What happens with remdesivir? Remember, I'm sorry. Remember, remdesivir is not a natural product. So why it's what it's doing? And we did some modeling, and actually what you see is that remdesivir work like if it was an analog of adenosine. You see all the recognition is beautiful. Even the nitro group is tolerated. I won't say that it's happy, but it's tolerated in the active side. Okay, so at this point, we try to learn how the enzyme works. And my original idea is that it will be a very inefficient enzyme, just because the years of evolutions that is behind this enzyme. So we have eukaryotic enzymes, all of them have millions and millions of evolution time. This has thousands of years of evolution. So why is efficient, or whether or not it is efficient? And so we did this. I'm not going to tell you about the details. So what you have here is a reaction coordinate. You see, this is the incoming nucleotide. You can see here the incoming nucleotide. And this is the receptor site, and this is the transfer. You see here that this phosphate is moving and is transferring from this. So at this point, the viral genome is one step more. So it's one base more. In this case, the incoming nucleotide is an uridine. At the bottom of the slide should be a pairing adenosine, and this is a reaction-free adenine. And this is a first surprise. So I will expect from, this is a normal nucleoside, this is uridine. There is a barrier of 15 kcal-mole, more or less. If you compare the same reaction in human, would be in the order of 13 kcal-mole. So this is a very, very efficient enzyme. Despite the low evolution that it has suffered, it is very enzyme. It's very effective. It is very active. This is why the virus is so good. But this is the second surprise. So our original thought was that remdesivir was not incorporated, that it was a kind of non-bonded inhibitor that will be placed in the binding site and actually stop the proliferation, but actually is not. So this barrier between 15 and 16 is not far away from 17. So our results really suggest, very strongly suggest, that is not only a very efficient enzyme, but remdesivir is actually incorporated into the nascent RNA. It's doing something, and these are results from modeling. Here, instead of an adenosine, you have remdesivir here, and you can see that it's doing nothing. So the RNA duplex, it will be the nascent RNA, is fully stable. So it's not, at least based on MD simulation, nothing is happening. It makes the standard Watson-Crick pairing that you have here, and you know, it's okay, it's tolerated. So why then remdesivir is inhibited? So we know from this structure here that indeed experimentally remdesivir is incorporated into the RNA, but if it's incorporated into the RNA, why is inhibiting the enzyme? So okay, the step you expect to make the inhibition is when it is incorporated, but it's incorporated and nothing really happens. And we found this at that point was unpublished, is now already published, that very strongly suggests that actually remdesivir does not inhibit the enzyme at the point of insertion. It's not inhibiting the enzyme at the place where it is incorporated. It inhibits the enzyme a few steps later. And this is rather unique. It's very, very rare. So we start then thinking what can happen, why the enzyme is stopped a few steps after remdesivir is incorporated. And then we make, what we did was to screw the DNA through the exit channel and see where this suspicious group was placed in it. And again, you can see that after a few steps have going on, you find this group here, this nitro group, close to this serine here. And actually, there is a water molecule here. And if you're a chemist, you immediately think in a reaction that is called Pine reaction. A Pine reaction, I try to show you here the reaction. A Pine reaction is a reaction where this cyano group here and this serine are the main players. You see that this serine gives a proton to the water. This water gives the proton to the cyano group, and at the end of the day, you have this complex here. So you have a covalent complex, and this reaction has a free energy that is quite low. It's about 15 kcal-mole, while the reaction of incorporation was about 16 or 17 kcal-mole. This is the reason why we believe it's actually inhibiting the enzyme. So remdesivir is incorporated, and in a slide, through the channel, and when it reaches the vicinities of serine 861, that is a serine that probably was placed there by evolution to check that it was an RNA. Actually, it makes this reaction. You can see that it's not a perfect inhibitor, and there is quite a lot of room to improve it because there is only 1-2 kcal-mole between the progression of the reaction. So the enzyme put one more nucleotide and then slide the RNA out of this serine. So it's not a perfect inhibitor, but it's a good inhibitor in the sense that this reaction happens very efficiently, and as you see, with a very favorable reaction-free energy. See again, the water, proton transfer, and then you have the covalent complex. This is a kind of cartoon representation of what we think is happening, and nobody has shown that it actually can be a covalent inhibitor. This is a kind of a schematic representation of what it's doing and explain why it is a delay in inhibition. This is the binding site, this is the active site, this is where polymerization happens, and this is the place where, according to our calculations, remdesivir catches the enzyme and keep it covalently bound. Do we have a clear evidence that this is true? We still don't have experimental evidence. Actually, we suggest that this is a reversible covalent interaction. In acidic media, it can break and recover the non-covalent complex. But if you look at the literature, there are many, many evidence of many enzymes that are actually showing this type of covalent inhibition with a very similar thing. This is why no pharmaceutical company wants to see a nitrile group, because it's a very reactive group that if it has a hydroxyl or a sulfur group here, group around, it will make a covalent complex. Okay, why is not toxic? So it's beautiful to see that you inhibit the viral enzyme, but the problem is that, of course, you should not kill the you know,
Dr. Orozco, I just want to let you know you have about 2 minutes left.
Okay, I'm finishing. It's very important that it's not killing the cell. And then we look at the available structures of human polymerases. There is nothing close to the natural group that can make a covalent complex. But very surprisingly, all the coronavirus that we know the remdesivir is efficient has the same serine. And actually, we cannot show because the alignment are poor. It happens also with Ebola. In the place that we suggest or we expect that remdesivir will pass near, there is a serine, and we guess that in case of Ebola, is the same type of mechanism. Okay, is the virus becoming resistant to remdesivir? Are the mutations of the virus consistent with the reaction mechanism? So with the help of Nuria Lopez, we plot all the mutations. I can say you there is no mutation that compromise the mechanism of reaction. It's something that you expect, you know, it won't be selected. There is nothing around the active site, around the progression site, and there is no mutations in the remdesivir. So based on our data, at least in terms of the covalent binding, right now we are quite safe. This is my last slide, just to tell you that what most of the things that I explained is the work of a postdoc in the group, Juan Aranda, who did all this QMMM. So thank him for his work, and thank you for your attention.
All right, thank you very much. We'll do one question here. I've got an internal question actually coming in from Dr. Hugh Ogilvie. The question is, would widespread use of remdesivir lead to the replacement of the RDRP serine, or would this be too costly to the virus due to, for example, increased replication errors?
I think it's mostly this. So the enzyme has a good recognition pattern, and there is another enzyme that actually is an exonuclease that is a proofreading protein that, in principle, warranty that the level of mutation is low. Actually, the level of mutation by the virus is quite low. So it's not like H or other RNA virus. This has a very long genome, and then a long RNA genome means that the virus has to take care to have a good replication, fidelity, and actually there is this double checking. So I think it's not really that is increasing the level of mutation. I think that is a direct inhibition of the enzyme.
Yep, thank you. With that, I think it's time to go ahead and turn it back over to a second round of Covert Lightning Talks. Afshin, feel free to take it away.
Yeah,
thank you.
Thanks, Leo. Yeah. So we're gonna again have 3 talks, lightning talks. It'll be the next 20 minutes. And again, each of these will highlight different subgroups from Covert. And we're gonna start off with the virus group from Todd, the other co-lead of or co-president of Covert. And I'll let you take it away. Go ahead, Todd.
Yep, thanks, Afshin. So I'm just going to cover today very briefly the work that's going on in the SARS-CoV virus subgroup in Covert. And just to remind everyone, I'm Tom Trehagen, assistant professor at Rice University and co-lead of Covert. Okay. So I also want to just recognize all of the members. We're fortunate to have a very broad and diverse group in the virus subgroup. Very excited to work with everyone listed here. So it's been a great experience. And I also wanted to take a moment to highlight a Rice COVID Genome Sleuth REU that Leo and I have been leading. And we have this Project 2, which is on transmission analysis. We have 2 Rice undergrads who've been helping out with that. So shout out to them for all of their help on the transmission analysis. Okay, so I'll just kind of go back to the start here with this limited time I have today. And so I'm leading the virus subgroup in Covert. And of course, I always like to go back to the beginning of anything just to make sure that I'm capturing all of the facets we need to capture. And we don't, we can go back to 2003, this paper published in Nature Reviews Microbiology, and it was SARS beginning to understand a new virus, right? And you read through that paper and it really kind of gives you some good perspective on the things you need to consider when sitting down and try to understand SARS-CoV-2. And not only that, SARS, of course, is another coronavirus. So it's a really, really nice paper to pick up and read before really getting started into SARS-CoV-2, viral analysis. Okay, and this is a great graphic for anyone that I have the link there, and I'll share my slides afterwards. This is from the LA Times. I really enjoyed this graphic, but essentially just kind of the main point of these few minutes I'm going to share with you is that coronaviruses use spike proteins to infiltrate cells. And so just two main points. Number one, these viruses depend on host cells. They need to get into a host cell. And then once they're inside, they make copies of themselves. So on this left-hand side of the slide, you have this very small, hard to see coronavirus particle, and then this very large host cell. It gets into that via spike protein and other processes. And then once it's inside, it will make a lot of copies of itself. And it's really important because inside of every person that's COVID positive, you're gonna have many copies of the virus, okay? So just emphasizing that point is once a virus infected cell, once a virus is inside an infected cell, it can produce hundreds to thousands of viruses, okay? And that's for each infected cell, right? And then it'll just this process will continue. So we really need to think about this. We want to look at kind of SARS-CoV-2 about, there's an actual population of viruses once you get infected, not just a single one, okay? And that's kind of a really key point to what I'll say and what we're looking into in the subgroup, right? And to put a finer point on this is thanks to one of my PhD students, Maria Wang. It's really thinking about consensus level information. We have this amazing resource, Jusei, that is collecting a lot of genomic information. I think it's up to about 40,000 genomes sequenced either on, I think, primarily Illumina or nanopore platforms, okay? But that's all at the consensus level. So if we're thinking back to this population again, essentially what that consensus means is you're kind of taking an average across that entire population. You're representing that entire population of hundreds and thousands of viruses that you're infected with a single 30,000 character string, okay? So the purpose of the subgroup is to really look behind the scenes and look at the hidden diversity that we have in the reads underneath that, which are not always shared. But if you look behind the scenes, you'll see that there's a lot of diversity there actually. And this could be a useful signal. So that's really what this presentation is about, what the subgroup is about, is really looking behind the scenes, inside it, within person variation, how diverse is this population of viral cells specific to SARS-CoV-2, and how can we use them, and what are implications, okay? So, and then this graphic here is kind of highlighting what I just said. You have two individuals, a donor and a recipient. Someone may cough or sneeze, and you have on the left-hand side maybe a population of viruses, and then on the right-hand side, someone that will receive some subset of that population. And then we want to look at, compare, and use information to see if we can help with transmission analyses, et cetera, okay? And really relevant to this talk is we have very fortunate to have a speaker later in the day, Dr. Don Milton. He had a letter that was titled, "It is Time to Address Airborne Transmission of COVID." And this really related to what I'm saying. So just keep that in mind when he's talking, that it relates back to some of the things I pointed out. I'll just end with saying there's a concept I can't introduce today, but there's this idea of a bottleneck. How many individual cells viral cells or virions are transferred when someone sneezes or coughs and will arrive to that second individual. But using some of the sequencing, deep sequencing technology we're using, you can actually look and interrogate and kind of compare and contrast and see what type of patterns you can see. Okay, and that's what this slide is showing. Essentially, you have a donor population, you have a recipient, which will be a founder population of that donor, and we're looking and comparing and contrasting this information to see how much you could really tease out and how much this can help with tracing. Okay, so as inside of the paper that I'll share briefly, you can take a look for a given person. These are the blue is a sample from, and red are samples from the same individual spread out by some amount of time. Okay, and you can actually look. The differences within a person over time, the same person are pretty consistent, and that gives us some hope. This can be a reliable signal that you can use to later look at actually transmission. Okay, sorry about that. So with that, I'll just jump ahead to the preprint. This is a preprint that the viral subgroup has produced, and it's on bioRxiv. The DOI is at the top of the slide. I would like to thank all of Covert again, just amazing team. And with that, I'll stop.
All right, good snippet of what to expect from the viral group. We have one question, so it was on there. Actually, it disappeared. Okay, how are you distinguishing variation in genome from genomic fragments?
Yeah, so that's it's ongoing work. Part of it is looking actually at the structural variants, which I didn't get to say. This is in collaboration with my collaborator, Dr. Fritz Sazlacek from Baylor College of Medicine, where we're looking at structural variation and single nucleotide variation. But of course, with short read sequencing data, and this all came from RNA-seq data from the Mason lab, we might need longer read sequencing and different strategies to pull out some of the subgenomic information. But we're looking into phasing. We're looking into different strategies we could possibly tie together to help get at some additional information there. But very good question.
All right. Well, any other questions, put in the Q&A and Todd will directly answer that. So we'll go on to the next quick lightning talk. So this next subgroup is a newer subgroup. We're calling it, well, right now it's ethnicity, but it could be more ancestral genetics. We're discussion to change the name, but it's Eve Worley from Iowa State University. So Eve, if you want to share your screen, you got about 5 minutes, a little more than 5 minutes. Oh, is Eve there? I don't see Eve. Let's see.
Eve, I see Eve. She's there. I don't know if I see her listed.
Maybe she's an attendee. We have to make her. Is she attendee or is she?
I think it's just an attendee. Actually, it looks like. So I'm gonna promote her.
Okay.
I'll promote the panelist.
Yeah.
She's now a panelist.
Okay. So maybe you can talk now. Or maybe we'll come back to you.
Okay.
Oh, there you go.
You got me. Okay. Hi, how are you?
Good.
So let me share my screen.
Yeah. We'll give you a couple of more minutes. Since,
I'm good. Okay. Oops. Okay. Oof. Okay, so our project's only a week old, really. We had our first meeting a week ago, so I'm going to mostly talk about the work of my own group, and because I'm not really familiar with everybody else's work right now, and a little bit of Bruce Arnaud's work. So we have in our project a group of amazing people, including domain experts in different diseases and cellular components, computational genomicists, and data science scientists. And our goal is to look at the role of ancestry in COVID-19 infection. So here we go. Okay, so we want to develop precision therapeutics for COVID-19, and we have two hypotheses we're working on. The first is that gene expression will vary by the population origin, and this will help us to understand different aspects of COVID-19 disease biology. The second is that orphan genes will enhance the prognostic signatures of COVID-19 disease and will also reveal aspects of COVID-19 and help us to understand it better. So we have a pipeline for looking at large amounts of data, and we're doing this whole project in the context of all the data actually we can get our hands on in COVID, involving COVID-19 and other data as well. And we're particularly looking at RNA-seq data, but we hope to also incorporate proteomics data and other data as well. So we're getting data from Covert itself and from SRA at NCBI, and we hope to get about 40,000 samples from 40,000 humans total. And we have a pipeline for raw read processing, which is both reproducible, and it also not only identifies annotated genes, but it also identifies novel genes that may also be important in COVID-19. So we've quantified expression of about 50,000 annotated and novel unannotated candidate genes in about 20,000 samples to date. We also take the metadata from those samples, and then we can explore that data. And our goal is to look to identify new candidates for experimental testing and precision medicine. King Jordan and others are developing a sample pipeline to infer the ancestry of individuals. And once we have that, that will go feed into this pipeline and be a really exciting part to help us understand the role of ancestry in COVID. So far we have 28,000 samples processed, including these large NIH fantastic studies on normal and cancer tissues. And we are accumulating all the data we can from the bulk RNA and single cell RNA studies on COVID-19, and we just keep on accumulating the data. A basic question that we have is why do African American populations, why are they so disproportionately affected by COVID-19. And obviously there's a lot of reasons for that. And they can depend on socioeconomic conditions, environmental conditions, and genetic. And gene expression itself is the interaction of these two factors. And by analyzing the data that we've done to date, we find thousands of genes that are differentially expressed in African American versus European American populations across all the different tissues and cancers that we investigated. So when we look at the overrepresentation of GO terms, what are the main biological processes involved in these differentially regulated genes, we find that a lot of them have to do with infection, inflammation, and immunity. So if you look at thyroid carcinoma, you can see these GO terms, they're all involved in these processes. And these are the highest, in the most significantly enriched GO terms. And it's true of many other of the conditions as well. This is one example of one gene, which is the F8A2 gene. And F8A1, which is a different gene encoding an identical protein in humans, is essential for early endosome trafficking, and it mitigates Huntington's disease, and it's required for Huntington's disease. It binds Rab5 protein with the Huntingtin protein and causes the endosomes to basically to slow down in their trafficking. So it's really important in this disease, and we don't know if it's important in COVID, but we do know that, for example, in the liver, there's a 41-fold change in expression. It's 41-fold greater in African Americans than European Americans. And actually this gene is often not even tracked in RNA-seq studies because it was not present. The initial studies were done with Caucasians, and this gene was very low, with Caucasians here, and the gene was often very lowly expressed, so they don't even notice it typically. And yet in African Americans, it's 41-fold higher. This is really cool cell map view developed by Bruce Mrna, and this basically is an atlas of RNA-seq data, single-cell RNA-seq data from esophagus, and it shows the various cell types in 6 normal individuals, and it clusters gene expression of 5,400 genes according to these presence in these different cells. And so you can see, for example, in this particular epithelial cell type, here's a cluster of co-expressed genes, and you can recognize what cell you're looking at by what set of genes are highly expressed. And he overlaid some of the genes that we got.
Sorry to interrupt, probably you got 30 seconds more.
That's all I got. He overlaid the single cell atlas of esophagus with the differentially expressed genes that we found in esophagus. And what he finds is that there's an extensive enrichment for the submucosal gland cells, including the duct and the mucous gland cells. And this is really interesting because these are the cells that are initially receive the virus, and it may be very significant that these are upregulated in African Americans. I want to end with a plea that we need to dissect genomic from socioeconomic effects. And for this, we need better metadata, both in reference to the genetics and race and various proxies for socioeconomic conditions. And without this kind of data, it's really hard to figure out what are the factors that are really important in terms of this disease. Thank you.
Thanks, Eve. If you have any questions for her, put it in the Q&A and she could directly answer you there. So yeah, thanks, Eve. That was good. So it's a little snippet of what we're doing with the ancestry ethnicity group. And we're going to end this lightning talk round with Bruce Mrna. And he's going to talk about the long haulers or the long-term impact of COVID-19. I guess, Eve, if you could stop sharing your screen.
Okay, got it, sorry.
No problem. So Bruce, it's all yours. Bruce is there earlier. Oh, I think you're on mute, Bruce.
Hear me now?
I could hear you now. Yeah. So okay, it's all.
Gosh, the gods of computer technology. Okay, I'm going to try and go through this in a lightning fashion. and see if it works. This is a new area for us to get into. It's a very important area. And why?
Hey, I think you're sharing your whiteboard. You gotta unshare this and then share your screen. Oh, there you go. Now we see your screen. Perfect.
And can I just hit the presentation mode? Is it coming through okay now?
Yeah, it's good now. Perfect.
Oh, fantastic. Okay, so we want to talk about the emergence of long-haul COVID-19 disease. Incredibly important thing. I think we're just starting to see the tip of the iceberg of it. I get to work with a lot of amazing people. Deanne Taylor is amazing. Steve Balin at Johns Hopkins, Robert Miller, and our leader Ashton is amazing. And I have a great team behind me, and I get to work with a lot of other people, and I'm really grateful for support too from places like that. Okay, so most of our research in COVID, I mean this in the biomedical community overall, is really about trying to understand differential severity on the initial acute course of the disease. And so we've got tons of data that's coming in from many different places, lots of beautiful data. This is the kind of stuff I do here, comparing gene expression profiles in single cells from bronchoalveolar lavage and PBMC and lung parenchyma from people who have differential presentations of COVID and differential survivals in response to drugs. Here's a friend of mine's dad who just who went through 19 days of extracorporeal membrane oxygenation and survived and came out. And let's see if I can close this off. But what happens next? And discharge after 48 days. And what is the status of all of his systems in this recovery phase? Well, there's a lot of signal that we're getting that it's not so simple. And why can't I? Even though we're maybe hovering as acute care practice is improving, and it really is. We're hovering, let's say, somewhere between 0.5 and 2% of strong cases that result in deaths. The rest of people who survive it are not out of the woods. And so with increasing frequency, we're starting to hear stories about ongoing symptoms, weeks, months after having cleared the disease relatively well, for just a week, 2 week, 3 week illness, and then things just don't get better, and new symptoms start to appear. And so there's some people who've written about it very, very nicely, that recoveries are taking far longer than what the WHO is still pretty much sticking to. And many times the symptoms are way more significant than the initial presentation symptoms, neurological, musculoskeletal, skin, kidney disease, liver disease, heart disease, a lot of cryptic heart disease. And so the idea that if you're in the 98% that don't get that severe version of the disease, that you're out of the woods is really wrong. And so just July 9th is the first, and most of these reports are anecdotal, but the magnitude of these anecdotal reports is gigantic. Here's a relatively systematic study from Italy where they took 143 patients, and they found that very high percentage of them, 88% of them, have ongoing symptoms that are really significant. And if you just compare the symptoms that they had on acute phase presentation, and then the percent of those that are staying with them, it looks like, yeah, you've got your things are going not so well in the recovery. But the truth is that there's a lot more symptoms coming out that are beyond those of the presentational phase, depending on the severity, depending on the individual and other things. So we need, this is going back in SARS from 2003, and finding lots of outcomes that have gone very bad: pulmonary fibrosis and femoral head necrosis, all of these symptoms are showing up in spades in SARS-CoV-2. So this, there are lots of groups. We want to collate, get these groups together. We want to leverage it, take multiple approaches. So that's the goal of this new interest group that we've got in Covert, trying to form an agenda around trying to study this. And we're going to need to study it in different clusters of individuals that where they're showing different symptomatologies that are, cardiac, skin, immune, neurological. All these sort of systems need to be dissected and so this is kind of the agenda that we're thinking about in going those directions, forming large cohorts, putting together multiple cohorts, extending current cohorts. We've got Melissa Handel coming up to talk to us about a very large all of us cohort that, where this extending into that is looks like it's going to be very possible. So trying to get biomarkers at every level. There's circadian aspects of the disease, really trying to do mechanistic dissection of it, understand these kinds of one-off uses of that individuals are trying. They'll try different medications, and there's some angles that where antioxidants and different nutritional strategies and maybe trophic factors, all of that stuff. We'll get some data around that, people's underlying genetics, really mechanistically dissect that, and then really consider what's our next strategy for therapeutic modalities that can both block the occurrence of these things going forward, as well as act as an intervention once somebody's in this state. It's very reminiscent of like Lyme disease and chronic fatigue syndrome. But now we're going to see this at a scale that we've never really had the chance to do, and then we'll be able to track back to different presentational versions of it, see how the immune system morphed in the course of the disease, and then what has that left people with. So that's the basic idea. I didn't get a chance to talk about some of these other red flags that are coming up, like the loss of going seronegative from a mild version of the disease. It's really an unprecedented phenomenon, implying that there's viral toxicity that's ongoing late in this. And so if that's viral toxicity is through an epigenetic or some sort of an imprint response syndrome, the pattern that's set up in the immune system that acts against itself, or if there's viral proteins that are coming out and really acting as toxic agents against different arms of the immune system, still very likely. That's why we need this host viral interactome to be really dissected in a different context. Thank you. It's really fun to work with this group.
Yeah, so thanks, Bruce. Let's see if you have one question. Actually, we do. We have one question. Enough time for that too. So, does the long haul syndrome include circadian-related changes? Any observations? And that's a good question because I know you know the answer to that.
So many levels. The thing that one of my sister-in-laws, the lead in flight attendants, and so the groups that they talk about and they talk to, there's just this the way that sets of symptoms just come up like clockwork. Like this thing is starting to happen to me at 2 o'clock in the morning routinely every night, sweats. And they'll say, yes, that happened to me too. This mental absence, fogginess, and then just being great in the morning, intolerance of exercise. There's so many different intricate features of symptoms that are shared over so many different individuals. So, and it's going to cluster out into different forms, but and then we'll be able to really go high resolution into those individuals, but. That's one answer.
I think that's good. Yeah, maybe just a break time. We'll do one more question and then take a break just as we're running. But another question is: do long-term SXS correlate with antibody immunology or do long-term serology?
Great question. Yes.
Yeah, antibody immunological capacity to fight infection if it hits again. I think some of the earlier speakers might have hit on that, but you could discuss that further.
Well, I mean, the short answer is obviously it's not known, but there's some strong signal implicating that a lot of these individuals are seronegative, and they've become seronegative. And so I'm sure everybody's aware of the recent studies from from China and Sweden, where there's not just a diminution of positivity of titers, but a true conversion to seronegativity. And so, the initial, as of a month ago, any doc seeing seronegative in somebody who's 2 months post-COVID-like phenotype, you didn't have it. I understand that you think it's a lot like what everybody's having, but you couldn't have had it. Well, guess what? That's wrong. And so that, and then what, on the other hand, the Swedish guys showed that there are these antigen-specific T cells that are there poised to react. And then there's a big question about are those cells adequate to really have a neutralizing immunity for someone? And there's, I think, the people who are well informed in this area are not being eagerly saying yes. That the strong possibility is that it's no. Therefore, these individuals are going to need to get vaccinated too. We don't know if it's a different safety protocol that's going to need to be followed in order to validate that it's okay to vaccinate them with vaccine 1, 2, or 3, approach. So that all of that needs to be worked out, and some of it is going to have to be probably with ex vivo samples.
Yeah, I think that's great, Bruce. I think we'll wrap it up. Any more questions for Bruce? Put in the Q&A, and then Bruce could actually just address it directly to you. Type it in. But thanks, Bruce. Yeah, I think this is an important question that we're trying to tackle now in the Covert too. But the problem is not enough data yet.
But we're with this consortium like ours, we're going to be teaming up with Melissa Handel. I think we're going to be in a really good position to make a full assault on this.
Yeah, perfect. All right, so we'll take a break until 2:20 Central, 3:20 Eastern, and we'll come back, and Leah will take it back from there. I'll put a slide up for a temporary break, so everyone knows. So then, yeah, thanks, and we'll see you guys in a few minutes.
Dr. Barnes, I think I see you and signed in. Are you here? We can just go ahead and start getting your screen set up if you want to.
Yeah, I'm here. Go ahead and share.
Sure. We'll kick it off in about a minute or two.
All right, that's good.
All right, I think I can go ahead and start with the introduction, at least here. So our next speaker is going to be Dr. Christopher Barnes, who is an HHMI Hannah Gray postdoctoral fellow in the lab of Dr. Pamela Bjorkman at Caltech, where he utilizes structural biology techniques to define the structural correlates of broad and potent antibody-mediated neutralization of HIV-1. Recently, he shifted his efforts to understanding the immune response to SARS-CoV-2 infection, having solved numerous structures of antibodies bound to the SARS-CoV-2 spike glycoprotein. By combining structural information with biochemical techniques, he hopes to rationally engineer improved antibody therapeutics, as well as protein reagents that can be utilized in vaccination efforts. With that, I think you can go ahead and get started if you'd like. Welcome, Dr. Barnes.
All right, well, thank you so much for the invitation. It's an honor to be here and present my work with you all today. So yeah, today I'm going to talk about our studies with convalescent individuals and antibodies that we have actually pulled out of convalescent individuals that neutralize SARS-CoV-2. So as we all know, SARS-CoV-2, this is the virus. On the surface of the virus are these spike proteins shown here in the multicolors, which represent the envelope or the protein utilized by the virus to enter into host cells. And on top of that, since this is the largest protein on the surface of the virus, it's one of the major targets, if not the sole target, of neutralizing antibodies, from an adaptive immune response. And so one thing to know about the spike glycoprotein is that it has a switch into a conformation that flips the RBD into this up conformation, which allows it to interact with the ACE2 receptor on the host cell. This leads to a conformational rearrangement, which then allows fusion of the viral membrane with the host cell membrane, either outside the cell or within endosomal compartments. So one way antibodies might interact with the virus and spike glycoprotein is by interacting with this receptor binding domain shown here now in red. So this binding domain can exist from structural studies in two different conformations, in which you have all down RBDs shown here on the left-hand side, versus up RBD, or in this case, 2 down and 1 up RBD shown here on the right-hand side. And so this up conformation is necessary for it to actually attach and to hook into the target host cell receptor with ACE2. And so one way antibodies work is by blocking this interaction by either binding to either the up conformation or the down conformation of the receptor binding domain. So just so everyone's on the same page with me, antibodies, of course, are serum proteins that combat pathogens. They are very specific, and in this case, we're talking about IgG antibodies. They are what you consider antibodies circulating in sera, and it's going to give you the neutralization that you would want. So one thing to point out is that, in my talk today, I'm going to be talking about Fab fragments, and these are the regions shown here in the multicolor. So the light chain shown here in dark blue, and the heavy chain, which represents the variable region that interacts with the surface of the antigen shown here. So the Fab fragment makes up this right here, and you have two arms that allow for bivalent binding in the terms of IgG, and the Fab itself would bind monovalently by itself. So when we talk about neutralizing antibodies, these are antibodies that interact with the virus in a way that prevents the virus from infecting this target cell. And if you look at, structures that have been solved, there have been a number of antibodies solved, isolated from both SARS infected individuals, including CR3022 and S309, which cross-react with the RBD of SARS-CoV-2, as well as a number of numerous antibodies isolated from SARS-2 infected individuals that recognize different regions of the RBD and recognize different epitopes. So there are a number of antibodies that have been pulled out and structurally solved. And here's just a quick showing that they can all bind at very different angles of the RBD. And so what we want to do is kind of understand the correlates of this antibody-mediated protection in the context of not only the polyclonal response seen from recovered individuals, like looking at the convalescent plasma and sera, but then also looking at monoclonal antibodies and understand what is actually leading to protection. And so to answer the first question, we utilize two different assays: a binding assay, ELISA binding assay, which allows us to understand potentially cross-reactivity between plasmas with different, not only SARS-CoV-2, but other coronavirus strains. And then we use a technique called negative stain polyclonal epitope mapping, which I'll talk about in a little bit in more detail later. Then on top of that, to answer the second question, we use single particle cryo-EM methods to solve Fab-S complex structures. So going for the first thing, we want to understand what was happening in patients that had recovered, right? These convalescent individuals we've all heard about. Here are just numerous headlines over the last several months from the New York Times talking about convalescent blood, convalescent plasma, and how it can be used in therapies. And so what is actually in this blood and plasma that's going to be therapeutic, and can we understand it better? And are there varying degrees between individuals? And so we work in collaboration with Michelle Nussenswag, who is at Rockefeller University in New York. And Michelle did one of the largest studies here in the United States of convalescent individuals. And so in a paper reported in Nature last month, led by Davide Robbiani, they studied and analyzed convalescent plasma from over 150 individuals, including several hospitalized individuals. And what they showed was that there was a wide range of neutralizing activity. So what you see here on the left, this plot here, is illustrating the dilution at which the ND50 or the NT50 dilution of convalescent plasma, which neutralized the virus. And so you can see, like for this individual 47 and 21, they had very high titers, had very high neutralization potential from their convalescent plasma. But then you had a lot of individuals whose neutralization capabilities were less than 1 to 1,000 in a dilution. And for the most part, a third of the individuals they looked at had either weak or no activity, and that their dilutions were less than 1 to 50, which is really low when you think about neutralizing plasmas, which is kind of illustrated down here with this, these sets of individuals here. So we knew there was a range of potential reactivity between different convalescent patients. So we wanted to study it. So some of the star patients shown here, 21, 72, 57, 96, and 107, we decided to study further in our group. So what we did was we took the convalescent plasma from these individuals that was collected at Rockefeller and then shipped to us. And then we took that and purified out the IgGs from the convalescent plasma. And so that way now we have only the IgG portion of the plasma that we then decided to study further. For structural studies, we had to cleave the IgGs into the Fab fragments. Again, so you have a bivalent Fab versus a monovalent, or bivalent IgG versus a monovalent Fab. And this allowed us to do some of the analysis that we wanted, including the negative stain and structural work. So from here, we purified the fragments and then tested binding as well as this negative stain MPIM. And so what I'm showing here is just a kind of a summary of some of the binding that we did. And so we took 10 different individuals and we tested, the potential of their IgGs, shown here in the dark, either blue and orange, versus the binding of the Fab fragment, shown here in light blue or light orange, against SARS-CoV-2 S proteins, shown here in blue, or the RBD, shown here in the 2 orange colors. And then we tested that also against SARS-CoV, MERS-CoV, that should be MERS-CoV, not MERS-CoV-2, as well as a number of common cold circulating strains. And so what you see for the SARS-CoV-2, at least, right, there's a range of binding in that they all bind pretty nicely to the S protein, but there is a differential range of binding when it comes to the RBD itself, suggesting that some of these individuals, like number 21 and 47, which show very high titers of RBD binders, these were, if you remember, some of the most potent convalescent serum in the previous study or slide that I showed, versus 107 that had very weak titers against the RBD. Right, so this correlation between RBD neutralizing titers or RBD specific titers in the convalescent plasma positively correlated with neutralization activity. And then another thing that we showed was that with these same individuals, they did show some cross-reactivity between SARS, which we expected, since it is 80% similar to the SARS-CoV-2 S protein, as well as some cross-reactivity to MERS. But what you don't see is cross-reactivity to the RBD in MERS, which makes sense because it utilizes a different host cell receptor. Whereas we did see some cross-reactivity between SARS-RBD, which at the time, was kind of collaborated or corroborated, validated some of the other studies that showed that you could pull out cross-reactive monoclonal antibodies that combine the SARS-RBD as well as SARS-2-RBD. So this all kind of matched and was nice to show. So now that we have this data, we want to know, okay, well, can we actually identify what epitopes are being targeted by this convalescent sera? And then whether or not we can then say if we can see actually figure out the cross-reactive epitopes, right? Because in this one individual, say like 134 or 42, right, you see very strong binding to the S protein, you see binding to the S protein of SARS, and you also see binding to the S protein of MERS. So, right, so can we actually identify this cross-reactive epitope between these three viruses? To do this, we turned to a technique that was first pioneered by Andrew Ward down at Scripps. And it was utilized really for the HIV field. And so this is called negative stain EM polyclonal epitope mapping. And in this case, what you're doing is you're taking Fab fragments, you're testing them for binding, and then actually you see binding, you're going to then complex this with an antigen. So in this case, we're looking at Fab binding after rabbit or immunization for HIV envelope. And then we're looking at binding to envelope of these Fabs. And so you can see a shift in, so we use SEC chromatography to purify the complex, so you can see a shift, and then we image it using negative stain EM to the point where you get these nice 2D classes, which then allows you to reconstruct antibodies floating around in the serum. Right, so this was done using the same pipeline for these SARS-CoV-2 individuals. And what we were able to show, just kind of skipping some of the methods, but we were able to show the predominant epitopes of two individuals, individual 21, individual 57. And so if you remember from the ELISA binding data, individual 21 had the highest titers against the RBD of its purified plasma. So it made sense, right, that the epitope we observed being targeted by its convalescent serum was to the RBD. Individual 57 actually had very low titers, but they still had strong binding to the S protein. And sure enough, when you look at where the Fabs, the predominant epitope of the Fabs from this individual, it seems to bind a domain in the S1 region of the protein. Individual 107, right, had very low titers altogether, and we didn't see any binding. So we could, this was the first time, to our knowledge, that this technique was used to characterize convalescent or antibodies floating in sera after an infection by any type of virus. And from structures that had been reported, we could kind of tell for two where these were binding. So again, we could fit in the model of the SARS-CoV-2 structure that had been solved and kind of understand where it was binding. And so in the case of the COV-21, again, we could see that it was binding to a up RBD. And at the time, there was no other structure of an up RBD with the antibody bound. So we compared it to the SARS monoclonal antibody S230 and showed that this was kind of an overlapping region on the SARS S230 with SARS S230, suggesting, right, that this antibody, this predominant epitope being targeted was most likely be a neutralizing epitope, since this antibody we knew was a neutralizing epitope in SARS. For the individual COV-57, again, we saw targeting to the S1A domain, and at the first, this was the first time showing binding of anything outside the RBD. And since then, there have been a number, I think one other structure reported showing targeting the S1A domain, and that structure actually was of a neutralizing epitope. So this suggests again that, the predominant neutralizing epitope targeted by this patient or individual may be neutralizing, or targets a region outside the RBD, which may be neutralizing. And so one thing we want to also determine is whether or not, these antibodies could bind with avidity. And so this is based off of modeling from a cryo-ET structure of a SARS virus at the time, and we knew the spacing between the S proteins, and we could see from the spacing that, targeting of the predominant epitope seen in individual COV-21 most likely represented epitope that could be targeted with avidity, whereas COV-57 could not. At least we thought so at first, or at least when this paper was published, we said so. But since then, the cryo-ET reconstructions of the SARS virus or SARS-CoV-2 virus has since been reported. And what we see in this virus is that, the S protein can adopt many different conformations relative to the membrane, this angle relative to the membrane. And when we look at this in the context of code57, we can see if you had two S proteins with this tilted angle side by side, you could probably likely bind with avidity in this sense, which kind of makes sense because in our neutralization assay, when we looked at neutralization of IgG versus neutralization of the Fab alone, if avidity effects didn't play a role, you would see you would see these curves very close together. But the fact that we saw, basically the same molecular nuclear ratio between the IgG and Fab, between code 21 and 57, suggested that 57 could bind with avidity, and this cryo-ET kind of helped explain that. So that was nice there. But again, how do we know these are neutralizing epitopes? So we decided to look at a monoclonal antibody pulled out of an individual 107 actually, which if you remember had very low neutralizing titers, but we were able to identify, or collaborators were able to identify a number of monoclonal antibodies from this individual that were potent. And so I decided to do single particle cryo-EM. So here's just kind of a quick flow through. We have particles in ice. We were able to image them, get these nice 2D classifications, and then solve a 3-dimensional structure. And what we saw, again, just kind of a workflow, but what we saw was two populations of this antibody called C105 in complex with the S trimer. And in this case, we saw a population that had 2 RBDs in this up conformation and 1 RBD down. And then we had another state where we had all 3 RBDs in this up conformation. And in each case, the up conformation showed the antibody or a Fab fragment bound to the RBD. And so what we concluded from this is that this class of antibody, the C105 antibody, could only recognize the epitope when the RBD in this up conformation. So how does that relate to ACE2? So we then mapped, of course, the epitope on RBD that C105 targets. And if you compare it to the region which ACE2 targets, you can see that this region really overlaps quite well with ACE2's footprint, right? So basically the mechanism of neutralization is most likely effectively blocking ACE2 from binding to the RBD. What was interesting though about this antibody and this class of antibodies is that it utilizes regions of the antibody encoded by the V gene. So these are the variable regions that are encoded, germline, and these are common amongst antibody strains, where you have the most diversity between antibodies in a certain class by the CDRH3. But it doesn't utilize the CDRH3. It utilizes these loops in this germline encoded regions, which is really important because not only did we not see a lot of somatic hypermutation within these regions, meaning antibodies usually mature by developing mutations to have higher affinity towards its antigen or its epitope. But in this case, they don't have lots of somatic hypermutation. And in fact, C105 has 1 mutation between both its variable light and heavy chains, right? So these basically are like similar to germline antibodies that are floating around in your body as a naive repertoire. And there has since been a number of papers reporting this phenomenon, in that these are very low mutated antibodies. And even naive human repertoires actually have these antibodies. So from a vaccine standpoint, it's actually really good because that means you can probably elicit these antibodies quite quickly in vaccines.
Dr. Barnes, you've got about 3 minutes left if you want to get some questions also into the mix here.
Yeah, this is the last slide. Okay. And so to finish up, so then we can also compare this binding to the code21. So this is just a low resolution blob of where the code21 Fabs would have been. You can see that it most likely overlaps with this epitope, again, suggesting that that epitope seen in the convalescent plasma of code21 was indeed a neutralizing epitope. Yeah, so I've already talked about this, but lastly, C105 represents this recurring class of antibodies that are low mutated and highly potently neutralizing. And we were the first to report this, but since then there have been a number of papers reporting neutralization by this class of antibodies called 3-53 or 3-66. So this is really great because you have a recurring class, low mutation that indeed could be potently neutralizes SARS-CoV-2. So with that, just quickly summarize, we tested binding, we showed the polyclonal response, we did the high resolution structures, and you can read more about a lot of work that I didn't talk about today in the paper that was reported in Cell last couple of weeks ago. So this was a huge effort, collaborative effort between our group and the group of Michelle Nussenswag and Paul Benash at Rockefeller. So thank you to everyone that was involved, including Davide, on Michelle's side, and of course, EM Centers and SSRL, all of our funding, and most importantly, the individuals who donated their plasma who were recovered from SARS-CoV-2. So with that, I'll take some questions.
Okay, a couple audience questions. First, to kind of set up the next one, what switches the RBD between up and down conformations?
Yeah, so it's not quite understood what switches it from up and down. We know it's just a highly flexible domain, and there are some studies being looked at to understand, these switches that could occur. And so there's a preprint out now looking at pH dependence on switching RBD from this up and down conformation, including our aspartic acid switch a network of switches. So people are investigating this, but it's not well understood what actually triggers the RBD to flip up.
Yep, and following up on this, I think you maybe real briefly mentioned it, but the question is, do you think the up and down movement is one of the major determinants for antibody binding? And how do you think that happens, which I think is kind of the same as the last one?
Right, so obviously the virus presents, from the Sars cryo-ET, we can see virus or spike proteins on the surface of the virus that have both this up and down conformation. And there have been a number of studies reported, and I think these papers will soon come out of antibodies binding to the down conformation. So it doesn't necessarily have to bind the up conformation to be neutralizing. So yeah, it can target either conformation, which is good.
All right, and last quick one here. How do you think the glycan shield on the spike protein affects antibody recognition and binding?
Right. So at the apex of where the RBD sits, there are two glycans that could potentially protect conserve epitopes, and actually it does. So the one antibody that binds a conserved core region that cross-reacts with SARS virus actually has a glycan there to try to protect that. So it uses glycans to kind of protect the core conserved regions, but for the most part, these antibodies don't seem to mind and can accommodate glycans quite easily.
Thank you very much.
Thank you.
Yeah. Dr. Stadler, are you here and ready to go? And you can just go ahead and start sharing your screen if you'd like.
I'm here. Here we go.
All right. Okay, our next speaker is Dr. Lauren Stadler, who is an assistant professor in the Department of Civil and Environmental Engineering at Rice University. Her research is focused on advancing sustainable and safe wastewater management systems, and she works closely with the City of Houston's Water Utility and Health Department. Before joining the faculty at Rice, Dr. Stadler earned her BS from Swarthmore College and MS and PhD in environmental engineering from the University of Michigan. Welcome, Dr. Stadler.
Thank you so much for that introduction. I'm going to assume everybody can see the slides and hear me.
Yep.
Good, great. Okay. So as I just said, I am an assistant professor in civil and environmental engineering at Rice. I am an environmental engineer and I study wastewater. And so I'll talk today a little bit about the work we've been doing to use wastewater to track community infection dynamics in the city of Houston. First, I want to start with some acknowledgments. This work has been a huge effort from a lot of people, really headed up by the health department and Lauren Hopkins, but also with the Public Works Department and Houston Water that's helped us collect these samples. We've been working very closely with the Taylor Group at Baylor College of Medicine, and I want to acknowledge our team here, Kathy Inzer in statistics, my research group, lots of graduate students, postdocs, research scientists who've helped contribute to this. And this started with a seed grant from NSF, NSF Rapid Grant. Okay, so to jump into it, what do I mean by wastewater surveillance? Very simply, we know that infected persons, both asymptomatic and symptomatic, shed virus in feces. And that feces ends up in the sewage network, and it travels through the sewer system to our centralized wastewater treatment facilities. And we can grab a sample there. And that sample represents an aggregate or pooled sample of the entire community that wastewater treatment plant serves. So really that wastewater sample is a really powerful sample in that it's this pooled sample that can be used to assess community health. So we can analyze that sample specifically and look for specific pathogens like SARS-CoV-2. And we use very similar molecular assays to actually detect this virus. So we use things that target the viral RNA. And that viral RNA can also be sequenced to get some more information about the diversity of strains that are present in a community out of the wastewater. So the form of these results, they can be qualitative, so similar to a clinical assay, positive or negative. Is there virus present in the community or not, based on the wastewater? It can be quantitative, so what is the concentration of virus present in the wastewater? Is that concentration increasing or decreasing over time? And then you also can, from the sequencing information, look at what strains are predominant in a community. And maybe how those strains relate to other strains that have been observed around the world. I'm going to talk mostly about using it in a quantitative fashion and how we use it to look at whether concentrations are increasing or decreasing to gain temporal and spatial information about infection prevalence in communities. I want to mention that there's a global interest in using this approach to track community infection dynamics. And this is just a map that shows, that aggregates all the publications or preprints that have detected SARS-CoV-2 in wastewater samples to date. So there's lots of people working on this. To illustrate why it is such a compelling and promising tool, I want to highlight two really powerful uses of wastewater surveillance. So the first is the fact that it could potentially be a leading indicator of COVID-19 outbreaks. So here are two figures that I pulled from a preprint out of a group from MIT. So the first one here on the left is showing viral titers, so the concentration of virus in the wastewater in the blue dots, and then new cases in the orange dots. And then the shaded portions are just the smooth data, so you can see the trends. But what this illustrates is that this blue shaded area definitely leads this orange shaded area. And when they looked at how much it leads, they looked at where the highest correlation between virus concentration and daily new cases was. They saw it was when there was a 4-day lag applied to the wastewater data. And so this study, as well as a number of others, have begun to suggest that wastewater, so the virus will appear in the wastewater kind of 4 days to a week, and maybe even potentially a little over a week before we start seeing that outbreak play out in the clinical case data. So if we think about that, it could be used as an early warning system to basically predict where and when we're going to likely see an outbreak in a community. And a week is a lot of time. If you think about how much could be done if interventions were put in place to stem an outbreak, a week gives very valuable time when it comes to implementing interventions. The second really compelling reason to use wastewater surveillance is that it is a more cost-effective and rapid way to do a screen of a community. This study from Hart and Halden said that if we just grab samples from the existing 15,000 wastewater treatment plants that exist in the US, we could basically cover 70% of the US population, and it would cost a tiny fraction of the amount that it would cost to do clinical testing of those individuals. And it's much more rapid in that you don't have to organize getting all these people to come and get tested and get their results and process them, because everybody goes to the bathroom. It also is unbiased because it doesn't rely on people having to volunteer to come in to get tested. If you wanted to do randomized testing, everybody goes to the bathroom. That wastewater has to go somewhere, and we can grab it at the wastewater treatment plant and use it to basically screen entire communities for the presence of this virus in the wastewater. So now I want to talk about 4 different possible use cases. So how assuming a community decides to implement wastewater surveillance, how are the different ways they can use this type of data? So the first one is for trend tracking. So basically looking at changes in concentrations of the virus in the wastewater over time. And like I mentioned earlier, this could be used for early detection of the virus, so second waves, for example, but also potentially look at how well interventions are working. So if you see a decrease or a plateauing of the virus in the wastewater, that could be indicative of an intervention being effective in an area. So, and I would say this approach is one what we feel most confident in as a scientific community and the ability to do right now, and that we can use concentration data over time to gain some information about how the infection is burdening a community. Another possible use case, which I'd say we're a little bit, has a lot more uncertainty associated with it, is being able to take a viral RNA concentration that we measure in wastewater and actually use that to back calculate the estimated infection prevalence in a community, so the number of infected people. And that's just because there's a lot of uncertainty associated with how this signal decays in a wastewater system, how much virus is shed per person, through the course of infection, whether those shedding rates are different between asymptomatic or pre-symptomatic or symptomatic, asymptomatic individuals. But I think this is one of the ultimate goals of this, is basically being able to come up with accurate predictions of infection prevalence from wastewater. Another potentially very powerful use case is to look at viral evolution. So as many of the other speakers today have talked about, we can look at the diversity of circulating strains by looking at them in the wastewater sample. And this could be very powerful because it also picks up on a lot of the asymptomatic fraction, again, because everybody goes to the bathroom, and if the asymptomatic population represents a large burden of the infection in a community, this would be picked up, and we could understand better if the diversity of circulating strains is different when you have a really large asymptomatic population in your community. And then finally, identification of geographic areas of concern. So in addition to just looking at temporal changes over time, we can do targeted wastewater surveillance where we collect samples from more specific communities and do spatial analysis and identify which areas appear to be bearing the largest infection burden based on their wastewater data. So one question that we have to ask if we want to use this approach is how sensitive it is. So basically, how many people have to be infected before we're able to pick it up as a signal in the wastewater. So here I'm just showing this heat map from the Hart and Halden study that's saying that this is a very, the sensitivity of this approach is a function or very sensitive to the wastewater generation rate, which is basically how dilute your wastewater is, or how dilute the signal is in the wastewater, and also very sensitive to the fecal shedding rate of the virus, and that's on the x-axis. But if we kind of show a typical wastewater treatment plant that serves a population that produces wastewater at about 300 liters per person per day, which we have a number of treatment plants in Houston that kind of fall in this range. And we look at reported fecal shedding rates in the literature to date. Wastewater surveillance approaches have the potential to be sensitive enough based on what we know about a limits detection of this method to pick up one infected person in a population of 30,000. Again, this speaks to kind of the power of this approach to be a pretty sensitive method. There's a lot we don't know about how much fecal shedding occurs and the range of fecal shedding in a population. But again, picking up a 0.003% prevalence is a powerful tool. There are a number of things that are going to impact the signal of the virus in the wastewater and our ability to measure it. And I just want to point to a couple of these. So aside from just fecal shedding rate and wastewater generation rate, we know that this virus this wastewater travels through our sewer system, and it spends on the order of hours and sometimes up to maybe even a day in very, very large systems before we pick up that sample at the head of a wastewater treatment plant. And so that viral signal will undergo decay. That's very temperature dependent. It will be diluted by lots of things, including infiltration and inflow. And we also know that signal will be much stronger from the people who live close to the treatment plant, as opposed to kind of the distal contributors, the people who live very far from the treatment plant. And so we're trying to develop better models to basically capture that. But another approach that we're taking is by doing some more targeted sampling in manholes and lift stations, where we actually go into the sewer collection system and collect samples that serve smaller neighborhoods, so that we can get more spatially resolved information about virus concentrations. And so now I'll tell you a little bit about what we're doing here in Houston, and show some data that we have indicating how we're going to use this data. So here I'm showing Houston's wastewater sheds. So Houston has 39 wastewater treatment plants, which is actually kind of unique. Most major metropolitan areas really just have a handful, maybe 1, 2, or 3 major wastewater treatment plants. We actually have 39 treatment plants in Houston. In this case, I think it's a huge advantage for doing wastewater-based surveillance, because we can collect samples from each of these treatment plants, and we can get this spatial resolution based on these individual sewer sheds and get a picture about what's going on in each of these individual sewer sheds by looking at their wastewater. So we've been collecting weekly samples and quantifying the amount of virus present in them. I'm not going to spend time talking much about our methods, except for to say we have spent quite a bit of time developing a more sensitive method because this virus is extremely dilute in wastewater, and wastewater is also full of lots of things, as you can imagine. It's pretty, it's a messy sample. And so we've had to develop both a very sensitive method, but also a high throughput method, so that we can turn around data in a time frame that's useful to make decisions with. So I'm going to jump into showing a little bit of the data that we have so far. So here, and I'll talk about how it's being used. So here I'm showing all the different sites that we've been monitoring over the past few weeks. And the 2 different color lines are just the 2 different targets that we've been quantifying. So it's the CDC N1 and N2. And the way this data is being used is to both track trends. So basically look and see where we see significant increases in viral loads over time. And as you can see, generally, if you look at all of these, as a big picture, we see this increase in concentration over time. And some of these, it's hard to see this axis, but I'll say that these are often several log fold changes, even these small ones. And the other thing we can do with this, because we have 39 treatment plants, is we can do this spatial analysis to identify hotspots. So you can look at this and you can very quickly see, when you look at all these panels together, that there are specific panels that we see, bigger spikes in some areas than others. And so this allows basically the health department to use this data to identify geographic areas of concern and use this information to decide where to deploy additional resources, where to do additional clinical testing. And the idea is that you could have, even if you don't have 39 wastewater treatment plants, a city could decide to implement a wastewater surveillance program, pulling from manholes, and identify geographic areas of concern just by looking at the wastewater of the communities that serve that manhole. And so as a community, I think ultimately we want to be able to use this data and compare the data that we're generating in Houston to data that's being generated all over the world and across the US. And so, for example, being able to compare the concentrations and trends in wastewater concentration data that we're seeing in Houston to other places. But the challenge right now is that there's no standardized method associated with measuring virus concentrations in wastewater. So it makes it nearly impossible for me to compare the concentrations of virus that we see in wastewater in Houston with, for example, one from the Netherlands, because you used a totally different method. And we know that there's huge differences in method recovery and sensitivity when you change the method that's used. There's also a huge difference just based on the characteristics of the wastewater that impact the ability to compare across sites and across studies.
Dr. Stedler, I just want to let you know there's about 3 minutes left and there's a few questions in the queue.
Okay, got it. I will, this is the last slide. So
sounds good.
All I want to say is what we're doing, because there's no standardized method, and I actually don't think there should be, because we need to figure out what the best method is before we say this is the way to do it. We're spiking in internal controls. So we spike in field spike a bovine coronavirus, an attenuated bovine coronavirus in the field. We can quantify how much of that virus we get back because we know how much we put in. And I just want to say here, you can see there's quite a bit of difference just in our 40 treatment plants that we're monitoring here in Houston. And our recoveries range, it's made, from close to nothing, it looks like, though we are detecting virus in basically all these samples, to 10%. And so I think these internal controls are extremely important as people begin to do more wastewater surveillance across the country. So that's all I have. I just want to say I think this is a really powerful tool that can help use to inform the efficacy of interventions, enable more targeted direction of resources, and it's sensitive, rapid, and inexpensive. So I'd be happy to take any questions.
All right, we have some questions. We also have some compliments. I guess I'll just state those real quick. One is "this is amazing," and one is "this is extremely interesting and with lots of potential." So maybe we'll start there. As far as questions go, and I agree with those. As far as questions go, is it possible to use this technique in more populated communities, especially in countries of South Asian continents where there are a lot of other factors that come into play in wastewater?
I guess I'm not sure what you are referring to in terms of other factors, but I actually think this is a tool that could be used globally, and that it is significantly less expensive as a monitoring tool than kind of traditional clinical testing approaches. So I do think, and the methods are relatively straightforward in that, there's a concentration step, and then there's the similar molecular tools that we use to do the clinical assays. So yeah, I think it's very possible to do this. And I think there's a lot of groups that are working to try and implement this, all over the globe.
All right. What about looking for like micropopulation trends? For instance, like you see with spike D614G, are you able to look for like micropopulation trends and also data publicly available? Yes, no, on these kind of things?
Yeah. I don't know for the answer to the first part of the question. I think if somebody wanted to try that, that'd be really exciting. We haven't done any sequencing of the viruses from these samples yet. I think there are some other groups that are working on that. I think this, again, this wastewater samples have so much potential. So I think if we excite someone on this call about getting interested about doing this, I think that's something that certainly could come out of this. What was the second question?
Actually, I don't see it anymore, but let me throw this one in as the last one. What about the viability of the recovered virus from the wastewater? Is there anything
that's a great question. So this doesn't indicate viable virus. In fact, it's very possible we're just detecting viral fragments. There are groups trying to see if it is viable to assess risk, right, via water spread, but there's very little indication, I think, based on what we know right now, that much, if any, of the virus that we find in wastewater is viable. I don't want to say none of it is, but I don't think anyone has recovered viable virus yet from wastewater. People have from stool, so it's possible that it is also viable in the wastewater, but I don't know if we know enough yet to answer that question.
Thank you very much.
Thank you.
Dr. Milton.
Quick question.
Yeah, I unfortunately, I'm sorry. I think maybe she can ask in the chat. Dr. Milton, are you ready to go for the for your talk? Are you here? We can't hear you. If you are on mute. By the way, I do see you logged in.
Okay, I'm here.
Okay.
Right.
Yep. Feels free.
Apologize. One of my mics died yesterday, so I'm using an alternate one. I hope it's working.
Yeah. Yep.
And so let's see.
Yep. And if you want to just go ahead and share your screen there. And I'll give you a quick intro here. Our next speaker is going to be Dr. Don Milton, who is a professor of environmental and occupational health at the University of Maryland School of Public Health. He has 40 years of experience in occupational and environmental health research and clinical practice with a focus on bioaerosols. Among his contributions are a mathematical model of airborne infection transmission risk using simple carbon dioxide measurements and 3 patents on exhaled breath bioaerosol collection and analysis methods. A recent paper in Nature Medicine using his methods demonstrated the potential for surgical masks to limit shedding of seasonal coronavirus aerosols from infected patients. And it looks like I can see your screen. I think you're ready to go. We can't see you, just by the way, as long as you're fine with that, just letting you know.
Well, I turned on my video, I thought, but and the camera says it's on. So I've had a lot of trouble with this. Zoom has been acting up funny recently.
Yeah, no problem.
All right. Well, thank you. I'm happy to be able to be here today and give this talk. I'm going to talk about infectious drops and aerosols and about the modes of respiratory virus transmission, and really thinking about this in the context of SARS-CoV-2 and much of the confusion about transmission modes and the different languages that different groups of people are using that I think is resulting in a lot of confusion for many in the public about how to interpret CDC and WHO, and then groups of aerosol scientists and aerobiologists pushing back about the term airborne infection. So let me talk about transmission mode in the context of saying that we have contact transmission, patient to finger, finger to nose or eyes, or patient to fomite, fomite to finger, and then finger to eye, nose, or mouth as one mode of transmission that we believe can happen. Although you'd be surprised at how little actual evidence there is for this mode for any infection. Even with rhinovirus, where it's thought to play a big role, attempts to transmit rhinovirus by having people play poker with cards and notepads and pencils for hours until they got soggy, then taking them to another room and giving them to another group to use, and forcing those people to stop every 15 minutes and pick their nose and rub their eyes, was not able to transmit rhinovirus. The only way it could be transmitted by contact was by having someone cough on their hand while their hand was still wet, shake hands with somebody else, and then have that person immediately inoculate their nose. That works. So contact is something we believe in, but has never actually been proven, particularly around fomites for respiratory viruses. Splash and spray transmission, where you have large drops or droplets that transmit, where they are released from the source in with momentum travel and direct hit on the target of your eyes, or nose, or mouth, or contaminate objects in the vicinity and then end up with fomite transmission. But direct splash and spray can be an issue, especially for people who are up real close to somebody and their cough right in your face or sneeze right in your face. Inhalation is where droplets are floating in the air. They are not being carried by momentum, but rather by air currents. They are being moved by the air molecules around them, and it can range in different sizes. And we speak about them in exposure science as inhalable, thoracic, or respirable, depending on where they land in the respiratory tree. So you will sometimes hear, and there's an article out in JAMA this week saying that, well, let me go back here. It was saying that, we're talking about droplet transmission and aerosol transmission, and defining aerosols as only those particles less than 5 microns. Yet we know that aerosols are much larger, and I'll talk more about that in a minute, that aerosols can cover a wide range of sizes up to 100 microns, and some would argue that ballistic droplets are aerosols too. I don't use that term for them because I think they have they're kind of a different mode. But inhalation means that it gets, you're breathing it in. But medically, we tend to talk about droplets in a different way. There's also in that article saying that, well, you know, SARS-2 can't be airborne because it's not like measles. Measles has an R naught of greater than 10, 18 or something like that. And but ignoring that there's another known aerosol transmitted respiratory infection called tuberculosis that has a much lower R naught. And one of the key differences and signatures between these infections is that in tuberculosis, you have a very high rate of infectious dose generation. In measles, you have a very high rate of infectious dose generation, and in tuberculosis, it's much lower. You can see here that we're talking about orders of magnitude difference going from 2 to 10 per minute for a few days for measles to something around 1 per hour for months from tuberculosis. So clearly, if people didn't shed tuberculosis for months, they probably wouldn't infect anybody. The target in both cases can be the alveolar macrophage, but for measles, airway dendritic cells, which populate the airways all the way up to the larger airways and even into the nasopharynx, can be a target. Whereas, and then one other argument that is said is, well, you know, you can detect the RNA in the air, but we've never been able to culture it. Well, in the case of measles, there's only one report of anybody ever sampling the air using molecular methods, and they did detect RNA in the air, but they weren't able to culture it. Furthermore, they found that there was a lot more on surfaces, and it was much easier to detect on surfaces than in the air. On the other hand, with tuberculosis, nobody's ever been able to detect it in the air in a medical facility. People are just not shedding enough into the air, and unless you're right where an aerosol is generated as you're doing bronchoscopy or something, it's impossible to find it, except by having people cough through a tube into a box where you contain the aerosol and focus it right on the sampler. Then people have been able to collect it and culture it. The dogma for a long time, up until the 1950s, was that TB was transmitted by large droplets and contact because it was easy to get from surfaces and because prolonged close contact was required to transmit infection. Now that prevalence is very low, it's possible to see that in fact people can transmit it over long distances. But back in the day, you couldn't tell that because it was everywhere. So measles, on the other hand, you don't necessarily have to have prolonged contact. It can be very brief because the dose rate is so high that the person is going to load the room up with infectious droplets. And long-range transmission, again, nowadays you can identify that this happens because there's not much of it around. But when it was everywhere, it was very hard to tell where it came from. Face masks have been shown to be effective as source control on TB patients to preventing transmission of infection. This work was done fairly recently in South Africa in an experimental facility where the TB patient's breath is monitored by feeding it essentially to guinea pigs. Face masks had not been looked at in measles. So why focus on less than 5 microns and call that aerosols? Well, some of that has to do with this work that William Wells did, showing that if you expose the animal on the left to TB aerosols in very large particles, coarse aerosols larger than 5 microns, they didn't get TB. Whereas if they were exposed with smaller aerosols, they got TB. The reason is because to get TB, they had to get an infection into the alveoli to infect the alveolar macrophage. So another aspect of this story is that influenza has been shown to be anisotropic. That is, the dose and route of infection matter. That with aerosol inoculation of humans, the dose can be vanishingly small, around 1 tissue culture infectious dose, 50%, is enough to infect humans who don't have antibody against that strain of virus. And it reproduces full-blown influenza-like illness with fever, even at that tiny dose. Sometimes at that dose, people developed prolonged wheezing for up to 6 days and nausea and vomiting. I mean, it was like they had fevers of over 39 degrees. They were sick. Nasal inoculation, on the other hand, requires a 100-fold higher dose in order to initiate infection. Usually they just seroconvert and have no symptoms at all, but sometimes they'll have very minimal sniffle. To get influenza-like symptoms, you have to go much higher, up to 80 to almost 200,000 TCID50s. And fever and lower respiratory symptoms are rarely seen with this. Even at those high doses, they'll have good upper respiratory cold symptoms though. And this was shown first in the 1940s, later in the 60s at Fort Detrick. But since that time, people have not been using aerosols to inoculate people because it makes them too sick. So where does the Sars virus bind and infect? The recent work looking at the distribution of ACE2 receptors shows that they are plentiful in the bronchus, in the lung, and the conjunctiva. And that the SARS-CoV-2 virus can bind very effectively at all these sites. The SARS-CoV virus, which has lower affinity for ACE2, doesn't bind as well, particularly at the conjunctiva. Also, SARS-CoV-2 binds quite well throughout the oral mucosa. So we can get it in the mouth, we can get it in the eye, the nose, bronchus, and all the way out at the alveoli. So the entire respiratory tract is a target for this virus. Is it more effective? Does it require lower dose at one level or another? We don't know that yet. But we do know that the co-expression of proteolytic enzymes is important for entry of the virus, and how that plays out with the expression of ACE2 is still to be determined. So where do aerosols deposit? Well, things bigger than 10 or 15 microns deposit in the nose. When you get below that, you begin to get position in the tracheobronchial tree, labeled TB here, or in the alveoli, labeled ALV. And you can see at about 2 microns, you have a peak of deposition in the alveolus. But even at 2 microns, most of it is getting into the head airways. Even at 1 micron, you have a large fraction in the head airways. So even with fairly small particles, you're getting a lot of deposition in the upper airways, but as they get smaller, they start penetrating deeper and deeper, and you can see that there's a lot of overlap here between these curves. So,
Dr. Milton, you got about 3 minutes left, including question time. Just want to let you know.
Okay, so we can define things either as respiratory droplets or as aerosols. And in exposure science, we talk about these things as aerosols across a range, not just less than 5 microns. indoor air, we worry about how far these droplets travel. But you know, even though they settle fairly quickly after a few minutes, indoor air is not still air, it's moving. And so even 20 micron droplet can move 4 to 15 meters indoors. And when we've tried to do transmission studies with influenza, we find it's very difficult to get people who are nasally inoculated with the virus to transmit. The results of this study published earlier this week suggest that ventilation is making a difference in influenza and reducing the attack rate, and that fomites and large droplets, splash and spray are not very important modes of transmission. In SARS, we know that in the Amoy Gardens, we saw transmission between buildings by aerosols, and MERS has been cultured from the air. We know that the virus is present in fine particle aerosols, as shown over here, based on studies done in a containment room in Singapore, as well as work in Wuhan hospitals here with even smaller diameter particles, and at the University of Nebraska Hospital, where a particular concern is the high rate of isolation and personal samples of healthcare workers. This outbreak in a restaurant in Guangzhou clearly showed that transmission can occur over much more than a 1 to 2 meter distance here, and seems to be due to recirculating of air without exhaust. And our studies have been working on looking at virus in exhaled breath, very directly collecting it with this device and testing masks. And we have shown that masks work very well to eliminate the coarse aerosols, and they reduce the fine aerosols to some extent, and overall cut out about 2/3 of the virus shed into the air. With coronaviruses, that works quite well. This paper published in April, it may not, with larger sample size, look quite this good, but we definitely see an effect. We are now recruiting cases. We're doing 2 cases today. We have some coming in tomorrow as the second wave that seems to be ramping up in Maryland. We are actively recruiting to test how much virus is in breath, working with Matt Freeman around culture and looking at masks and maybe face shields as containment. And I want to thank the team of people that make this possible. Thank you.
Thank you very much. Someone asked about a fan. I think you already kind of showed that a little bit in that figure. So the question I'm going to ask is, should N95 masks be used by the public in certain settings where aerosol exposures are more likely?
Well, I think that what we need to be doing in public places is not having, the 3 C's: closed environments with poor ventilation, crowding, and close contact. And when you line these 3 things up together, that's when you're in trouble. And so if you have to have a group of people in a closed environment with poor ventilation, and there's a lot of people and they're going to be close together, well then, yeah, maybe you need an N95. But the real answer is, don't do that.
Okay. All right, thank you. I think we're going to have to switch to our next speaker. Thank you very much, Dr. Milton.
Thank you.
Thank you. Adrienne, are you here and ready to go? If so, you can go ahead and just share your screen. Our next speaker, great. Our next speaker is Adrienne Gladden-Young, who is a senior research associate in Participetti's lab at the Broad Institute of MIT and Harvard. She's an alumna of Tufts University and the University of Maryland. Adrienne is focused on advancing laboratory methods to enable genomic studies of infectious diseases. She's been crucially involved with the lab's efforts to sequence and track the viruses responsible for some of the world's most severe and deadly outbreaks. She's led efforts to sequence Ebola, Zika, hepatitis A, and numerous other viruses. Most recently, she's leading efforts to sequence and publish genomes of SARS-CoV-2 amidst the COVID-19 pandemic. Welcome, Adrienne.
Great, thank you. Thank you for
and
that.
I can't see you, by the way. I don't know if you want to show yourself or not. You don't have to, but just letting you know.
Okay. Okay, yeah, that's fine. That's fine. Okay, all right. Thank you. And thank you for this opportunity to share our work. I'm going to describe the work that we've been doing to understand the spread of COVID-19 in Massachusetts through sequencing and phylogenetic analyses of SARS-CoV-2 genomes recovered from patient clinical samples on behalf of the Subedi lab at the Broad Institute of MIT and Harvard, Massachusetts General Hospital, the Massachusetts Department of Public Health, and our many other partners. So what can we learn about viral genomes? Well, I know we've been talking about it a lot lately, so this is probably redundant, but I think we're seeing during this pandemic just how valuable viral genomic sequencing is. And we are able to detect emerging pathogens and learn more about their structures and function. We can use sequence analysis as an epidemiological tool to understand transmission and the ways that pathogens change over time as they adapt to different hosts. And we can use to help explain clinical data and to reveal targets for diagnostics, treatments, and vaccines. So again, sequencing technologies have become increasingly valuable for the study of infectious pathogens. And there are many ways to sequence their genomes. We used, for this study, we used the metagenomic sequencing approach to sequence all of our viral genomes. And it's useful because it's unbiased and it allows us to sequence everything. That's all the genetic material present in samples, including pathogens that cause new or unknown infections or co-infections without needing any information about what's in the sample. In fact, metagenomic sequencing made it possible to identify SARS-CoV-2 before we knew much about it. And here, phylogenetics, which is the study of the evolutionary relationships between different organisms, made it possible to characterize SARS-CoV-2 as soon as the first genome became available. Here, based on the virus's relationship to other viruses, it was found to be a beta coronavirus with a single-stranded positive sense RNA genome that's about 30 kb long. So, for our study, we use phylogenetic analysis of SARS-CoV-2 genomes as an epidemiological tool. And as we're able to collect more data, we've been able to look at places like Massachusetts, and more specifically in cities like Boston, to learn about the ways that the virus is spreading and being transmitted through outbreaks in these areas, and how different cases or clusters may be linked together. So far in Massachusetts, there have been 106,000 COVID-19 confirmed cases and more than 8,000 deaths. And our sample set includes the first of these confirmed cases in Massachusetts, and nearly all of the positive test samples, mostly from the Boston area, starting from early March, when testing and sample collection began in Massachusetts. Our sample set also includes samples from suspected super spreading events in different parts of the state. So how did we sequence the viral genomes? Well, we attempted to recover viral genomes from patient clinical MP swab samples. Our method takes about 2 to 3 days to complete and consists of RNA extraction, then removal of human and bacterial or other background RNA to enrich for the viral RNA, then cDNA synthesis, unbiased RNA library construction, sequencing, and reference-based assembly. So we were able to sequence 772 complete or nearly complete SARS-CoV-2 genomes. And as expected, we observed a strong correlation between the sample viral load and mean genome coverage. So here's the tree with our the genomes that we sequenced, and it also includes approximately 10,000 genomes subsampled from the global GISAID data set. Well, there's a lot here, and I'll focus on a few key aspects of the data, but it's freely available, and you can find our interactive trees and explore the data more here at this site. Sorry. Trying to get that. Okay, sorry. Okay, so here's our tree again. The Massachusetts genomes are in red and can be seen more clearly here. In the background, you can see the European genomes are pink, North American genomes are green, and genomes from Asia are blue. And here is the first sequenced genome from Massachusetts. And we can see that, that most of the genomes are closely related to ones from Europe and other parts of the US. And the genomes are scattered throughout the tree. It looks like there are many, at least tens of different introductions of the virus into specifically the Boston area during the early part of the epidemic in Massachusetts. And also, by looking at the relationships of the genome sequences, we can start to notice clusters where many cases or genomes are closely related or appear to be linked by one common ancestor. And it's important to understand these clusters. They may present as sharp increases in case counts in a certain geographic area or during a specific time period. And in that case, it could mean that it's super spreading, that there is a super spreading event that occurred. And so it's thought that super spreading, which causes unusually high numbers of infections linked to a single source, has been a major contributor to the spread of SARS-CoV-2 during this pandemic. And we've seen, super spreading events reported repeatedly throughout this pandemic in churches, congregate living facilities, prisons, and other places. So we for this study, we intentionally sampled from several suspected super spreading events in Massachusetts. And nearly half of our data set includes genomes that could be linked either directly or indirectly to these events. So I'll briefly describe two examples here. The first, okay, so in late February, there was a large business conference held in Boston. Conference participants traveled from all over locally within the United States and internationally to attend this conference. And it took place in the same building over 3 days. And when it ended, everyone traveled back home. Well, a few days after that, some people who had attended the conference, they started developing symptoms of COVID-19, and they were tested. And I think eventually, well, at least most of the people associated with the conference were eventually tested. And we were able to sequence 28 complete SARS-CoV-2 genomes from a subset of the clinical samples that were collected from patients directly associated with the conference. All of the conference-related genomes, including the ones from outside of Massachusetts, appear to be linked to a single source. And they appear to have what appears to be clade-specific mutations, which we later see in other genomes from cases reported weeks after the conference ended. So we were able to identify many cases that seem to have descended from this event, indicating widespread community transmission in Boston.
and spread to other parts of the US and the world. Okay. And now this second example, it took place in a skilled nursing facility where residents were mostly senior citizens, and they were preparing to move to a different location. So because of their older age and pre-existing health conditions, this population was considered to be a physically vulnerable group, and it was recommended that they be tested for SARS-CoV-2 as a precaution before relocation. At the time, there was no COVID-19 outbreak that was suspected because none of the residents or staff had reported any symptoms at the time of testing. But eventually what we found was that 85% of the 97 residents and 37% of the staff tested positive for SARS-CoV-2 before showing symptoms. And we were able to sequence 63 complete genomes from clinical samples collected for testing. Interestingly, while we did find that the virus was introduced a few different times into this facility, 90% of the infections came from a single source. So here you can see the cluster here on the tree, and then zoomed in here, you can see the clonal expansion of the virus here. And this shows just how easily the virus can spread in congregate living spaces when just one person is infected. Okay. So for future directions, again, we see just how significant super spreading events are and how focus on them will be important for outbreak containment efforts. So, we can start to look to see if there are any phenotypes associated with super spreading and which characteristics, if any, allow these viruses to be transmitted so easily. Also, our data set holds very valuable information that can be used to characterize early infection, especially during the pre-symptomatic or non-symptomatic phase, that as we saw at the skilled nursing facility. We can ask questions like what do we see in the viral genomic populations during this non-symptomatic phase? Does this determine how the virus will spread or which viruses will be transmitted? And just in general, it'll be important to confirm the timeline for the start and spread of global outbreaks, and more specifically the outbreaks in Massachusetts, so that we can better understand these evolutionary relationships that we've seen in our data. And viral genomic sequencing will be useful to understand, especially as the more society we're returning back to normal. If there are any flare-ups, we can understand this, the sources of these flare-ups and continue surveillance. So there's a lot that we can pull from this valuable data set, and we hope that it can be a resource to inform public health efforts during this pandemic and beyond. So I just want to say thank you, especially to Dr. Party Sabetti, who's the PI of the Sabetti lab, and all of the groups at, this was truly a team effort. And so I thank all the groups at the Broad and Mass General Hospital and Mass DPH, and a special thanks to NIH and Illuma for their generous donations.
All right, thank you very much. So a couple questions here. A lot of the stuff that you're describing, following the different phylogenetic trees and following the transmissions and the epidemiology, there's been a lot of talk of the D614G mutation. In fact, today it's been brought up a few times. Can you say anything about that?
Yeah, well, we have seen that mutation in our data set. In fact, we saw increased as time went on, and it increased in frequency. The mutation increased in frequency, but other than that, we haven't drawn any conclusions yet.
Sure. And as far as the metagenomic sequences, has that revealed any information about like co-infection rates?
Yeah, well, so we have seen some co-infection, just like common respiratory infections, but not a lot. It could just be because we were, when we started sampling, we were at the end of our, we were at the end of the flu season, and just the season for other respiratory infections. So we could have just missed a lot of the co-infections.
Mm-hmm. All right. Well, thank you very much for joining us today. Oh, yep. Oh, yeah. We have time for one last one.
Adrian, that was fantastic. Great job. Thank you so much for giving that and doing your work. So when you look back at those sentinel events, can you tell, like in that healthcare facility and the super spreader and the Biogen conference, could you tell whether it was on the exact same day or was it spread out over multiple days? Were the people that got the infection on different phases? And then what were the states of the individuals that were super spreaders? Were they actively sneezing? What kind of fluids were they putting out, and how were they, kind of interacting with fomites and everything like that?
Yeah, well, interestingly, in the nursing facility, no one had any symptoms, or at least no one reported any symptoms. So it's possible that maybe they missed some symptoms, but for the most part, no one had any symptoms. So that's what's so interesting, because this is truly the earliest, very early infection. So I think we haven't yet, but we will be able to go back and look at the genomes to see, what do we see there? Is it, just one virus that was transmitted at the time, or is there maybe a dominant virus, and then, some other virus that also, snuck through and was transmitted. It will be interesting to see what we uncover once we go back and look at our data again.
Is there any chance that there's spreading from diarrhea or fecal materials rather than sneezing, coughing?
I don't know. Maybe it's possible. I'm not sure. Because there were no symptoms reported. I don't think it was like it would have been likely at that time. I would think that they would have noticed, increased diarrhea or other symptoms that they could connect to the transmission of the virus, but there are just no symptoms reported. So.
Okay, one last really quick question from Michael Yakum here, actually. And I think actually several of us are probably wondering this, so good question. Are the raw sequences for the samples available?
Yes. I have it in the slides. Yes, all of the sequences are publicly available.
Yep. All right, great. Thank you very much. And thank you for joining. All right, Dr. Handel, you're up next, and I see you are here, so that's great. If you want to just go ahead and share your screen, and I'll just give you a quick introduction here.
Perfect.
Okay. Our next speaker is going to be Dr. Melissa Handel, who is the director of the Center for Data to Health at Oregon Health and Science University and the director of Translational Data Science at Oregon State University. Her background is in both wet and dry lab translational science, with a focus on the development of ontologies, semantic engineering technologies, and open science infrastructure programs. Dr. Handel's vision is to weave together healthcare systems, basic science research, and patient-generated data through development of data integration technologies and innovative data capture strategies. She co-leads the Monarch Initiative, an international consortium dedicated to utilizing model organism genotype-phenotype data, deep phenotyping, and graph-based integration techniques to improve rare disease diagnosis. She also co-leads the NCATS Data Translator, which aims to integrate hundreds of data resources for mechanism and drug discovery, as well as co-leading the GA4GH clinical and phenotypic workstream, supporting the development of standards for clinical genetics and improving access to data across the world. welcome Dr. Handel.
Thank you very much. So it's really a great pleasure to be here, especially kind of wrapping up the day. I basically want to sort of give you an overview of what we're doing in the clinical data space, but with a challenge to you all to sort of have a think, which is why I think it's wonderful that I kind of got to go towards the end of the day, because what we really need is to be able to connect some of the amazing work that's going on in all the different contexts, everything from the talk we just heard about understanding how we can do public tracking. And I am now losing this because my sister is texting me. Sorry. Where did my, sorry about that, guys. Just lost my window. There we go. I don't, for some reason, sharing again. Nothing like having a technical glitch in front of 200 people. We've all been there. Okay, there we go again.
Okay.
All right, so
you can see it.
Okay, perfect. So the idea is that we've been really thinking about how do we help support the use of clinical data in combination with many other kinds of data modalities, everything from sewer tracking and public health monitoring to viral genome tracking to public applications that patients themselves are downloading and using to track their own symptoms, wearable devices, and all these kinds of data sources that fundamentally depend in our understanding of clinical outcomes and how to actually reduce the severity of this disease. How do we actually best leverage the clinical data to support all of the different kinds of things that we've heard about throughout all of today, and how do we connect those data in meaningful ways? So today I'm going to tell you about a national initiative called the National COVID Cohort Collaborative, or N3C. This is an initiative that's really led by a very large number of people. In this country, we do not have a common care system for a common centralized healthcare system, and therefore we do not have a common healthcare data system in this country. And so if we want to do really large-scale, robust machine learning and discovery-oriented applications to understand those clinical data, we really don't have the data that we need in order to discover those things. And so, kind of towards that notion, this pandemic really highlights these urgent needs, not only to understand this new disease by understanding the data that we're seeing in the clinic as the disease and pandemic progresses, everything from diagnosis to triage to predictive algorithms, understanding how to best care for individual patients in a more precise way. We have a desperate need for data harmonization and integration across all those spaces that I just mentioned. Decision support for resource allocation as our hospitals get filled. How do we best make the best decisions in the most ethical manner? How do we expedite drug and mechanism discovery? Clinical guidelines to reduce disease severity. How do we decide them and how do we get them out the door as quickly as possible? And then how do we understand whether or not they're actually reducing disease severity across the nation? Improved coordination of biomedical ecosystem overall to maximize efficiency. And while I'm here in the US and many of you are as well, this is an international consortium. And so all of the barriers that we have in sort of moving data into and out of a given country is also a big challenge. And so all of these things are benefited by the creation of a comprehensive clinical data set. So how do we actually do that? How do we actually aggregate and harmonize clinical data for accomplishing all of these goals, knowing that we really need certain PHI, personal health information data in order to understand the pandemic, specifically geolocation and time of service or temporality. So the N3C is set up into 5 different work streams, and they really serve kind of the main functions of the program to help us create exactly that, an environment where we can
aggregate and harmonize the clinical data from across the US. So it starts with data partnership and governance, which has relied very heavily on partnership with the community, the NIH, as well as different clinical data model networks and data sharing communities such as ACT, Trinetics, PCORNet, and OMOP Odyssey, without whose partnerships the N3C would not be realizable. So each of these clinical data models has a large group of organizations that have instantiated clinical data in those models that can then be drawn upon, and their expertise has been incredibly valuable in instantiating the N3C. So then the phenotype and data acquisition is in charge of deciding which phenotypes, which patients to include in this cohort of patients that we're aggregating data about. And then how to actually get the data out of those institutions and into a centralized location, which is located at NIH in the NCATS cloud, where the data ingest and harmonization team takes on the data and works to harmonize the data from across all the different sites and all of the different, in whatever flavor clinical data model that site might provision the data with a lot of data quality assurance. And then we have our collaborative analytics environment where we have a secure enclave, which allows software tools and analytical workflows of whatever innovations and ingenuity the community can come up with to be able to run those workflows over these aggregated and harmonized clinical data that provisions access to patient level data in this secure enclave. Because it is a secure enclave and no data can go out, we also have a synthetic clinical data pilot where we're working to determine if we can create synthetic data as a derivative from the PHI containing row level clinical data that's located in the collaborative analytical environment so that we can more widely distribute it. So I'm going to give you an overview of each of these, some of the activities of each of these things that are going on with an eye towards some of the covert community and how we can actually try to join forces. So the data partnership and governance is led by Christine Suver at Sage Bionetworks. And I should also mention that I'm obviously not at this alone. I have two key partners, Ken Gersing, who's really led a lot of the NIH aspects, but in particular the governance, and also Chris Shute, who's leading data harmonization, which I'll talk about in a minute. So on the data partnership and governance, there's quite a complex set of regulatory structures that have been put into place. And one of the biggest things that the N3C has really overcome is that, in order to create this, really unique, first of its kind in the US anyway, data set that would provide broad access to investigators from any institution that's willing to sign a data use agreement and verify that the investigator has human subjects training, is really unprecedented to have something of this magnitude with this broad of an access. And it's really due to Ken and Christine and many others' efforts to overcome some of the regulatory hurdles in order to do this. And I'm really highlighting this because when it comes to connecting other kinds of data, the kinds of data that many of you all are working on, these are the kinds of regulatory hurdles that we together need to overcome even further. So the way it works thus far is that at the top we have institutions that are contributing data. They sign a data transfer agreement with the NIH. They sign an IRB, which can either be their own IRB or a central IRB using Smart IRB and hosted through Johns Hopkins. Those original limited data sets then are provisioned to the NIH. I'm not sure if you can see my arrow, where we will undertake the harmonization process, which I'll talk about in a minute. And then this data is distributed into different data tiers for different types of access. At the bottom, we have data users. And I, again, should mention that in most contexts, clinical data sharing is limited to those who put data in can access the greater data that's been collected. But in this case, we have an incredibly broad access policy where really all you have to do is go through this process in order to access the data. And so we really want to encourage those that are experts in analytics, machine learning, AI, statistics, all kinds of different technologies that may not be normally applied in your own given field to clinical data, to come in and really leverage the clinical data environment and the analytical environment that we have around the data. And so it's a broad call for participation. So data users, they can register and then support the community guiding principles, which I'll talk about in a minute, but really support attribution and good behavior. They fill out a data use request, which attests to the user code of conduct. They will use the data for COVID research, that you will not take screenshots or attempt any data egress. Not that it's so easy to do so in any case, but basically promising to be good citizens. And then that data use request gets approved through a data access committee. Your institution must also sign a data use agreement for you to participate, and in that context, attest that you have human subjects training. Once the data access committee has given you approval, you can access HIPAA safe harbor data, which does not have the geocodes and the date stamps as exact. They've been obfuscated according to safe harbor rules. And we also have aggregate data in that context, which are summary level statistics and the like. If you have a specific project, and in this controlled data tier, you can run tools and algorithms over the patient level data, but you cannot see the patient level data. If you have a project that requires seeing the patient level data, you must have a specialized IRB and have a good reason, basically, in order to access that level of data access. So these tiers kind of provide basically your study design necessitates different tiers, and you get we aim to have you work in the lowest tier possible and only expose the most controlled data in the most controlled ways. And then, as I mentioned earlier, we're also working on having a synthetic derivative. And while we're in the testing phase for that, it's currently still going through the data use request. But eventually, if the pilot is successful, we will make it available via simple registration, and you will not need to go through all the regulatory control. So these are just a summary of those different data types and sort of the descriptions. So I've kind of already mentioned those, but again, the controlled plus data contains dates, full zip codes, and age. This safe harbor data has been stripped of this. Aggregate data is counts and summary statistics, and then the synthetic data is computational data that is a derivative that statistically resembles the original data. And if anybody here is experts in that, we would very much welcome participation in that pilot. So to date, some statistics of how much data do we have. We have 51 different institutions that have signed data transfer agreements with the NIH. This is an unprecedented level of institutional clinical data sharing in the US. 27 IRB protocols have been approved. 24 have both approved. Some institutions take longer on one than the other. 36 have already met with the data acquisition group, and 12 have deposited data in all 4 different models. We have 85% of the Centers for Translational Science Award sites participating in the US, and 229 different organizations with 751 members to date. So it's grown quite quickly, and it's because of all of these institutions and people that it's really been truly possible. So a little bit about attribution. One of the really exciting things about the environment is that because of the security requirements, in particular, the system tracks every single change, every single version, every single user's action. And because of that, it actually supports a really robust attribution model as well. And we had been working on a very simple attribution model that essentially says that an agent, a person, group, or organization makes a contribution that is a role that that person or agent had in the creation of some artifact, which is any data, data quality, tool, terminology, algorithm, or software. And the N3C analytics platform will support this kind of attribution in this model, and the data use agreement will require attribution of all scientific outcomes to everyone that is contributed. So what this means is that we really hope to garner a really robust collaborative environment where everyone who contributes in any way can be attributed, so that we can expedite the sharing of results. And in fact, it's a requirement of the DUA, as well as to really support all the different kinds of roles that one might have. So our example here is Tiffany Callahan is a graduate student. She has a curator role in the creation of a open biomedical ontologies to OMOP mapping file that's going to be used in the N3C, and that file has got her role and her ORCID associated with it. And so anytime anybody uses that file, then in the system, she will be attributed for having curated that file. So on to the phenotype and data acquisition workstream. This is Emily Pfaff at UNC. This is a community designed phenotype. And by phenotype here, I mean essentially inclusion criteria of which patients should be included in the cohort. And so there's a list there on the left of all the different data types that are pulled from once a patient is deemed included. And so, essentially pulling anything that's relevant to any kind of analysis related to COVID. And then the inclusion criteria is all ages. So we have pediatric cases as well. I know there's a strong interest in that in Covert. And essentially, there are positive cases as confirmed by a LOINC code positive results and negative cases as confirmed by a LOINC code negative result. But we also have likely positive and possible positive cases as well. So essentially you have kind of the spectrum of positive cases, negative controls, and everything in between. And then once these phenotypes have been decided by the community, and these are based upon many other communities deciding which clinical cohorts are relevant to COVID research in those different communities. So it's sort of a consensus phenotype. There's a set of community maintained scripts that helps each site pull the data out, those data that are pulled. There's a set of scripts for each model, and the idea is to try to execute those up to 2 times a week. And then there's some data quality assurance processes and a transfer to NIH. And so this is just an example of what the metrics might look like. So for example, there at site 2, we have 2,550 COVID positive patients as publicly reported by the site. When we do the N3C
Dr. Handel, I'm sorry to interrupt.
Yep.
Also, I just want to tell you, we've got about a minute left until we're going to probably need to move to the next segment.
Okay, why don't I just move on to the very end and talk about the challenge to you all. So one of the challenges that we have is that there are way too many different kinds of data being collected in different environments, and a huge lack of interoperability and access from one to another. And in particular, this relates to phenotyping data. So patients might post information on social media, use self-phenotyping apps, web searches. We have our infection monitoring, our wastewater monitoring, case report forms. I've been collecting those there in that link. Then none of this information really makes it into the clinical EHR record. We take the data out of the clinical record and put the clinical record data into the N3C, but much of the other data that a patient might have had collected about them during that time would go to other repositories such as genomic and imaging repositories for both genomic viral sequences as well as host sequences, other kinds of biospecimen and omics data repositories. And it's very difficult to reconnect these data. But the discovery really lays in understanding the longitudinal outcomes for the patient by understanding how do we actually connect these data. So my challenge to you all is to help overcome both the data interoperability issues as well as the regulatory barriers in reconnecting a single person's data over a large number of patients in order to overcome those technical challenges. So just want to say thanks, and sorry I had so many technical problems.
No problem. Thank you very much. Well, in the interest of time, I think I'd like to go ahead and just turn it on back over to Afshin for the last round of lightning talks.
Thanks, Leo, and thanks, Melissa. If there's any question, Melissa, put it in the Q&A. So let's just last round, as Leo said, before we wrap up, covert lightning talks. We're going to have the modeling group, subgroup Kyle Hernandez from University of Chicago. You there? You're going to share your screen. I'll let you take it away. In about 5 minutes. I think you're on mute, Kyle. We don't hear you.
Yeah, it wasn't letting me. All right.
Okay, now we hear you.
You're seeing my screen, right?
Yep.
Okay, great. Okay, yes. So I'm going to give an overview of what we are doing in the modeling subgroup. This group is led by me. I'm at the University of Chicago, and also Sylvain Costas, who is at NASA in the Space Biosciences Research Branch. So this group kind of, our long run goal is to kind of help bring together a lot of the different pieces of data that is, generated by the various groups around Covert, which those groups will be finding, their own signatures and interesting targets. And we hope to kind of help bridge, these groups so that we get kind of interesting things across these different dimensions of data sets with the interdisciplinary analysis and modeling approach, and have some outcomes of like therapeutic targets, intervention impact, biomarker detection, outbreak prediction, those kinds of things. So we'll be highly collaborative. Some of the data that I'm going to start out with that we have been working on currently comes from Sylvain's efforts. So what we have here on the left is starting when the death toll was about 4. Sylvain went and looked at the total number of reported deaths and got doubling time. And in the first few time points, I think about 12, he built a projection based on the first few time points. And so the lines would be that projected trend from the first 12 time points, and then the dots would be the actual observed values. And so he went out a few days, I think about 35 days considered at most. Again, this is at the beginning of the outbreaks, and kind of compared the observed values versus the actual, what the model predicted, kind of getting at, how maybe early measures taken by different countries could have kind of mitigated some of the deaths that we see. You see some very large, potential lives saved from Spain, around 3 million. However, we look at the US and we see really the US started with a pretty slow doubling time of 4.4 days. But really, our action was kind of slow initially, and it seemed to potentially led to a loss of lives, and it is the only country with a negative value in this model. Now, there's a lot that goes into trying to look at these types of data, and one thing that somebody want to look at is some interesting cofactors like how much mobility is happening to non-essential types of places, as well as the density of counties of a county. And so you can see things like the doubling time being slower in low density counties, as well as in counties where a trip to the, these kind of non-essential movement is lower. And with mortality rate, it seems that it's actually higher for low density counties. However, this really may reflect the lack of testing in these counties, only testing kind of critical patients. He also saw that mortality rates clearly increased with counties with increased visits to stores. The gist of this is that you really need to kind of correct for behavior and local policy, population density, all these kind of factors to characterize things like transmissibility and other epidemiological phenomenon. We've really focused on mortality, and a lot of people out there doing various models seem to have a consensus that, probably at least in the US, the death data, death counts are probably more reliable than perhaps case counts. So a lot of people have been building models off of death data. So our next project that is led by Catherine Conley from NASA, also with a lot of help from Ben Steer from Children's Hospital of Philadelphia. She kind of was looking at, metrics to evaluate some of this death data. When we're building models, the quality of the models you build will really depend on the quality of the source data. So in this plot that you see here, it's various select causes of death from CDC. You can see the sources here. And what you can really see over time, and this is all kind of percentage, you really see the impact of COVID-19 deaths right here, around March. You can really see its growth and decline. One thing that is really interesting, though, that she noticed. Yeah.
Sorry to interrupt. Yeah, 1 minute.
Okay, cool. One thing that she noticed here is that the growth of abnormal clinical and laboratory findings was actually growing. Are there COVID deaths categorized here? So what she then did is remove the COVID effect, and you really do see this growth of abnormal clinical and laboratory findings, which is kind of concerning. Perhaps some of these are COVID deaths, so there might be something strange going on with reporting. And we also see that the other causes remain relatively stable. Some other interesting things are from Sam Duda, who also is studying things like viral latent aerosol transport, kind of like Don Melton that we heard earlier. We have some imaging and prediction from imaging for COVID, as well as been doing some large-scale analysis of COVID resources on GitHub. This is our team. We have a diverse team, and we are just now kind of getting into a lot of our research. So thank you.
Thanks, Kyle. If you have questions for the sake of time, Q&A, I think there was some in the chat, Kyle, you might want to address, but thanks. Let's move on to the inflammation subgroup. Michael Topper will be representing them. If you wanted to share your slides and start. Michael?
Yep, I'm doing it now.
Very good. I'll give you a minute warning before we're done.
Sure, sounds great. So I'm Michael Topper. I'm from Johns Hopkins Medicine. I am presenting on behalf of the inflammation subgroup. We are a team across several different institutes, and we meet on a regular basis. So Stephen Balin and Deanne Taylor are our leads, and most of the analysis I'm going to show today, in fact, all of it, is derived from Deanne's work. So I'm just relaying it. Okay. So this is a diagrammatic representation of our current processes of interest. We may move from these, but currently this is what we're looking at, and it's basically differential cytokine production and immune cell recruitment. And we're also really interested in looking at inflammation-mediated cell death and the interaction between these two different processes. So the questions that we have are looking at how variable the inflammatory processes and immune cell recruitment is on a patient-by-patient basis, so the patient heterogeneity. Also, what is the impact of viral load? And what is specific to SARS-CoV-2 relative to other viral infections? So the way that we're addressing this is basically by mining an existing RNA-seq data set. This is the nasopharyngeal swabs provided by the Mason Lab, and it's total RNA-seq data. And on the left, you can see a volcano plot that's actually from their bioRxiv paper. And this is looking at SARS-CoV-2 positive samples relative to negative. And you can see here quite clearly that there is a marked induction of interferon-related genes, as well as Th1/2 type chemokines. I guess that's not incredibly surprising, but it's good to sort of establish a baseline for the data set that we're looking at. And downregulated, they note some ribosomal-related genes. And in agreement with these data, they actually saw an enrichment of pathways that are predominantly viral and immune-related, which can be seen here on the right. So our aim actually is to evaluate adaptive versus innate immune signaling as a result of differential CoV-2 status, that being high versus low, and also patients that are negative versus positive, as well as comparing SARS-CoV-2 versus other viral infections and its intersection with cell death mechanisms. So I'm going to just present a couple of vignettes of what's been done from the analysis side of things. And a good place to start would be looking at the DC2 differential gene expression, where a comparison between high SARS-CoV-2 versus none was conducted. And you should leap right out of you right away that this sort of recapitulates a lot that what was shown in the Mason data set. So you can see here that double-stranded RNA sensors, which are known to initiate antiviral responses, are induced. as well as a panoply of different interferon-related genes, Th1/2 type chemokines, interestingly, a monocyte neutrophil attracting chemokine, CCL8. And downregulated, actually some interesting candidates emerged. Hemoglobin-related genes, which could be linked to the complement-mediated hemolysis that's seen in COVID patients, and also mitochondrial localized gene that's been associated with thrombosis when there's an inactivating mutation. So these data are interesting, and they are definitely in agreement with what's been shown by the Mason lab. So the next question we had was, how does the transcriptional response vary as a function of SARS-CoV-2 sequencing counts? And once again, it's important to note that this analysis was done by Dean Taylor. So this analysis actually is a DC2 gradient analysis, and it looks at differential gene expression versus increasing SARS-CoV sequencing counts on a sample by sample basis. And what kind of emerges from this is that it's clear that there's interferon genes, antiviral sensors, and innate immunity track with increasing SARS-CoV-2 sequencing counts. It's also important to note that this gradient analysis is preliminary and still ongoing, but it sort of agrees with the previous data looking at SARS-CoV-2 high versus none. And so the next question we had was, how does high SARS-CoV-2 transcriptional responses compare to more general viral responses? And to get at that, we did a DC2 differential expression analysis where high SARS-CoV-2 was compared with other viral, and this is a DC2 analysis. So what emerges from these data is that interferon and pathogen sensors are induced, which I guess isn't so surprising. And what's interesting is that actually class 2 MHC, B cell, and T cell related genes are part of the downregulated, so they're actually lower in high SARS-CoV-2 relative to other viral infection. And our hemoglobin genes and also the thrombosis related genes are downregulated in this data set. And then interestingly, a link RNA that's been shown to be differentially expressed by metformin and PBMCs is also downregulated, but the functionality, I do not know. So our future directions are basically to establish functional roles for SARS-CoV-2 associated differentially expressed genes. What are they actually doing as opposed to just making observations? And then to apply findings derived from RNA-seq to the analysis to the design of a serum-based biomarker assay. So we've already started on this. And then of course, we would love to apply therapeutic discoveries. We would love to make therapeutic discoveries based on inflammation profiles. And that's the end of my presentation. So if you guys have any questions, let me know.
Thanks, Michael. We do have time for one quick question, but I don't see any. So if you have any questions for Michael, put it in the Q&A and he could directly address it. So let's go on to our last, thanks, Michael. So let's go on to our last subgroup lightning talk by Sonia Villapol from Houston Methodist. She's going to talk about neurology type symptoms of COVID-19 and their work. Sonia, I think oh yeah, we can hear you now. Perfect.
Yeah. Hello. Yeah, hello. So I'm going to present like a short introduction or subgroup. This is the neurology subgroup. These are the members of the group in COVID, and we're working basically in try to establish the links between the COVID-19 in association with the brain and with the central nervous system, and who these neurological symptoms can be affected on the COVID-19 patients. There are a few data so far, but this is relevant, is important, and we know several kits that are important for that. So we know that half of more than half those hospitalized patients, they develop neurological symptoms. So that was demonstrated that the entrance of the virus into the brain is through the different pathways, and probably the viral entrance through the olfactory bulbs there is the most relevant, but also the coronavirus could maybe reshare the cerebral vasculature through the general circulation, because can bind the ACE2 receptors in the vessels. So there are several issues here to explore, but definitely there are brain damage and brain injury, and that also affects the immune response. So the inflammation and the cytokine storm can cause neurological deficits. So the symptoms, there are not specific to the brain, like muscle pain or headaches or dizziness, but they are related with neurological affectation. But the more severe symptoms, for example, there are in the first stage of the disease, there are a loss of taste or the smell that was shown in several reports, and it's around the 5-6% of the cases. Stroke, the ischemic stroke, hemorrhagic stroke. So there are is related to the hypercoagulability that is increased, so can affect the thrombosis and can generate multiple damage in these patients. You can see this imaging here of the small vessel with the thrombus into the brain. Another disease that were observed, they are rare, that is the Guillain-Barré syndrome that affects the immune system and that can damage the nerves. That is, there are a few cases reported in encephalitis as well. That is caused through the acute inflammation to the brain. And you can see here in this neuroimaging picture that corresponds to the woman 57 years old, that they are inflamed and they show this encephalitis into the brain. So that was associated, that occurs after a stroke and was associated with the infection, with the disease. Another disorders like the delirium, and that is the most common in older patients that there are in the UC, and there are really severe cases. So the common types of the coronavirus infections with potential central nervous disease and injury were pretty similar to SARS and MERS. There are stroke was common, and another neuroinflammatory disease related to the brain. But it's important to take a home message that all these consequences, all these neurological manifestations that we are looking now and we are observing now in the initial reports, maybe have a long-term medical consequence, and that is very relevant, and it's important to the incidence and the development neurodegenerative disease, as can be Parkinson or Alzheimer's later. So we need to target this and try to explore a little bit more what is happening, we can stop it. And finally, briefly, I want to say that the brain and the gut is connected, and we in our subgroup have a study to analyze this connection with the inflammation, how the brain can connect with the gut, and as well with related with the gut microbiome dysbiosis and the early phase of the COVID disease. So we can stop or reduce the inflammation at this level, and then we can probably reduce the severity of disease later. And that is related with the with the enteric system and all this neuroinflammatory cascade that occurs in the gut in response to inflammation. That is the physiology of this disease. So definitely analyzing the gut microbiome and modulating the diet and using probiotics or fecal transplants could be another tool useful to improve the neurological dysfunctions. So in my lab, for example, we have this clinical research that we're collecting data, and our group is going to analyze later, and our main goal is analyze if the gut microbiome could be an indicator of the susceptibility or the severity of the COVID-19. Is the major goal, that we can analyze that in the early phases of the disease. And that's it. Thank you so much, and thanks for the opportunity to present our group here.
Thanks, Sonia. I think we don't have time for questions, so yes, 5:20. So if you have any questions for Sonia, please put it in the Q&A or chat. Yeah. So thanks. That was a good way to wrap up all the lightning talks on Covert. I think we'll give it now to either Todd or Leo to introduce the Provost Reginald DeRoches for closing remarks.
Yep, Dr. DeRoche, I don't know if you have any slides you want to share or anything like that, or if you just want to just give a talk.
But
okay.
Honestly, I'm impressed that you guys are exactly on time. That
is. So for the closing remarks today, we have Dr. Reginald DeRoche, who is Rice University's provost and serves as professor of civil and environmental engineering and mechanical engineering. As the university's chief academic officer, Dr. DeRoche works closely with the president to advance the university's teaching, research, and service mission. He previously served as the William and Stephanie Sick Dean of Engineering at the George R. Brown School of Engineering at Rice University. His primary research interests are in design of resilient infrastructure systems under extreme loads and the application of smart materials. In 2020, he was elected a member of the National Academy of Engineering. Welcome, Dr. DeRoche, and thanks for agreeing to help us close this out.
Great. Thank you, Leo. Can you hear me okay?
Yeah.
Great. So it's an honor to provide a few remarks at the closing remarks at the second Coronavirus International Research Team Symposium. The symposium brought together an exceptional group of scientists and engineers to speak on a range of topics, from vaccine and therapeutics to using wastewater to understand the regional impacts of COVID-19. I want to thank the Ken Kennedy Institute for their leadership, and specifically thank the organizers, Afshin and Todd, for their hard work in pulling together such an exceptional group of speakers. While we regret not being together in person, the benefits of this format is that it allows us to bring people from all over the world to learn and to collaborate in a meaningful way. COVID-19 may very well be the greatest challenge of our generation, and it will take bringing together the best and the brightest from all sectors of the community to address the challenges. Shortly after the outbreak, Rice University announced a competition to fund a series of projects that can have an impact on our understanding of the disease and our ability to address it. We funded nearly 20 research projects across 6 different schools at Rice in a range of topics, including one of the projects discussed today on wastewater monitoring, which was a seed project, which ended up being also supported by the city of Houston, by Dr. Lauren Stadler. The diversity of the projects ranged from understanding the steps that Harris County can take to make voting safer in the face of COVID-19, to working with the Houston Symphony to investigate proper social distancing protocols for rehearsal and performances by musicians and singers. We have a team that's studying the airflow created by wind instruments and singers using high-speed imaging. Despite all the challenges that have resulted from COVID-19, I think we're seeing 2 good and hopefully lasting changes in our professions as scientists and engineers. First, we're seeing a greater respect for the role of science and technology in addressing the greatest challenges. Unfortunately, this is not in all segments of society, but certainly in many, we're seeing how people realize the importance of science and in addressing the nation's challenges. Hopefully, this will result in many more young children pursuing careers in science and engineering. And second, you're seeing some truly unique partnerships develop across private industry, academia, national labs, medical clinicians, and others. Even the interdisciplinary nature of our work has been quite unique. We've seen, again, musicians working with electrical engineers, social scientists working with computer scientists, and much more. And I hope this, again, will also be a lasting change in the world of science and engineering. So finally, I want to once again thank all of you for your participation, and thank the organizers for their leadership. And I hope there'll be soon a third symposium in this space. And so have a good and a safe day. And thank you for allowing me to say a few words.
Thanks so much, Provost DeRoche and Reggie. Wonderful to have you here. It's really great to pull this together. And again, this is thanks to a lot of people, but really, really nice to see our local response here in Houston at Rice. But then, like you said, across the globe, which has been, just the participants we had today, it's been, amazing. 20 countries spanning, 34. I mean, just really, really unbelievable. So thanks to everyone who attended today and participated and contributed Q&A questions, and really appreciate you giving some closing comments. So thanks to you, and thanks to everyone.
Thank you, John.
Leo, I guess maybe pass it back to you.
Sure. So we're wrapping up here, everyone. Thank you for joining today. I'll give just a few final comments. As far as slides, which a lot of people have been asking about the availability of the slides, these can be sent out pending speaker approval. But what I would just personally recommend for everyone is that there should be a recording of the whole symposium available immediately on Facebook after we end. So if you want to go back and reference any particular slides right away, you always have that as an option. Just search up Covert or the COVID-19 symposium on Facebook. You should be able to find it either through that route or otherwise. We also have a Covert website, and there's other social media platforms for Covert like Twitter. And the only other things I have to say other than that is just thank you to people. So for instance, thank you again to the Provost DeRoche. Thank you very much to the Ken Kennedy Institute, to Angela Wilkins, the Executive Director of the Ken Kennedy Institute. Thank you to Rice University, obviously, for sponsoring this event. Thank you, Nick Sapovell, Hugh Ogilvie, and Kristen Curry for actually helping me field questions during the event. Thank you very much, guys. And thank you for the huge number of people who helped make this such a successful event, this and the last one, which unfortunately would be a bit too long of a list for me to name everyone explicitly, but thank you very much, everyone. And lastly, I'll just give my personal thanks to Afshin and Todd one more time. Afshin, thank you for very much for co-organizing, promoting the event, helping organize and run the lightning talks and all the other things you've helped with. And for Todd, what can I say here? He's been my mentor for the last year. I can't believe how much I've already learned in that time. So thank you very much for that. Thank you for consistently putting me in just great positions and giving me amazing opportunities. For instance, like being involved in these. Thank you very much, and for co-organizing the symposiums. And I'll just add a final thank you, which is just to say thank you for just being just a kind, nice person. That's my personal touch there, just being an actual nice person. I appreciate that very much. So thank you very much to everyone. And unless anyone else has anything else to say, that'll be the end.
Yeah, well, Leo, that was very flattering. Thank you. Well, we have to thank you as well. So I will virtually applaud you for excellent, as our provost said, hitting the mark through a long day is very difficult to do. So thanks for great moderation and providing questions and all that. So awesome job again. And overall, really, really happy with how everything went. Of course, Ashvin, I want to pass it back to you if you have any final comments, of course, before we close it out.
Yeah, no, I think you guys covered it. And yeah, special thanks to Leo, of course, and all the speakers and Covert for actually making it happen. Because without Covert, we can't be doing this, all the members in Covert. So, and of course, you, Todd. So thanks to everyone. All right. We'll have another symposium probably in 3-4 months, right? So keep an eye out for our emails and our badgering of you to attend.
Yeah. Thanks, guys. Thanks, everyone.